So we are going to start the meeting. A few people still to arrive, but we should start, and then they can chip in whenever they arrive. Thank you very much all for coming, especially those that come from abroad and abroader. And it's a pleasure to be here. And well, we are gonna a few things. You need internet. It was there. So the internet. It changed the screen. Now it is back. That's the internet data. Okay. A few things. The bathroom is right in front of this room. We'll go for lunch at 1. The people that told me that they were coming for lunch, all the speakers are invited, and everyone else that said in advance that were coming, they are also coming. And I think this is everything for now. So I'm going to actually introduce the meeting. I'm going to introduce the meeting using my talk. And I'm going to introduce the speakers just through my talk. Okay. Okay. My talk has 5 parts, and I will include the presentations of all the talks within my own talk. And first, I'm going to talk about natural enemy spillover effects, then predator spillover effects driven by water in deciduous forests, the use of computer simulations to test predatory spillover effects. I'm going to introduce our framework, Weaver. And then I'll talk about the applied side. One of the main reasons we are here is natural enemy spillover effects within greenhouses and among greenhouses. And then I'll field experiment that we are doing about predatory spillover effects from resource islands. Okay, so we know that nutrients, litter, and organisms move across ecosystems and habitat hedges, and that this creates population dynamics, communities, and ecosystem processes. Affects that. And we have a view on predators in my lab, and we know predators are beautiful, and we like them because they are close to us. And I would say that this is also beautiful for an arachnologist. And we have this view, and we know that predators are also very important in ecosystems. When predators are not there, this review paper shows that there are several ecosystem processes that are change, like disease dynamics, wildfires, carbon sequestration, invasive species, and biogeochemical cycles. I want to recognize the figure of Gary Polles, who's first author in this paper that I just mentioned at the beginning, because he was an arachnologist and an ecologist. As a matter of fact, in his webpage, is listed as an arachnologist, not as an ecologist. Even his great contributions to ecology, his famous books, The Biology of Scorpions, which is still a main book in scorpions. And I talk it about arachnology because we do arachnology, but remarkably this book that was published after he died, and which talk about the subject exactly that we're talking about here, right? Food webs at the landscape level. And several papers, a couple of contributions here, and last senior author in this paper is Professor Holt, who is here in the audience and will give today the talk, The Ecological Effects of Special Subsidies and Temporal Pulses, a Theoretical Perspective. Bob Holt has worked on apparent competition. He coined apparent competition, apparent mutualism, extensive work in omnivory and intraguial predation, meta-communities, phenotypic plasticity, and you name it. This recent book with Andrew Thompson and David Tillman has short papers, short chapters by several top ecologists, and it addresses topics, interesting topics that are short and easy to read, so it's highly recommended. Okay, when we talk about predators, we are focusing on the cross-habitat spillover hypothesis of Czarnecki et al. In 2012, which states that landscape-moderated spillover of energy, resources, and organisms across habitats, including between managed and natural ecosystems, influences landscape-wide community structure and associated processes. And it's a pleasure also to introduce Jason Tilianakis from the University of Canterbury in New Zealand. He just arrived last night from New Zealand, who's going to present his talk on consumer spillover and the spatial propagation of habitat effects. Jason is second author in the paper of Czarnecki et al. Of 2012, and he has done extensive work on this and using also ecological networks. The reason that, this symposium is called Unnatural Enemies and not just predators is because they are the parasitoids too, right? Jason works with parasitoids, and they are also important in this top-down control. Okay. He has done experiments at very large scales that he will talk about it later. Okay, so when we talk about the cross-habitat spillover hypothesis for predators, predators may have effects for two reasons. One is dispersal, when they go from one ecosystem to another, or one habitat to another, and they move and they never return. And then foraging, where they may enter a different habitat, forage here, and come back, like in central place-loading predators. Okay, and predators can also have propagated effects without even moving from their own ecosystem. This is a very nice paper in 2005 in which that shows that fish feeding on odonate larvae, on dragonflies, affect the adults and indirectly positively affect pollinators and pollination. So this, without moving, effects propagate to terrestrial ecosystems. And I want to show here the scale of experiments that we can do. And I want to claim that water dynamics is a driver of predator-prey interactions. This is obvious in the savannas, right, where you have these concentrations of animals going where the water is, especially where drought is on. And you see that this may affect when predators are around, you have predation risk. But what's the potential of predator-prey interactions in these aggregations? We don't know. We cannot do experiments with this. And actually, the scale of cross-habitat foraging can go from long-distance migration to foraging among micropatches. And this, I would claim, and it's obvious, right, that the scale of these animals working is negatively correlated with experimental feasibility, field. To do field experiments, it's much easier with the smaller animals in macropatches than it is with very large animals that have effects, at least if you want to manipulate the actual animals. And I claim that this can be done in the forest. This is an image of the deciduous forest, the leaf litter food webs. So we have predators, large predators, small predators, the fungivores, mostly fungivores in the litter, and the basal resource that is usually fungus. And in a forest, what we have is areas where litter and water accumulates and areas that are drier. And if we see this paper from Shomi et al., we see that through time, the spatial distribution of water changes. So does that change the distribution of animals too? So what we did is to measure two microsites, some that are more or less on slopes, this is just in principle, and some that are in accumulation areas where after a rain, water, energy, and nutrients accumulate. And when this dries up, this becomes dry and poor, and this is a wet-rich area where water is maintained for longer periods of time, even in a deciduous forest. When we look at the abundances of animals here, we saw that indeed this rich, with more water, patches have more animals in volumetric density. And we can ask, does litter fauna move horizontally towards these wet and rich patches as water dries up, or what we are seeing is just local numerical responses to higher productivity from water and nutrients? And if they move, indeed, if fauna moves, what are the predator-prey interaction consequences of such aggregations? And the reason we do experiments is no doubt that of David Weiss. David Weiss is currently emeritus professor at the University of Illinois at Chicago. He was my mentor during my PhD, and also I did the first postdocs with him. So it's a pleasure to have him here today. And he's also an ecologist and an arachnologist. He wrote this very influential book in 1993, which, if you want to do experimental ecology, whether you are into spiders or not, is very worth reading it even nowadays. Even the statistics are from 1993. It is very relevant to understand many things that we do in experiments. It's a lesson of ecology, of experimental field ecology. And he's going to present a talk. It's called Two Different Approaches to Uncovering Patterns of Intrahill Predation in Forest Floor Food Webs. And as you can see, with this molecular, this molecular solve food webs, he's still into spiders in ecological webs, because these are mostly spiders. Well, what we did here to prove this, to show that what happens, to test what happens when animals move due to water, is to a four-patch bifactorial experiment in which we had water and fungi combined bifactorially and test where the animals would go. And what we found is there are additive effects, so animals would mostly go, the litter fauna would mostly go where more water and more fungi were available, with additive effects. And when we look at the path analysis, what we see is that prey, fungivores, respond directly to water and fungi, and predators mostly respond indirectly through responding to prey. At the end, what we see is that the number of predators to prey ratio is much higher in water plots than in drier plots that were experimentally created in the field. So, as a consequence, it looks like prey may have higher predation risk here because the ratio of predators to prey is higher. And what we did is we took the litter from these patches and removed the predators in half of these patches, in half of them, and the other half remained the predators. So this is a beach forest soil food web by Digel et al. It's not ours. And what we did basically is take all these predators out of the bags, and in half of the bags include them. And I'm talking about bags because what we wanted to know is the instant effect of this finding each other in these areas. What are the instant consequences of being there? And what we found is that prey mortality was higher, not lower, when predators were removed, which was unexpected. But what we found is that actually predators have higher mortality than prey, because they were there in higher abundances relative to prey, and that actually predators have higher mortality in places where there is more water, where they have aggregated. So water, spatiotemporal water dynamics drives biotic interactions, and removing predators increase prey mortality, perhaps from competition, perhaps for increased predation from small mite predators. Predator mortality was higher than that of prey and increased in water patches. And the question is that maybe they are ecological traps from IGP. They concentrate there and there is interspecific predation. So the predators are actually going after the prey, but they got then a higher probability of being killed, higher predation risk. And this experiment is also good to introduce our modeling background, modeling framework, And for that, I'm going to talk first about this next generation. Now everything is called next generation something. Next generation ecology and next generation individual-based models. So a next generation individual-based model is nothing but an individual-based model in which we in silico follow the fate of individuals that do something, mixed with other modeling approaches such as differential equations or difference equations. And highly related to that is next generation ecology, in which pattern-oriented modeling is the main framework. Pattern-oriented modeling means that if you have a pattern, try to test different mechanisms, different submodels that leads to that pattern that we observe in nature. If we come with the right sort of submodels that explain this pattern, we can talk about structural realism, so what we are mimicking is really close to what happens in nature. And a robustness analysis is when you try to fit different submodels, different mechanisms that explain this pattern, and you achieve structural realism. So in addition to sensitivity analysis, in which we have to compare the values of the parameters, different values of parameters, you have to test different models, different submodels, and different mechanisms. Weber is one of these models. It has realistic genetic phenotypic mapping using quantitative genetics and multiple genetic trade-offs. And this is one example from 2012, when we came with all this framework of genetic framework, in which we can have modularity. Different traits have different amounts of genetic correlation. Of course, this can be realistic if we have the data, or we can make assumptions and put these correlations and this variation, genetic variation. And other models can affect each other via epistasis and sort of phenotypic plasticity, for instance, for the response to temperature, right, from this via. And this is good for introducing talk by Carlos Melian. Carlos Melian is a theoretical ecologist with whom we have been collaborating for quite some time now. And his talk is going to call Trait Architecture and Coexistence in Especially Explicit Food Webs. And this framework reminds a little bit of what I showed you before, but I think that one of the main differences is that in this framework, he'll explain it, there is an explicit association between the traits and fitness, whereas in Weber, what we look at is more heuristic. We look at emergences of fitness from the traits. It's a bit different, but we can discuss this as we go in the meeting. Okay, then we have in the metabolic theory of ecology for developmental time, for metabolic rates, for thermal performance, the ecology of fear, so animals avoid predators and try to avoid predators, and there are ecological and evolutionary consequences of that. And then there are interaction outcomes, and the outcomes depend on the traits. And then we can have different food web configurations linking this to this. And since one of the traits is mobility, we can make mobility depend on the traits, is more accurate. And we can actually test this in space, and these are now a representation, graphical representation of these litter water pockets that we show in the field, that they are there and they have more fauna, and whether we were interested whether fauna concentrates here and what are the consequences of that. We can test that. An addition that we put in Weber now is that we have temperature and water time series that are especially explicit. So in each cell of the simulation, in each patch of the simulation, we can have different time series of temperature and relative humidity, which makes it more realistic with the data we could have from out there. Okay, why Weber? Because it's strange why someone would come with such a difficult, complex thing when answers can get more easily. Well, I have some eco-evolutionary questions in which I'm not going to get into detail, but for instance, temperature and water leads to diversification from biotic interactions. This is one of the hypotheses, and this framework is now ready to test this sort of hypothesis. Here, one of the claims is that every interaction in the life of individual matters, and this links nicely with the talk of Jose Maria Gomez from our institution, which is going to call modeling ecological interactions, a Heraclitean view. We have in common that we think that every interaction in the life of an individual matters, and his talk is going to address this nice thing, that interactions between species pairs are flexible in sign. Jose Maria Gomez has mainly experience in pollinator networks, pollinator plant interactions, and in dispersal animal plant interactions. Okay. And a second thing why Weaver exists is this hypothesis that was at the time was a little bit perhaps naive, but it's the hypothesis that higher amounts of genetic variation in the species in food webs would increase persistence. And we do a little test here, so Weaver can compare webs with different conformation, different structure, different degrees of connectance, well, higher connectance here, and different spatial setups where these islands are more or less separated in space, which is what happens really in nature, in the soil. We have these water pockets all over the place. And this is a good point also to introduce the talk by Oscar Godoy, Non-Random Interaction Shaped Species Coexistence in Multitrophic Communities. He's going to talk about communities that have herbivores, plants, and pollinators. And he's going to actually connect feasibility domains, the probability of coexistence, with connectance and other network metrics. I'm going to talk also about connectance. Here, when we test the hypothesis that genetic variation in 13 traits improve the persistence of the web, of course, this is very, very fuzzy. These are just transient dynamics. But it's still good to see that when you have more variation in the traits, this can stay for longer, and even though the extinction risk is high. But this will only happen when you have high connectance in the web and at intermediate island distances. What we think is happening here is predator-spatial coupling, which was already described by Holt in 1984, an American naturalist. So these next-generation individual-based models are very complex. They have lots of parameters, so they are dangerous in a way because you don't know if you're doing is accurate enough, or you can drift away from what happens in nature. But these others say that yes, we can, and yes, we must. We didn't know we were doing this sort of models. It was called like that when we were doing what we were doing. So the release of this paper was nice because we saw that some other people were doing these sort of things, especially these people that are the main promoters of individual-based models. And Weaver is listed there, and it has lots of features that are, lots of functionalities that are desirable in a model of this kind. There are some things that are still not there. We are working on all of them, but I'm going to just focus on two of them now. One is the microsite. We work in the forest just in microsites. This has spillover now to other systems. You started with beech forest, and of course we cannot parameterize every single species that is in the beech forest. That's not possible. But what we can parameterize is what Grimm and Berger call functional types. So different types of animals that are there, and build a simplified food web that we hope it works similarly as what a real food web works like. This is work of parameterization done by Dolores Ruiz-Luprión. During her PhD, she did excellent work on looking at the literature and using the scripts and parameterizing this entire thing. And this is now ready for simulations. One new feature that we have included here is to include ontogenetic growth. The reason for that, the main reason, there are many reasons, but the main reason is that if we relate only on time to decide when an animal would become adult, just based on what the metabolic theory would rule, we were having animals that would develop too fast, and they didn't have adult sizes as we see in nature. Using real curves, either from the lab or from the field, we can now actually have a double target and mimic the growth of animals much more accurately. And how Weaver works, we make an energetic mass length assumption. So using Peter's equivalence of 7 joules to 1 milligram of animal body mass, we know how much energy these animals spend per half the energy power. And then we use the mass-length relationships, allometric equations, which Dolores Ruiz-Upion, during her PhD, did among a comprehensive database based on most soil arthropods, and it is now published in this paper. So we have enough information to go from here to here. And of course, we now go from length to mass to energy. Let's focus on here. In addition of that, we can use these growth curves to have targets for how much food an animal needs every day. We can target, an animal would need a food slightly bigger because it has assimilation efficiencies, it has losses from respiration, metabolism. Slightly higher, we know that the energy that an animal needs every day, we have these targets, is slightly higher than what the curve that we know is published. So that with these targets, we also set limits to these targets. So plasticity is there, if there is more food or less food. Below this limit, the animal would die from starvation, and above this limit, it's not possible to feed anymore, so it has satiation effects. And this plasticity can be different among individuals too. Okay, and another feature is indeterminate growth. Indeterminate growth means that the animals can start reproducing before they reach maximum size. Okay, and a spillover, I was talking about the spillover effects of Weaver. We know that in dinosaurs there are lots of growth curves published because they have this histology similar like rings in trees where they can know the years and the life of this individual and turn it into a growth curve of that individual. So we have dinosaur growth ontogenetic curves, and we talked about this like 10 years ago with my high school teacher in biology and also in sports, and we have now ready food webs with dinosaurs that follow exactly the same rules in Weaver, and it works. So we have increased in 10 orders of magnitude this framework. And another spillover is a postdoc that is in our team now. This is Encarnacion Fenoy, who's sitting in the audience, who is now started parameterizing the food webs and of headwater streams that are subsidized from litter from terrestrial ecosystems. Also in the audience is Paula Lillo. She's going to talk about parameterizing soil nematodes in food webs, which is within this framework. With her PhD advisor, we've been working and talking about this for several years now. And we are going to include the nematodes. So we have the arthropods and the subweb of nematodes below that, and also the subweb of nematodes, because nematodes in themselves, they have predators, they have fungivores, they have bacterivores, they have omnivores. And she's going to talk today about the progress she's making with parameterizing bacterivore nematodes. They are almost ready to be run in Weaver. Okay. Basal resources have also been improved in Weaver. Now, Rmax depends on the metabolic theory, so basically on these equations by Savage et al. in AMNAT in 2004. With that, we have Rmax, and then we can have Rmax to have optimal maximum growth rate depending on relative humidity, and have different relative humidity niches for the basal resources. For one thing, bacteria grows at a much higher relative humidity than fungi. So we can sort of mimic that and assume that different basal resources have different optima. With this framework, we also include plant growth. Okay, newer things that we have included, we have diploid sexual reproduction and also haplodiploid sexual reproduction and asexual reproduction. This is new. Genetic maternal effects, so we can see the evolution of egg size. Resource preferences and profitabilities, which weren't there before, and consumer memory. So now consumers realize what they have eaten, so they can assess relative abundances and switch their preferences, typical predator switching, which increases probably the probability of coexistence. And in the metabolic theory of ecology, we have now gone to the right of the curve, to the decrease of the performance curve. And this is a big deal because before we had only this positive relationship in, and it makes a big difference to have all that in. And then we have included omnivorous insulato, which means that feeding on individuals and on basal resources. So on individuals at the individual level in the simulation and on these basal resources or alternative resources that grow according to recursive equations, as we have seen before. Okay, and now I'm going to show the patch algorithm because we have improved it too. Here, where patch to go, the animals is decided upon biomass of food and predation risk negatively. And with this, we have a parable that animals can actually have an optimal in abundance of conspecifics. So this term is about abundance of conspecifics. And this, all the parameter weights whether conspecific is more important or predator and food is more important. And this is a small parameter that has a very tiny random number. So if these two terms are 0, animals would move randomly. And this is a couple of examples. I have more. Odin has been recording more, but this is just a simple example in which here the predator and the prey will just follow each other. We have one here, and then the abundance, the biomass of conspecifics. And when we do that, now I'm going to put a video. What we see here is not a lot, meaning that you see the predators following each other, but they are not very successful at killing the prey, of course, because they are behaving not adaptively, right? They are just moving, following each other without caring what there is out there. Then we can have a second, which is this is not the second is this one, in which we have now here that predators move avoiding each other, is this parameter here, right? Like sort of territoriality, they avoid each other. And when I put this, what we find is that prey has much mortality. Even prey aggregate to each other. Prey still aggregates, predators are killing the prey. Okay. What you see the different colors in green is actually the value of the trophic cascade. So eventually you see that all the color is darker blue because most of prey has been killed. Just a small demo, that's nothing, just to show this algorithm. And highly related to this is David Garcia Callejas, whose very nice work I didn't know, and who is very related to this. He's currently postdoc in Jason Stylianakis' lab. He's going to go about signatures of spatial cascades mediated by dispersal and foraging in trophic meta-communities. David is a modeler, and he uses a strong mathematical background in his research. Okay, the second thing that Graham et al. Put it as potential here, it says that can explicitly incorporate human management actions. That's Weaver can potentially do that, and that's the main spillover of Weaver that brings us here. And now I'm going to start talking about Almeria and the sea of plastic. It's sort of southwestern Almeria, but this was a natural landscape that was a natural ecosystem of shrubs that are now endangered. And this has been changed over 40, 50 years like this. They say, I'm not sure, that you can see this from special, from special stations, space stations. But the thing is that in 2007, this biological control became important and mandatory because if you don't use this sort of control, it's unhealthy to people and it's not good in the market, fortunately. And here are the two heroes. The main reason we are here, because these ideas of spillover that, in my mind, they came, of course, by knowing your work, but because they were actually applying this here in Almería. They are responsible for making Almería green again. Stefania Rodriguez, who is going to talk about vegetation management and biological control in greenhouse crops. And Monica Gonzalez, who is going to talk about using functional biodiversity as a new tool in IPM programs. And this is their work. So we can see here these hedgerows that can actually prevent pests from going from one greenhouse to another. That's the idea. And of course, here we can talk about these propagation effects of predators, right? Because predators, in principle, they stay here and they will avoid pests from going from one greenhouse to another, so their effects are propagated through space without moving from the actual habitat. And these plants, or sort of these plants and other plants and native plants, are used inside greenhouses, and you can see greenhouses that are quite green with all these plants. The idea is that there will be predators inside that will have spillover effects all over the place in the greenhouse. Here lives a lizard. I put this a big lizard because it's, for us, a lagarto is a big lizard. It's a particular species of lizards that are quite big. And well, I don't want to stay on their talk anymore. I did enough. They will explain all these things. So if we seek for efficient BPC from natural enemy spillover, this is one thing that I put, is everything that could occur to us on this, right? So, but this to start fuel discussion. What plant size, abundance, diversity, and distribution, and what associated predator traits and food web structure can provide the best pest control in different crops, pests, and under which abiotic conditions. And because we know that there is rapid evolution in these animals that are usually evolved, what is the potential role of evolution and ecoevolutionary dynamics when we do all this? And related to this is the talk of my dear friend and colleague, Marta Montserrat, who is going to talk about a framework that we put together that is food web engineering, building community models to improve biological pest control. She's going to address how to use community models and evolutionary experiments to try to get strains that are more efficient at biological pest control. This is also linked to this IBM's framework, so that's why I said it was a spillover, because we are now parameterizing these webs and trying to see the applied side of this framework. And nicely, a recent call for the European Union. We got this grant, Food Web Engineering, Ecological Systems Biology for Biological Pest Control, from the program of Recuperación, Transición y Resiliencia of the Spanish Agencia de Investigación. And within the same program, but in the Junta de Andalucía, we got a computer scientist, a young computer scientist in the program Investigo, who is actually improving very fast all the functionalities of Weaver. Odi Moron is a postdoc that just joined us in the lab, and he's sitting in the audience and who prepared the videos that we are seeing. And many people in the audience here, of course. And this is a nice point to introduce the talk by Ines Fragata. A specific sequence of arrival promotes coexistence via a special niche preemption by the weak competitor. So here we have the spatial component of Tetranicus, which is a pest, knowing how competition, how species of Tetranicus compete is important for theoretical ecology itself, and for experimental ecology, but also it has implications for control. So here everything fits. And of course, I always ashamed to talk about all this. I'm putting all this together, and I have these books, but I read just parts of them which are overwhelming. People that are really working consciously and very meticulously in all these things. And of course, we use something that we use many parameters and many things that are realistic, but at the same time are not as mathematically oriented as all these frameworks. So actually, I was a little bit stressed out about preparing this talk because I was realizing very fast how much I didn't know. But of course, I cannot pretend to know everything. So we just step by step. And the nice thing is that I have the opportunity to now, using Weaver, learn about all this. I especially like this book of Mark McPeak, Coexistence in Ecology, because it builds webs step by step, and we can do that with Weaver too. His is a mechanistic perspective. I guess ours, we can actually see all this at once, so I guess it could be called a hyper-mechanistic perspective. Whether we are getting too far or not is something we can discuss. And of course, we are doing a field experiment in this grant. In this field experiment, we have resource islands, which are dwarf palms, in which we are manipulating predator densities and seeing the effect of these predators outside, in the matrix outside. So spillover effects from predators. And we are also using these roofs to see the effects of precipitation, lack of precipitation, from climate change in all these effects. These are the traps that we use with different layers and different mesh sizes to prevent predation or to minimize predation in the trap. And here is Eva de Mas, my dear wife and technician, who is actually building the traps. She built them themselves. And here we have some of the animals, the pictures that she's taking of every animal, so they're being ID'd by experts, and then we can actually try to ID them in the field. And of course, we have all this diversity. This is just a very small part of the diversity that we have. But the point I want to make here is that when we remove all these predators, as David Weiss knows very well, the effects that we get, or the answers that we get, can be any. And then we have to figure out what mechanisms led to these final answers that you have. David knows a lot about this. We are hoping that we can actually use Weaver with this to try to get what is going on in these experiments, in any other experiments. And of course, this is something that we see in the traps. We see predator-prey interactions. We measure the animals, and we don't see many, but the ones that we see are very valuable because this is very difficult to see in the field. These are those two nocturnal animals. But of course, you can go with a headlamp and with a UV light at night and do thousands of visits. And this is what actually Gary Pollis did to actually start a revolution in food web theory, right? Because he was seeing that spiders, scorpions, they were killing each other, and there was a lot of omnivory, a lot of intragill predation, his later coined, in natural food webs, which were not being considered, and the model said that omnivory couldn't be common in nature because it was destabilizing. So this made an important point in change. And when we see these interactions in the field, remind a lot about this work. Well, I have some points for discussion I'm not going to enter into. If there is 5 minutes, yes, there is 5 minutes. I'm going to put a couple of videos of the greenhouses that we just prepared, of artificial greenhouses, of course. This is something we can discuss later. It's just some thoughts for discussion. And I'm going to put a couple of videos that I forgot to actually include, which were prepared also. Okay, so if we have, first of all, let me stop. What we have here? We have a plant inside the greenhouse, so suppose of a plant, which has maybe a lot of pollen because it has a high productivity of alternative resources for mites. These are mites, these are parameterized like mites and within the Weaver framework, and this is just a pest that is spread all over this simulation. You can see the background. In principle, you're going to see that when there is more predators, the background is going to be lighter because more prey are going to be lighter because there is consumption, right? There is the pest is working. And what you can see here is all the mites are working. They come out. This is just right, so the mites are forced to migrate because there is a lot of food, but it finishes in the day. So they are born and they are forced to migrate. And look at, they cannot reach the left. This is just by chance. We didn't prepare this. And the pest or the herbivores just come back, right? So the spatial distribution of whatever you do is extremely important. We didn't provoke this. This was just by chance. Okay, and then we can see the effect of hedgerows. For instance, we can have what we could call 2 greenhouses, one on the right and one on the left. So the pest will move very fast to the left if there is nothing that prevents them from doing it. Of course, there are meshes, there are like nets and things to prevent this. What happens if we put a hedgerow and some plants? That's probably something very differently. These are 4 plants on the corners and a hedgerow that start building up predators because there is food for these predators. And this is the pest. And eventually, the pest disappears. This is so beautiful. It's just a demo. It's nothing but a demonstration of what we can do with this. And of course, many people has worked on this. And many thanks to everyone, and thank you very much for your attention. Thanks. You have any questions? We have a few minutes for questions. ¿Tienes tú el otro micro? Pásaselo. ¿Quién no va ese? No, it doesn't work. Maybe this one.
Now, hello. Okay. Yeah, thank you, Jordi. That was great. Very interesting. And I have a sort of a pragmatic question about Weaver and the IBM. What is the dimensionality? How many? I was thinking that these days my campus, for instance, has a supercomputer where you can have huge arrays, and it's very interesting to understand how some of these processes scale with the dimensionality of the system, how many individuals you're simulating, and so forth. I'm just curious what your group is doing.
So when you talk about dimensionality, it's how the same simulation scales up when you use different systems.
Well, you could imagine like different sizes of patches. Some that have a few hundred. I have a student, for instance, who's doing
so you're talking about
IBMs with for invasive species, and she has, grids of 100 by 100 and 1,000 by 1,000, and so each grid cell is a single tree, and you can see how processes involving interactions and so forth scale with the size of the system.
So you're not asking how well it scales with increasing, with changing
Yeah,
scale?
I'm just wondering technically what you're doing in terms of how big the systems are.
Okay, the system can go from 1 little square to kilometers. It is all the power of the machine. So now we are working on paralleling this because this is amazingly sequential. We haven't paralleled yet. But where we're hoping to go to this is with big supercomputers in Barcelona. Yeah, but because we can have gigantic systems that, and everything is ready to do that. And of course, we can see what happens in a real ecosystem with real size. I don't know if that was your question, but that's it. Yeah. Any more questions?
Thanks very much, Jordi. I have a question about the leaf litter experiment. Do you know anything about the dispersal distance of the predators versus the prey?
We haven't done that, no. Okay. No. Because one of the problems with that is that they grow and they lost the mold. But I bet nowadays there are ways to do that. The problem is that we have abandoned that system. We work now in Almeria, going to Asturias, which is the other part. You come from New Zealand, so what I'm talking about. But we are going, it was doing field work 1,000 kilometers from here. So it was a big deal, and it was very, very nice and very nice system. But for now, we have abandoned that.
Okay.
Yeah.
Great.
Jordi. Yeah, thank you for the introduction, Jordi. Lovely. I have a question. It's a tiny question in comparison with the amazing framework, right? You have been building for years. It's about the emergence of interactions. We can discuss this in more detail, but as fitness is emerging, right? You don't set a priori any function. So how are you dealing with many interactions, and are they fluctuating? Meaning, would this a negative and antagonistic can be a positive interaction and a mutualistic, or so you have some functions there where interactions fluctuate in space and time, or are they fixed a priori?
Interactions depend on traits, so the output of interactions depend on traits. I see this framework that was published a few years ago by people that are here in the room, which is very nice, which uses Bayesian a framework to calculate the probability of interactions given the traits. We do something similar, but we don't do that. But the thing is, so the traits, if you are in a food web, you know what positive interactions can be, right? They're indirect, right? Mostly. So I don't know if your question is if in this framework we are going to put things like pollination or things that are explicitly mutualistic. Is that your question? Okay, yeah, that's not there. That's not there. That's not there. So the evolution of interactions from negative to positive, or from mutualistic to symbiotic, blah blah blah. That's not it. That's not it. Not even considered so far. It's a food web. You can put things on top, but David.
Jordi, on behalf of everybody, I'd like to thank you for organizing this symposium, and to you and Marta for obtaining the grant that made this possible.
The what? Sorry?
For obtaining the grant that made this possible.
Ah, okay.
Very quick question. What exactly do you mean by pattern-oriented modeling?
You find a regular distribution in the field, and you want to know what mechanisms drive that regular distribution. Question an ecology from forever, and then you try different submodels and see how it changes with these different submodels until you get close to that pattern.
So you're referring to developing a model that will explain a particular spatial pattern that's observed in nature.
That's just one example. It can be anything that you can imagine, even evolutionary questions. It's just pattern. They say we know that after the evolutionary experiment that Marta is going to talk about, things change. how, and we got this pattern, she will explain, how did we get there mechanistically during the experimental evolution. That would be pattern-oriented model. Yes? Okay, so it's time. Thank you. It's time for the next talk by David García Callejas, Signatures of Spatial Cascades Mediated by Dispersal and Foraging in Trophic Meta-Communities.
Okay. Can you hear me? Yes, right? Is it okay? Shouldn't it occupy like the full screen? Let me check. Oh yeah, it's fine. Okay, thank you. Well, first of all, thanks a lot, Jordi, for organizing the workshop, and thanks everyone for attending. It's really a pleasure to be here among so many respected colleagues. And after the very nice introduction by Jordi, I'm going to perhaps step back and try to put you on a more general view of spillover effects, and in general indirect effects, because spillover effects can be considered a set of indirect effects across species. So what I'm going to talk about is a very general question. So how do indirect effects propagate across space? I mean, this is a very general question that we actually, at least when I started this study and this idea, there is not a lot of specific hypothesis on that. So we can think about very different outcomes from indirect effects across space. And since I was doing my PhD on community ecology and networks, I thought about taking a community perspective on studying indirect effects across trophic networks and overall large meta-communities. So I'm going to talk here about how indirect effects are emerging in a community perspective, so in assuming a set of n species across different trophic levels, and so on. So within this overall idea of studying indirect effects across species, like I said, what do we know about their spatial dimension? How do these indirect effects are propagated in space? And importantly, if we are talking about indirect effects in space, of course, the different movement of species has to play a role. So I wanted to look explicitly about that, about how different types of species movements across local communities mediate this signature of indirect effects between species. Like Jordi very nicely mentioned in the previous talk, we can think of two main modes of movement in meta-communities. Of course, this is a very, very broad simplification, and there is a lot of variability on this. But perhaps we can divide, at least theoretically, the types of movements of species between local communities, between dispersal and foraging types of movement. So if we think about foraging, we may have something like this situation in which a certain species, in this case species B, forages its prey in the local patch and also in a nearby patch. So here the direct effects can be positive in green or negative in red, and so likewise for the indirect effects. So a certain species may have a negative indirect effect on its prey in the same or in a different patch, and this cascades and generates a whole set of indirect effects in the local patch and in the adjacent patch. And this can be, of course, extended to any situation in which different species move, or we have more patches and so on. This is the situation for a foraging effect, and we can think of a similar situation for dispersal effects, in which a population in a single patch may have actual positive effects on the population on a nearby patch because of the positive flow of individuals to a nearby patch. So in theory, we could think of very generally these situations as generating different sets of indirect effects in the community, in a meta community, right? So that was my question. How would we expect spatially the indirect effects to be emerging in these different situations across space? And well, how do we test this? If I had known before the experiments that you're running, I probably would have contacted you, Jordi. But in the time of my PhD, I didn't know of any experiments that were actually looking at this type of questions. So, of course, I had to resort to other type of experiments in silico. So I developed a meta-community model in which, based on trophic relationships, we can build food webs of any number of species, and then connect the local communities of these food webs across space in different arrangements. So I'm not going to talk about the details of the model, but it's a fairly standard meta-community model based on Lotka-Volterra equations, in which you can derive stability patterns and whatever you decide. And you can, of course, tweak the dispersal and foraging patterns of the meta-community. But I'm not going to talk about that, and I'm going just to present the main results of this study and some takeaway messages. So what is the first thing we looked at? We wanted to know whether the indirect effects between two species were similar, or how similar they were to the direct effects between them. So what happens when we include all the network of indirect effects between species and compare that to the overall positive, negative effects that we have directly between two species? In this figure, let me explain that a little bit. We have in the vertical axis the net effect between any two species, so that's considering both direct and indirect effects. And here, likewise, but on the same patch. So here is the net effect on species that are in different patches, and here on the same patch. And the idea is that each point is a pair of species. So when points are in this or this quadrant, the intra-patch net effect, so the effect of a species over another in the same patch, is the same sign as it is when species interact indirectly in different patches. So species A with species A in the same patch, or in patch A and patch B. What is their net effect? Likewise here. And these two quadrants would mean that the sign of the effect, of the net effect between any pair of species, is switched between same patch and different patches. I don't know if that makes sense, but perhaps with the example here it will make more sense. So in the situations when we only have dispersal between patches, we've seen that the effects between two species on a given patch compared to their effects when the species are in different patches are mostly conserved in terms of sign. So they mostly are in these two quadrants. So if we can expect a positive effect between two species that are in the same local community, we might expect that if the meta-community is mostly dispersal driven, their net effects when they are far away would be similar. What happens when we include foraging? Well, things basically blow up, in the sense that we cannot predict anymore the sign of the net effect between any two species in two different patches, so that there is a lot of pairs of species that change the sign of their net effect, and with no discernible pattern. And when we include both dispersal and foraging, we see even more stochasticity, or rather more complexity arising from this more complex set of interactions. So what to make out of this? Well, the first take-home message is exactly that net effects between species can be highly complex depending on the spatial arrangement of interactions. So the next thing we looked at is the structure of the network of net effects between species. And very quickly, we found that, as expected, there are net effects between every two species in the meta-community. So there is a non-zero indirect effect between every pair of species in our setup. This is given by the connectance of one in the network of net effects, which is the light green here. And well, the rest we can discuss that, but the average path length and the modularity are also interesting to actually make sense of some of the patterns that we see, but these are perhaps a little secondary. So another important thing that we looked at is the distribution of these net effects with spatial distance in our meta-communities. And we found that the strongest relationship was to be observed between the effect, the net effect between any pair of species here in the vertical axis, and the average path length between these two species. So the number of steps, of trophic steps between any two species was a strong indicator of the strength of the net effect between any two species. And this happens for the three configurations, when we only have dispersal, when we only have foraging, and when we have both. Note also that when there is dispersal and foraging occurring in the meta-community, at least in these model meta-communities, the net effects tend to be higher than in any other case. So that's a little food for thought for the rest of the talks, which will be likely more concrete. So this, as I see, this type of studies can serve as a baseline, so as an overall idea of what to expect in ecosystems in which we have not yet spatial heterogeneity and in which many species can disperse or forage. So this can serve as a baseline, if you will, for the more specific details and field studies that we will see, and I'm looking forward to seeing the rest of the talks. So as a couple of take-home messages, at least from these model communities, we can conclude that indirect effects are really widespread, which is obvious, but hey, at least we have seen it mathematically and in trophic meta-communities. They affect every pair of species in these large model systems. The sign and magnitude of indirect effects quickly become unpredictable as soon as there are spatially explicit foraging interactions. When we only have dispersal, well, you can more or less predict the sign of the indirect effects, but as soon as there are some trophic interactions across the local meta-communities, across the local communities, it's very difficult to do so. The last take-home message is that the magnitude of the net effect between any two species is, as you may think, strongly related to the path length between them, but not so much to the spatial distance between the local communities. So what are some questions that could arise out of something like this? Well, is there a way to approximate indirect effects in nature more generally? Can we actually, without resorting to very specific field experiments, is there a way to actually approximate indirect effects across communities? What happens when we don't have a knowledge of the dynamics of the system to actually quantify these indirect effects? And also, which species are key for propagating these indirect effects? Are these the top predators, or why? These are some of the questions that I am actually looking at right now with Jason in our work, and I would be happy to tell you a little more about it. And if you want to take a deeper look at this study, we published that a couple of years ago. So this is the title of the paper. And of course, I again thank you, Jordi, for organizing the workshop, and thank you all for listening.
Thank you for your inspiring talk. It was really, really very nice. I have two small questions related with the assumption of your
Yes.
modeling setup. One is when you mean different patches, are you meaning different communities or
Yes.
different region of the same habitat? I mean, there are differences in the community context between patches. The structure, the diversity, the composition
Yes,
of the community
that's a very good
differ.
question. That's a very good question. In principle, the local communities have the same species pool, and they're like local food webs, but with the same number of species and same structure. So each one has their own dynamics, which are then also including the dispersal and foraging between the patches.
But basically, they are the same communities
Yes.
in different
Yes.
places or wherever.
Yes.
And the other question, if I can, is the space? Have you modeled the space in fine grain or coarse grains? I mean, can individuals move very easily during their lifespan between patches, or they have to move very slowly, and one individual only can be in one patch and then reproduce, and the descendant offspring going to the other patches?
Since this is based on a lot-cabal Terra framework, dispersal and foraging are modeled as a diffusion coefficients from the populations. So a population in patch A has a probability of, I don't know, 0.2 of moving. So it's the kind of population that moves with a certain number of individuals.
Okay, it's fine grain, I think.
Yes.
Okay, thank you.
Yes.
Thank you.
Thanks.
Can I make a question? So mine is very quick. So I had the impression from your talk that you were saying that what happened at the local community does not predict what happens at the bigger scale when you accumulate these old patches. But this does not mean that the interactions become stochastic. Rather, they become in a different way of what will happen at the beginning, right?
It's not that they become stochastic, but simply that the indirect effects overwhelm the direct effects, and then you cannot tell what happens globally from the local interaction.
Okay.
Yeah. Professor Holt had a question.
Okay, yeah, so yeah, very interesting talk, thank you very much. So with indirect interactions, there's often an important effect of time signal. So imagine you do an experiment where you perturb the system, okay? You do a pulse experiment where suddenly you start inputting a species in one place. Then how the indirect interactions play out, often even the sign can change over time, and that would be an interesting thing to look at in your setup, I think.
Absolutely, because here we let the dynamics go, and we looked at the coefficients, the Jacobian coefficients
kind of
at equilibrium.
Right.
So yeah.
So there's a transient effect. So this comes up in very practical ways. There's been beautiful work done in Canada with indirect effects between moose and caribou via wolves and other predators. And the short-term effect of removing moose is to increase predation upon caribou because the wolves suddenly switch over. But the longer-term effect is wolves move someplace else or their numbers decline, and you reduce wolf predation. So you get sort of transient negative effects of the perturbation go into positive effects. And that'd be an interesting thing to do with your
Absolutely.
model, I think.
That would be very beautiful to look at, indeed.
Quickly. Oh.
Okay, just while they load me up, thanks very much, Jordi, for organizing this and for dealing with what I imagine were hundreds of administrative emails. Unless I was
Sorry
the
about
only
that.
one, but I imagine you got nothing else done the past month, so thanks very much. Cool. Huh. Cool, and it's just the next one there, right, to jump forward? Perfect. Thanks very much, everyone, for coming. I was giving some thought to this question of spillover and trying to think what is it that we would really want to know, or what would be the key factors if we were to try and come to some kind of a predictive understanding of spillover effects. I'll apologize from the start. I'm not going to restrict myself to just predators. We've done a bit of work on pollination, so I'll kind of move back and forth between mutualistic and antagonistic interactions. But when thinking about predator spillover from one habitat to another, I was trying to think about what would be the critical elements. And the first thing we need to know is what are the species that are contained in different habitats, and to what extent can we begin, or can we know this from the beginning, or make some predictions? Then the next question is, of those species in different habitats, if we can predict what they would be, which ones of those, which subset will be the ones that are most likely to spill over? There's also the possibility that if two habitats are juxtaposed within a landscape, species will spill over in both directions. Can we be predictive about which of those directions spillover is likely to occur in? And then once species do arrive, what's going to be their impact on the communities? And related to what David was just talking about, what's the kind of spatial extent of those impacts? So these are all of the things I wanted to talk about today. I'll probably talk a little bit less about the spatial extent because that's the one we've done the least on. But starting off, just thinking about what's likely to be in different habitats, and we already have a fairly good idea of what this is likely to be. We know, for example, that disturbed systems, agricultural habitats, as well as smaller habitats, tend to have more habitat generalist species. This was some, I'm going to scrape the bottom of the barrel in this talk. This is some of my PhD research a while back, and looking at different habitats in coastal Ecuador, looking at cavity nesting bee and wasp communities. And this was, this figure here shows you the specificity, which is essentially the deviation from what you would expect based on random occurrence given the abundance sampled of different species. And long story short, the more on your right, the more natural habitats tend to have species that are only found in those habitats. The more intensive habitats tend to have species that are found pretty much everywhere. And so we have this sense of disturbed habitats having habitat generalists, and we also find that disturbed habitats and smaller fragments as well tend to have species that are more interaction generalist. And as I go through this talk, I'm going to try and make the point that those two things are probably not independent of each other. If a species can interact with lots of others, it's then able to find partners to interact with in other habitats. So this paper on your left, I wasn't involved in, but it was a nice global study of, I think, about 18 or 19 butterfly data sets from around the world. This is their summary figure, and the blue lines here are generalist species, and the red lines are specialist species. As area declines, so as you get into smaller fragments, the specialist species have a rapid drop in diversity, whereas the generalists less so. So in short, the generalists, which they measured as the number of plant families on which these larval butterflies fed, tend to be present or able to survive even in smaller fragments. And this was some work we did with Marcelo Eisen in Argentina, looking at pollination networks, and on the x-axis is interaction degree, so the average number of interaction partners. So this was looking at a particular interaction, and when both partners in that interaction were generalist, they would have a high interaction average interaction degree. If both of them were specialists, it would be low. And ubiquity was the number of fragments we found them over along a gradient of mainland island fragments, or as representing fragments. And what we see is that the interactions among generalists tend to be the ones that persist even in the smaller fragments. So we would expect that we can have some predictability about the habitat generality and the interaction generality of species in different habitats. We also know something about how trophic levels respond to habitat differences. There's been quite a bit of research on the vulnerability of higher trophic levels to different kinds of disturbance. This is a nice study led by Thomas Roslin, which I wasn't involved in, and it also built on some ideas, as with many ideas in ecology, if you follow them back far enough, they end with Bob Holt. But what this showed was looking at species across different island sizes in the Finnish archipelago, and you see that plant species richness is not particularly affected by island size. It's a relatively flat slope. Herbivores a bit steeper, primary parasitoids steeper, and secondary parasitoids steeper still. So higher trophic levels are increasingly vulnerable to small island effects. So we might also be expecting that smaller habitats or smaller habitat islands are going to be richer in lower trophic level species than higher trophic level species. And that matters if we're trying to manage predator spillover. And then lastly, if we think about productivity differences across habitats, which is a theme that's going to continue a little bit during this talk, we might also have some expectations about how productivity will drive differences across a community. And this was some work led by one of my former PhD students, Claudio De Sassi. It was done in New Zealand in the Cass University's field research station in the Cass Mountain area. These are plots where we exposed them to experimental warming and or factorial with nitrogen addition. And we looked at changes in the plant community, which were grasses that we planted, as well as some that colonized themselves, their lepidopteran herbivores and the parasitoids that fed on those. You can ignore the warming bit. It's not what I'm going to, the point I'm trying to make here, but if we look at the control plots in gray versus the nitrogen addition plots in white, we can see the effect of experimental productivity increases, and we saw an increase in plant biomass, an increase in herbivore biomass, but only a small and non-significant increase in parasitoid biomass. So the lower trophic levels seem to benefit from the resource addition. The higher trophic levels were unable to catch up with it. In this case, it was probably more of an aggregative response than a population level response, given the scale of the experiment. So we can have some predictions about what's likely to be in different habitats and how that'll respond to management intensity and productivity difference. But then we can ask ourselves which of those species are going to be the ones that are most likely to spill over. And the punchline of what I'm going to argue is that it's mostly the generalist species, both habitat generalists and interaction generalists. This was some work led by Guadalupe Peralta on the edge of... Somebody's shooting at me, it sounds. This was at the edge of natural forest and plantation forest in the north of the South Island in New Zealand. She looked at parasitoid host food webs on those habitat edges. Then she also looked at the food webs in the interiors of each of those habitats. She simulated a random sampling of what edge webs should be if they were just a random mix of the two interiors, and compared the observed ones to the random expectation. Each of these bars is one of her replicate sites. And just an example of a couple of network metrics: generality, the number of hosts used by each parasitoid, and linkage density, which is just the average number of connections per species. We can see that on the whole, the edge webs were more connected than you would have expected based on random mixing of species and interactions from each of those habitats. So you were getting a kind of, and she dug into this deeper and found that it was the species that were most generalist in each of the interiors were the ones that were most likely to occur at the edges. So the generalist species are accumulating at edges. And this was some work with Jane Mehmet's group led by Talia Hackett and Alex Salve, and this work we did it in the UK and in New Zealand. We had landscapes of different habitats. Inside each habitat, we looked at multi-trophic communities or meta networks, multi-layer networks containing lots of different kinds of interactions. And the main point I want to make here is we were looking at how species not only had roles connecting up different parts of these networks within their habitat, but also their role in connecting among habitats. And what we found was that the species that were most generalist in their own network were also the ones that were most likely to be found across different habitats. So the generalist species are definitely the ones that are connecting up the landscape. And also in the case of pollinators, we found that these generalist species tended to be also the larger bodied species, which are potentially the best able to disperse, at least in pollinator community. There's work from Claire Kremen's group showing that larger bodied pollinators are better dispersers. It's probably a fairly common phenomenon across a variety of taxa. So when large body size correlates with generality, then you're again going to get more generalists moving among those different habitats. So just to make that point about body size, the potential relationship between body size and generality, and how it's possibly a phenomenon that's consistent across lots of different kinds of species interactions. This was some data collected by Christy Weber during her master's, looking at pollinator body size and the number of interaction partners a pollinator uses, and there was a positive relationship. This was some work led by Guy Woodward, which I wasn't involved in, but it's a difficult figure, but the take-home message is pretty simple. These are predators of increasing size class. These are each dot is a prey species that the predator feeds on, and you can sort of see that this forms a wedge that as you get larger predators, they're feeding across a larger range of prey species. Those are in aquatic food webs. So within food webs, it's quite obvious. Bigger things have bigger mouths and can fit more different things into those mouths. We see a similar phenomenon in plant frugivore networks, and we also see it in pollination networks as well. Larger things tend to interact with more different partners, and because we also know that body size is correlated with dispersal, therefore, again, we're expecting that generalists are going to be the ones that are connecting up the landscape, interaction generalists, which are also habitat generalists. So if we have an idea of what's there and what's likely moving, the next thing to try and understand is which direction are things likely to be moving in. Are we putting in natural habitats for predators to move out into our crop environment, which is what we ideally want in biological control, or are we just setting up our natural environments to get hammered by enemies from those productive areas? So this was a paper with Tatiana Rand and Taya Chantke and I, and we hypothesized a few years ago that if you had a situation where you had a natural habitat and some kind of a crop environment, you could expect predator spillover from the natural habitat to the crop, but the high productivity of that crop would become a source for, or would have high productivity also of prey, and therefore could sustain higher predator populations. And you might then, after harvest, or even passively, continually through time, you might expect more spillover in the other direction, from the more productive habitat going back into the less productive habitat. So this was something we tested in a study again at the edge of native and plantation forests. I should mention plantation forests, Pinus radiata, primarily in New Zealand. It's got a much higher primary productivity than our native forests do in terms of the biomass that accumulates per year. It's not surprising, that's why you grow plantation forests, is because they grow quickly. And what Carol Frost did was look at this edge and look at consumers or high trophic levels consumers moving across that edge, and she put up flight intercept traps along this edge of forest types, and she could record the direction that things were coming in because it captured them separately from each direction. So she looked at the spillover in both directions, and what she found was that there was spillover in both directions. She looked at both vestibular wasps, which are more generalist predators, and parasitoids, which tend to be more specialist. And there was spillover of both in both directions, but it was asymmetric that there was definitely more spillover from the plantation into the native forest, which was roughly proportional to the double the productivity of the plantation forest and the native forest. And in fact, we even see this spillover according to productivity at smaller scale. This was some work led by one of my former postdocs, Warwick Allen. And the x-axes here talk about provenance of native versus exotics, but these were in experimental plant communities where the exotic species were the fastest growing. And what he found was that the number of herbivores that attacked those exotics was much higher. So he had experimental mesocosms where he had different plant communities in them, exposed them to experimental herbivore communities. The herbivores went for the exotic species, but then those exotic species shared a lot of herbivores with the native plants, and in fact were most likely to drive apparent competitive effects, or we would predict that they would have stronger apparent competitive effects on the natives by virtue of their higher productivity. And then in another field experiment, he actually showed this experimentally, that exotic plants were having negative indirect effects on native plants, largely by virtue of the high productivity or high biomass of those exotics. So from whether we're talking at very small scales of individual plants up to whole habitats, productivity is definitely going to matter, and we can expect asymmetric spillover from high productivity to lower productivity habitats. And really hitting the bottom of the barrel here, this was some work from back in my master's. I just wanted to make the point that you can even see spillover of energy coming from one habitat to another. So this was looking at planting floral patches in cereal fields to try and enhance parasitoids and controlling aphids. And essentially what we found was that when you were close to these floral patches, you'd get higher attack rates on aphids, and then as you got further away, the attack rates would drop. This is because floral resources provide energy for parasitoids, help them boost their egg load, and also help them live longer. But essentially what this is some kind of resource difference between those two environments, and the resource itself is moving within mobile consumers. So we can expect a spillover of energy, even if those consumers were originally already flying around in that cereal environment looking for prey. So what I want to spend most of the rest of this talk on is thinking about what's going to happen once species do spill over. What's likely to be the functional impact of spillover into recipient communities, and particularly if we begin with the hypothesis that the species doing the spillover are going to be more generalists. So what's likely to be that functional impact of bringing generalists into the environment? And when we think about interaction generalists, or when we think about interaction networks in general, they tie closely, implicitly to the previous research on biodiversity and ecosystem functioning, which probably most of you are familiar with. This work has shown that species that are complementary in their resource use, the early work by Tillman, for example, in grassland communities showed that species with complementary soil resource use functioned better. And complementarity in resource use is implied within the architecture of a network, that when species share few partners with each other, they're effectively partitioning the partner resource and have higher complementarity. In contrast, if they share a lot of partners, you would have lower complementarity, but you would have higher redundancy in partner use across that network. So we would expect potentially higher overall average functioning rates with high complementarity, but maybe more stability in functioning with high redundancy. And this was something Lupe Peralta again looked at in her PhD in parasitoid host communities. This was interaction complementarity, sharing of the high complementarity is not sharing of host partners among parasitoids was related to parasitism rates, whereas high redundancy, so when parasitoids shared a lot of partners, you tended to have less variability, although not quite significantly, but a tendency towards lower variability in parasitism rates across different sites. So species were able to fill the gap when their other parasitoids could fill the gap when one enemy was missing. So just briefly to make a point that here when you have high redundancy and high complementarity, it's coming from high generality of those interaction partners. So as we move, or if we expect that spillover is driving the movement of generalists, then we would expect we're moving from this high complementarity to low complementarity, high redundancy networks, which could potentially be worse for functioning, but better for stability and functioning. I'm also going to, this is going to seem like a bit of a detour, but it's going to take me back to something related to functioning. But I just wanted to mention briefly that interaction generalists also tend to choose their partners differently, it would seem. So interaction generalists are more likely to choose their partners based on what's available rather than based on their traits. So this was some work led by Lupe Peralta again on plant frugivore networks. She looked at how at the strength of trait matching in those networks, the extent to which frugivores were picking fruits that closely matched their beak size, which is known to be optimal for bird foraging. And we found that species that were more generalist tended to have less trait matching. So generalist species weren't picking the most optimal fruit, they were just eating anything. And this was particularly the case for interactions involving exotic species. They were just eating whatever. And in a separate study led by Camille Cooks, who did a PhD in my group, she showed that the flip side of this, that exotic species tended to interact more neutrally. So they would be more likely to feed on fruits that were available or abundant rather than showing specific preferences for particular kinds of fruits. In contrast, native bird species were more likely to find particular fruits even if they were rare and feed on those. And we also see this pattern in the phylogenetic matching of interactions too, that in this case this was some more work by Lupe Peralta in our edge of native and plantation forests. The plantation forests tended to have a stronger signal of coevolution, which meant more interactions with strong trait matching, and native forests tended to have weaker trait matching. Now, at first that seemed a little bit counterintuitive. Our hypothesis coming into this was that more natural systems would have a stronger signal of trait matching. But what we're starting to see now is that actually more stressful environments cause that the best matched traits are the ones that species can rely on most, and the other poorly matched traits, so opportunistic interactions with things that aren't particularly good partners, tend to be more flexible, but those are the ones that are lost in stressful environments. So what we see in more disturbed environments potentially is the falling away of these opportunistic interactions as species are forced to focus on their core interactions. And if anyone's interested in this, this is something we've been looking at more, but I won't talk more about it now because it's a bit of a digression. But the point was that if we're getting spillover of generalists, we're likely to be moving communities into these more neutral, interacting with whatever's there types of interactions rather than these more niche-driven interacting with species with shared coevolutionary history and matching traits. And unfortunately, in some more work led by Lupe, we found that trait matching, those interactions, this was in pollinator plant networks with best trait matching, were the most effective in terms of their pollination success. So as we're moving to communities with more generalists, they're likely to be interacting with less trait matching and therefore having poorer functional outcomes. again, like we're seeing the less interaction complementarity of those generalist species. So we might expect that a shift towards generalism is going to be worse for functional outcomes, or particularly for specific functional outcomes of a given pest species. And just to continue with the same linking of interaction generality with complementarity and potentially with functioning, this was some more work by Camille Cooks during her PhD. And this is an ordination of pollinator species in trait space. So each point in here, or each circle in here, is a pollinator species. Circles that are close together have traits that are quite similar. The overall variation of traits, the whole hypervolume, is an indicator of functional diversity. So species at the periphery of the functional trait space are the ones that are contributing the most to functional diversity, whereas species at the center have kind of average traits, just like everybody else. These circles here are colored by the number of interaction partners. So more red equals more generalist. And you can see here that the species closest to the centroid tended to be the most abundant, and that's partly because they pulled the centroid towards them. And the centroid, sorry, is like the multivariate average. So the traits that if you picked any individual out of the community at random, its traits would be most likely to be there. And we find that the red species, the generalists, are closest to the centroid, and the species with fewest partners are furthest away from the centroid. So there's a relationship between species generality and their trait distinctiveness. The species that have the most unique traits, potentially to escape competition with their own trophic level, lose interaction partners. Because I guess you could imagine being a flower that becomes so unlike every other flower that no pollinators want to visit you anymore. Maybe they don't even know you're a flower anymore. So there's some trade-off between interaction partners, maintaining interaction partners, and evolving different traits. And we see that there's this correlation between species that have the most unique traits, so contribute most to functional diversity, are also the species that tend to have the fewest interaction partners. Again, another piece of evidence that if we're getting spillover primarily of generalists, we'll be losing those species that contribute most to functional diversity and instead getting species that are most functionally average. So then moving from what are the kind of functional impacts more generally, but thinking about what are the population impacts of spillover predation, this was something that, again, in those edges of native and plantation forests in that project, in this case led by Carol Frost during her PhD, we were interested in trying to see the extent to which we could predict spillover and the extent to which we could predict the functional consequences of that spillover. So what Carol did was sampled parasitoid and host communities at the edges of those habitats, but within the native forest and within the plantation forest. Then she constructed networks of parasitoid host interactions, and then she was able to, for those working on networks, project the bipartite network into a unipartite projection of host sharing among parasitoids. So each of these nodes here is a host species, which would be a caterpillar in this case, a Lepidopteran larva. And these connections don't show a parasitoid attacking a host, but rather they show the sharing of parasitoids among two host species. So two hosts, that have a thick connection between them, like say this one here and that one there, share a lot of parasitoids with each other. The coloring of these links, the blue ones indicate hosts in native forests that are sharing parasitoids with each other. The red ones indicate hosts in plantation forests that are sharing parasitoids with one another, and the grey ones indicate hosts in one habitat that are sharing parasitoids with another. And the first thing you notice is that there are a lot of grey links, which means there's a lot of sharing of parasitoids across those two different habitats. If we know how much two hosts share parasitoids, we should be able to predict their indirect effects on one another, because if, for example, this host were to become really abundant, it should provide a bottom-up subsidy to its parasitoids, which should then increase attack rates on all of the hosts that it shares parasitoids with. Does that make sense? Cool. So what Carol did was take this overlap graph to make precisely that prediction for every host in these communities. So what she predicted was if the abundance of every host changed by a given amount, what should be the change of attack rate on every other host in the community, both in plantation forest and in the native forest adjacent, based on their sharing of parasitoids. Then she used those predictions combined with changes in abundance to see if she could predict accurately those changes in attack rates. And she got changes in abundance from two ways. She looked at just natural changes in abundance of hosts and how that translated into changed attack rates. But the problem with that was that those changes in host abundance could actually be related to the changes in attack rates, and it would be difficult to get at causality with that. So she also caused an experimental change in abundance in hosts in order to see whether that influenced her ability to predict changes in attack rates. So to do that, Carol flew over these habitat edges in a helicopter and used Bacillus thuringiensis, a caterpillar-specific bioinsecticide, in the plantation forests only, because obviously you don't want to be wiping out caterpillars in our native forest, but in the plantation forest they're quite happy for her to wipe out their pests. So she sprayed about 25 hectares of plantation forest with caterpillar-specific insecticide, more or less wiped out the hosts from that area, and was able to see how that changed parasitism rates in both of those habitats. And so she had those changes in host abundance. She used this network that she had sampled initially to predict how those changes in host abundance would translate into changed attack rates across the entire community. And she found that her expected change in attack rates was significantly predictive of the observed change in attack rates, although there is obviously a fair amount of noise in those. But the more interesting thing was not just that she could predict changes of attack rate within a habitat, but she could predict them just as well. The predictive ability was just as good across habitats. So changes in abundance in the plantation forest, changes in abundance of host, caused predictable changes in attack rates of hosts in the native forest. So the whole system is functioning essentially as one giant network, as one giant food web. And unfortunately, because the plantation forest is so productive, enemies from plantation forest are constantly hammering hosts in the native forest. So the spillover effects are driving changes in attack rates. And Carol was even able to show that those changes in attack rates and the expected parasitism rates were related to changes in abundance at the following time step of hosts. So they were having population level impacts on host abundances. So if we think of, often as ecologists we go to different habitats like a forest, and we call that a food web. We sample it and we say that's the food web of this forest. What we see here is that two different forests are actually functioning as one single food web. So then the question becomes, what's the limit to that? Over what scale would we cease to see these things functioning as a single food web? And that relates to what David had just talked about. And I think it's still an open question of what is the scale over which these indirect effects are likely to be propagating. We don't yet have an answer, but Lucas Pereira Martins, who's just finished a PhD with me and is still doing a postdoc with me now, looked at this in plant frugivore networks, and what he was interested in is there some upper limit to the potential for indirect effects across habitats. So he did a meta-analysis of nearly 200 plant frugivore networks around the world, with a lot of plant and bird species in them across 67 ecoregions. Each of these points is one of his local plant frugivore networks, and the connections among them indicate that interactions were shared across those two networks. And the blue links indicate that those networks were in the same biome, even though they were spatially distinct, and the red ones are in distinct biomes. And what you see straight away is that there are way fewer red links than there are blue links, which means that you're much more likely to share interactions across space if you're in the same biome than if you're not. And that's not just explained by the sharing of species, but the interactions, there's also a rewiring component to that. And he looked at this within ecoregions as well. So ecoregions being finer scale differences in the biotic and abiotic environment. And this here is a plot of change, of increasing spatial distance. And this is the turnover of interactions among those networks. And we see that there's a rapid decay in interaction similarity until about 2,500 kilometers. And then it more or less flattens off. So there's this fast distance decay in the sharing of interactions, but then that cutoff point gets brought closer, so you get a rapid turnover of interactions if you cross into different ecoregions. So these are ecoregions that are distinct ecoregions. This is the same ecoregion, and these are frequency distributions of interaction dissimilarity, with one being highest dissimilarity. Basically what this shows is that in distinct ecoregions there are lots of ones. So when two sites are in distinct ecoregions, they're likely to have complete dissimilarity of interactions. Whereas if they're in the same ecoregion, you're more likely to have some sharing of interactions. So it seems that ecoregions and biomes, ecoregions are nested within biomes, are providing some kind of a hard boundary for the sharing of interactions, and therefore potentially a hard boundary for the propagation of indirect effects. But again, I think it's still an open question how far those indirect effects can propagate. And still thinking about the functional impacts of the spillover of generalists, I wasn't involved in either of these, but I just wanted to point out that even though generalists can potentially interact with a lot of species, so any change or disturbance to generalists, any change in the frequency of generalists should drive or should impact a lot of species within the community. And they should create a lot of indirect effects because they shorten the paths between species. There's the potential, though, for those effects to be dissipated quite quickly. So we might see with spillover of generalists impacts across a lot of species, but not potentially such long distance impacts as more specialized species that have dynamics that are more closely coupled. So I think these are some hints to potentially starting to think about what is the scale over which indirect effects can propagate. I'm now getting into the most speculative stuff, but I thought it's something that's interesting to think about is the potential for evolutionary impact of spillover as well. So if we know that we're selecting for generalist species within habitats, so there's a form of ecological selection, there's also going to be genotypic selection within different habitats. And if we imagine here we've got a gradient from low to high productivity. This was just a thought piece with Lais Maya, one of my former PhD students. If you think about environments with high productivity, they're often quite harsh environments because of high chemical usage, for example, lots of insecticides or herbicides. So you tend to have low diversity in those environments. As I showed you earlier, they tend to be generalist species, but any species that can survive there has the perfect life because there's so much productivity there. If you can overcome the harshness of a productive environment, you have all of that food, all of that resource to make use of. So there's a high selective pressure to be able to survive in those environments, and therefore if you're getting high buildup of species that have been selected in productive environments, then you can get spillover of those genotypes into the surrounding landscape, which could potentially influence coevolutionary processes. And some work by Bob back in the, I want to say in the 90s, showed that you could even get through those kind of processes, you could get maladaptive traits becoming fixed outside in the surrounding habitat, so you could get a spillover if you've got one very productive habitat where a certain kind of trait is beneficial, it could spill out and become fixed in an environment where it's not beneficial, where it's maladaptive, purely on the basis of the high productivity of that environment. So we might expect an evolutionary consequence of spillover across the landscape, and this is something that I think we know, as ecologists, we know embarrassingly little about. So to put all of that together, I think if we were to make come to some general predictions about spillover across habitats, we would tend to predict that it's going to be stronger from high productivity to low productivity habitats. That's a problem if we're trying to have kind of nice natural reserve areas next to highly productive agricultural systems. But potentially in the case of your experiments where you're adding water and boosting productivity of those sort of more natural environments, potentially you might be more likely to get spillover out of them. And that's because the spillover would go in that direction, both because you'd have higher productivity in the more productive environment, providing a bottom-up resource subsidy, but also in more productive agricultural habitats generally, you tend to have more interaction generalists and habitat generalists, which are those species that are best able to spill over. We would also predict that spillover is more likely to involve lower trophic level species because the higher trophic level ones tend to get hit harder in smaller fragments or in agroecosystems. We would expect the spillover to be of large-bodied species because they can disperse further and also because they tend to be the most generalists. And we would expect this to be the case particularly when you're coming from small habitats, so habitat patches. So if you're trying to plant a small natural area within an agricultural landscape. We would expect that those generalists, when they do spill over, will contribute to functional redundancy but potentially reduce complementarity. And we would also expect those generalists to engage in more opportunistic interactions with whatever species happen to be available rather than interactions with species with well-matched traits. So it might be more difficult to predict based on trait matching the consequences or the impacts of those predators. But if you're trying to target a pest species that's abundant, then potentially that could be an advantage. And we might predict, but again, I think this is an open area that there's more to understand, that the spillover of generalists will directly affect more of the community than you would expect at random, but those indirect effects might dissipate quickly. But we do know that those indirect effects can propagate across habitat boundaries, so at least what we consider to be different habitats may be functioning as one, although we do know that there will be some upper limit biogeographic boundaries to that propagation. But where the propagation stops in between those scales, I think we don't yet know. And lastly, we need to consider not just ecological spillover, but also potential genotype spillover and the selective mosaic of a heterogeneous environment and how that could drive evolutionary processes. So there's a ton of people to thank, and I'm not going to try and list them all, but I've hopefully mentioned some of them as we've gone along, of all of the lab members and collaborators that we've worked with. This work was paid for by a lot of people, and thanks also for those people who paid for me to come here today. Thanks very much.
Thank you, Jason. That was great. Really a terrific summary of lots of intertwined ideas. I have an empirical question about your New Zealand system, the plantation to native forest. What happens at the lower trophic level? Do you find an impact in terms of like amount of leaf damage or anything like that?
That was hard to get at, and part of the problem was a lot of them are up in the canopy, so we had to kind of use pruning, long-range pruning, long-range pruners on the end of a stick to try and get branches down and sample the canopy and things. So I think we probably didn't have the right system to be able to get at leaf damage. There was some damage in the understory, but from memory, we weren't able to really predict that very well.
Yes,
But
quickly.
certainly in the plantation forest, most of the productivity is up in the canopy anyway. Carlos.
Yeah, thank you, Jason. Quickly, I still miss one component. I think this was a great summary for spillover effects. But from your patterns, it seems generalists, they produce weaker interaction strengths, and the population effect can predict a spillover. So, for example, for the network you saw in New Zealand, did you try to overlap the potential effect of generalists and specialists to predict the spillover? So did you do a partition? This is what I mean, because you need some strong interactions somehow to really consolidate, right, the spillover effect, or you don't need this? You only need the generalist species. This is my main.
Well, I think that's a good question. We separated them in that example of Carol's, where she just Carol Frost's, where she measured the spillover. So we only separated them as far as vespular wasps versus the parasitoids, which tend to be more specialized. But there are parasitoids that are more generalist and some that are more specialist. And in the following work where we predicted the indirect effects, we used all of the parasitoids together, but none of the wasps. So there's likely to also be indirect effects driven by the vespular wasps, but we didn't measure those. So I guess to answer your question, even with relatively specialized, relatively rare things, there was a population impact.
It's time for a coffee break. We'll be back here at 11:30.
Oh, well, it's not on. Ah, okay, okay.
Okay.
Ready. Testing? There, it's on. Okay.
Okay. We'll establish now with the talk by David Wise from the University of Illinois at Chicago, 2 different approaches to uncovering patterns of intrahill predation in forest floor.
When Jordi first wrote me from Barcelona about being his co-advisor for the PhD, I never dreamed that as an emeritus professor, Jordi and I would still be collaborating on research. And this collaboration has been really one of the highlights of my entire career, so I want to thank Jordi right now for that. Jason's already talked on this. I think I couldn't begin to. Do I have to find it? I thought someone back below it. I'll let someone else handle the tech. Yep, and then yeah, this controls the slide. Which one? Right. Okay. 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 Let's see if we can go. There we go. Okay, now I got it figured out. Adding detritus, such as mulch, to vegetable crops can increase populations of fungivores, boosting densities of natural enemies, such as spiders and carabid beetles, thereby potentially increasing vegetable production. This approach was integral to my biocontrol research at the University of Kentucky. In 1997, during a meeting of the Ecological Society of America, Gary Polis suggested to me at breakfast one morning that I consider this detrital addition in a broad theoretical context. Thus, in a manuscript that we submitted just a few months later after that breakfast, we described our proposed biocontrol technique as an allochthonous energy subsidy via multi-channel omnivory, quoting papers by Gary and Don Strong. That's a very high density of jargon per linear centimeter. Allochthonous because we added detritus from outside the system to increase the strength of trophic cascades in the green channel via the link of generalist predators. In reality, however, most webs do not consist of neatly separated trophic chains. And Gary earlier had argued strongly and convincingly, as has been mentioned already today, that terrestrial food webs exhibit widespread trophic level omnivory, that is, feeding by generalist predators on 2 or more trophic levels. Gary and Bob Holt published a highly influential set of papers on a widespread type of trophic level omnivory called intraguild predation, or IGP. Conventionally, the top predator in this food web module is the IG predator, and the intermediate level predator, the IG prey. This figure depicts asymmetrical or non-reciprocal IGP. That is, the IG prey does not prey upon the IG predator. Today I will describe 2 approaches to understanding the dynamics of intraguild predation in food webs on the forest floor. The first study examines how an experimentally imposed allochthonous input of detritus affects the dynamics of a postulated IGP module in the soil food web. Our approach is radically unconventional because we define an entire community of soil microarthropods, mites, and collembola as a single IGP module. The second study examines the temporal stability of a forest floor food web, a spider-focused forest floor food web, in which practically all interactions are IGP. These two approaches implicitly ignore the complications of spatial and food web heterogeneity. We examine properties of two complex food webs by describing them in the simplest terms possible. And our goal was to discover what insights might be gained about a system by first ignoring its complexity. Thus, these two studies can be viewed as a foil, if you will, a contrast to research that explicitly recognizes heterogeneity to examine the strength and dynamics of spillover effects. Early theoretical research by Bob and Gary revealed that the IGP module is dynamically fragile. Coexistence of all nodes requires a balance between gains to the IG prey from exploitative competition for the shared resource and gains to the IG predator from consuming the IG prey. The rate of energy and nutrient input to the shared resource is critical. Too low in the IG prey outcompetes the IG predator, driving it to extinction. Too high in the IG predator persists and the IG prey disappears. Just right, and all three coexist. Let's imagine an IGP module of three coexisting nodes at equilibrium. What happens if we increase the input of energy or nutrients to the shared resource? Classical theory predicts the following response, which reflects the tension between bottom-up and top-down control. Densities of the resource and the IG predator will increase, and densities of the IG prey will decrease. As the energy put increases, the IG predator eventually will exclude the IG prey. More recent mathematical modeling, employing slightly different assumptions, such as that of Sabina Volrap and her colleagues, leads to the same predictions. Monica Farfan and I used this prediction to determine if the community of soil mites and collembola behaves as a single IGP module within the soil food web. This community of mites and collembola occupies two horizons, the humic layer and the litter horizon. Fungal hyphae in both layers support many species of mites and collembola, which are fungivores. We'll consider them strictly fungivores for now, although they also consume detritus and so on, which are fed upon by a wide spectrum of predaceous mites. Based upon known feeding behaviors and relative body sizes of adult and juvenile stages, 17 families of predaceous mites were classified as either IG predators or IG prey, with 23 families of fungivores being the shared resource. Such diversity clearly includes numerous IGP modules. Nevertheless, if IGP is widespread throughout this community, one would expect the dynamics of some of these IGP modules to be synchronous. And if the dominant IGP modules exhibit synchronous dynamics, then IGP would be an dominant organizing principle or force in this community. Thus, we hypothesize that this community could be considered as a single IGP module within the soil food web. Previous research by Kendra Lawrence found that adding artificial detritus, a combination of dry fruit fly medium flakes and chopped mushrooms and potatoes, could double or triple the abundance of fungal hyphae in the leaf litter horizon. How would densities of fungivores in the two categories of predaceous mites respond to a similar input of detritus in the system Monica studied? This pattern of response would be confirmation of our hypothesis that this complex microarthropod community could be modeled as a single IGP module. Monica and her assistants located 200 1-square-meter plots within a forest outside Chicago. 150 of these were fenced. The other plots were left open as a reference treatment. 3 levels of detrital supplementation were added randomly to the fenced plots. The rate of addition to the low treatment, the 1x treatment, was similar to that employed by Benrong Chen in a previous experiment in the mountains of Kentucky, in which densities of mites and collembola had increased 2 to 5 times after 3 months of addition of the same kind of detrital supplement. This rate of addition that Monica used and Ben used was the same that increased fungal densities in the previous experiment conducted by Kendra Lawrence in the same Kentucky forest. Detrital enhancement was applied every 2 weeks for 3 months by Monica and her assistants in year 1 and 4 months in the second year. And to document initial conditions, microarthropods were sampled a week before addition started in year 1. Arthropods were again sampled before the resumption of a detrital supplementation in year 2. And the microarthropods were sampled then at the end of each of the 2 field seasons. Over 45,000 mites and collembola were collected, identified to family level, and assigned to one of the 3 nodes in the hypothesized IGP module. Did the system respond according to theory? So let's look first at how densities of the 3 nodes changed. These plots show mean abundances per sample with 95% confidence intervals for the 3 compartments. The red line along the abscissa represents weeks when detritus were added, and month is the number of months since the beginning of the experiment. Let's focus on the high treatment, the blue circles, which display the most pronounced responses based upon comparisons with the none treatment, the gray circles. Densities of all 3 nodes had increased in size in response to the detrital treatment at the end of the first season. In fact, if you look at the entire pattern over 2 years, the treatment times time interaction is statistically significant, to use those forbidden terms, that forbidden term, with ranging from a p-value of 0.0001 to 0.02. However, if you look closely, you'll see the responses were not identical. After 1 season, all 3 nodes had increased. And the positive effect of adding detritus appears, the high level of detritus appears to have persisted to the end of the experiment for the IG predators and also for the shared resource. In contrast, the response seems to be declining for the IG prey. Thus, the pattern appears to confirm theory, at least for the IG predators and the shared resource, the fungivores, but only partially for the IG prey. Might this pattern suggest an eventual trajectory of IG prey densities in the high treatment becoming less than those in the none treatment, as predicted by theory? That was a tantalizing first glance at the pattern, but unfortunately, this possible congruence between empirical and mathematical models is misleading. For one, the broad confidence interval on the mean density of the IG prey after just 3 months makes interpretation of a temporal pattern very problematic. And even more importantly, comparing densities in detrital addition plots with the no addition plots is not a direct test of treatment effects. Instead, statistical modeling of effect sizes over time is the more powerful approach. First, let's examine how the IG predators responded. Confidence intervals for effect sizes at the end of both field seasons for both treatment levels, that is the high rate of addition and the low rate of addition, the high is in blue, the low is in black, do not overlap with 0. The effect size of the high treatment for IG prey is about 50% that of the IG predators, but the 2 predator nodes both respond similarly to the low treatment. Thus, IG prey exhibit a weak but consistent positive response to detrital supplementation, certainly not in agreement with what IG integral predation theory would predict. The pattern for the shared prey, the fungivores, is even less supportive of the hypothesis that this simple trophic network is an IGP module. For example, compare the patterns. Is that a signal that? Oh, still here. Okay. All right. We've just established that I'm not doing anything. Okay. Okay, that's good. Compare the patterns for months 9 and 13 for all 3 nodes. Fungivores exhibit the weakest overall response. Thus, the response of the empirical model was not congruent with our hypothesis that this was a single IGP module. In fact, effect sizes reveal this to be the response of the system to increased supply of nutrients and energy. So the community is clearly not behaving as our hypothesized IGP module. In fact, it may be even more interesting, actually, when the predictions of a particular module model fail. In the words of Bob Holt. It's always fun to quote Bob. I enjoy quoting Bob because I agree with so many of his quotations. So, where do we go from here? What do these results suggest about future research directions? We could connect this module with other predators and prey in the soil food web, but we grouped these 40 families of microarthropods into a single community because of similarities in size, body plan, and behaviors. Thus, it might be more productive at this stage to consider what these results might reveal about the trophic connections within this module. As an example, let's focus on better defining the intermediate trophic level, which we no longer will call IG prey. Other trophic structures might be more congruent with the results of our field experiment. Diagram A in the upper left depicts the behavior of our empirical model. I want to compare this response to several predictions of Volrop and her colleagues, their mathematical model of 2 alternate energy pathways that are linked by a top generalist predator. In that model, increasing energy input to the system supports greater trophic complexity. In looking for a match between diagrams B through E to our empirical model, we're going to assume that there are groups of intermediate level prey that we fail to recognize, that we fail to distinguish. So we already saw that configuration B is not congruent with the empirical model. But what about the next configuration, the 2 linked 3 trophic level chains? Remember that we assume that C1A and C1B are 2 undetected groupings of predaceous mites that we mistakenly lump together as IG prey. In this model, their opposite responses cancel out so that the overall group would show no response to the tridal addition. But in this model, neither the top predator nor the resource increase, so this model has to be ruled out. So we eliminate that possibility. We're left with configurations D and C, which do match the behavior of our empirical model if the intermediate trophic levels in the models are collapsed into a single node. Congruence between the behavior of our empirical model and the collapsed mathematical model of more complex trophic connections suggests that it would be productive at this point to refine our knowledge of the natural history of predaceous soil mites. Additional knowledge might reveal an empirical model with more nodes that is congruent with the results of Monica's experiment. Of course, the number of possible nodes and connections is almost too many to contemplate. But before we even start that contemplation, let's return to an important assumption of this approach we've been taking. We've assumed that mathematical and empirical models have reached a new equilibrium after the resource addition. We don't have time to explore this depth of equilibrium in the ecological systems here, of course, but we can examine evidence of whether IGP modules on the forest floor show a constant structure over time in the absence of an externally imposed perturbation. So to do that, let's move to another forest floor system, again in the Chicago area, in a 240-acre nature preserve, the Swallow Cliff Nature Preserve, just about 30 minutes from downtown Chicago, or 60 minutes or more depending on traffic. I'm going to present research conducted by Robin Morris. It was just submitted to +1. Research that was made possible by a vast array of numerous undergraduate assistants, mostly volunteers. Every spring, summer, and fall for 4 years, Robin and her assistants collected spiders from leaf litter, bare ground, logs, tree trunks, and low vegetation. They collected about 14,000 spiders from 20 families from 90 different areas across the reserve. 80% came from the litter horizon and 10% from low vegetation under 1 meter. And they also collected non-spider prey, and spiders that were to be analyzed for gut contents were placed on ice and kept at -20. And also spiders and non-spider prey were collected for primer development, and they were kept alive in the lab. And to develop primers, Robin used multiplex PCR to sequence DNA from 10 spiders, at least 10 spiders from each family, and at least 10 specimens from 11 broad non-spider prey categories. And after a prey assay had been optimized and developed and optimized for particular prey, field-caught adult spiders were then analyzed for DNA from all of the potential prey in the system. Spiders were selected at random from 1 to 5 genera, with the 11 most abundant spider families to yield a total of 300 samples per family of spider. Of 3,300 analyzed spiders, 45% tested positive for prey DNA. Only 17 of these spiders tested positive for 2 prey items. These were considered individual interactions. These results were then converted into 11 times 22 interaction matrix, and because we couldn't detect within family predation by this technique, there were 11 times 21 or 231 potentially detectable predator-prey interactions. The full matrix with years and seasons pooled yields the summary food web. The middle panel illustrates consumers, the 11 spider families, and solid bars in this diagram are the cursorial spiders, and the ones with lines are the web spinning spiders. The relative width of the triangle connecting a consumer to a prey category reflects percentage of that prey in the consumer's diet. Spiders that were prey appear in the top panel, and non-spider prey are in the bottom panel. The most common families of spider prey were wolf spiders, pissards, and crab spiders among the cursorial spiders, and linniphiids among the web-building spiders. And among the non-spider prey, Diptera, Leptera, and Colembala were the most common prey. Note that about 90% of the spiders consumed by cursorial spiders were other cursorial non-web-building spiders. And about 90% of the web-spinning prey were other web-spinning spiders. Food web structure appears similar over the 4 years of the study, with seasons pooled. And this similarity is reflected by constancy in 2 indices. These are bootstrapped 95% confidence intervals for restricted connectance and interaction evenness. There also appears to be seasonal variation, minimal seasonal variation in these 2 indices. In contrast to that pattern, notice that the relative number of spider prey in summer compared to non-spider prey is much greater compared to spring or fall. So there seems to be a seasonal difference in the percent of non-spider and spider prey in the diet. Are these spiders that are prey of other spiders, are they IG prey? That is, are they part of an IGP module? They need not be. It depends upon whether the spider predator and its spider prey share in common a non-spider prey taxon. It so happens that if you look at the summary web, 99% of spider-spider predation events involved an IG predator and an IG prey. And among the non-spider prey, 97% of detected predation events were part of an IGP module. So IGP dominated this spider-focused food web. There was little variation in percent IG prey over the 4 years of the study. But if you look at the seasonal pattern, you'll see quite a difference, with almost twice as much IGP prey in the diets in the summer compared to spring and fall. And what's really interesting about this pattern is that this seasonal pattern is constant over the 4 years of the experiment. Mathematical models of food web structure and dynamics ultimately must be tested by comparing model predictions with the behavior of widely distributed natural food webs, that is, powerful empirical models. When mathematical models assume an equilibrium state, comparable empirical models should also be at equilibrium. Determining whether an ecological system is at equilibrium is, of course, a contentious issue, but nevertheless, I argue that the natural food web being compared with model predictions should exhibit a high degree of temporal constancy in structure, or if variable, this pattern of variability should be repeatable. Thus, the widely distributed spider-focused web of the forest floor is an excellent candidate for an empirical model to investigate the dynamics of IGP. And the striking advantage is its proven suitability for controlled, replicated manipulations of the detrital resource base, as well as experimental manipulations of long-term rainfall, spider densities, and densities of other arthropod predators. And these long-term perturbation experiments will be particularly powerful if they're combined with monitoring feeding connections via PCR, fatty acid analysis, stable isotope techniques, and so on. What about the soil component of this community? Constancy of effect size suggests that this system may have reached a new equilibrium, that is, in Monica's experiment, after 2 field seasons. Now, it's much more difficult to directly manipulate components of the mite-focused food web. One reason I never worked with mites directly. But it's still feasible to uncover interaction pathways with DNA and stable isotope techniques and other biochemical approaches. Thus, we must include mites, of course, in this hypothesized empirical model. Another advantage of the forest floor model is its clear vertical stratification into humic horizon, litter layer, and low understory vegetation. Many arthropods are confined, more or less, to one of these layers, but several move from one layer to the other at different rates and in different directions during development. In fact, within the layer of litter, there is stratification of spiders, as these ingenious traps made by James Wagner and Søren Toft illustrate. Thus, the forest floor, because of this clearly discernible heterogeneity, is a promising empirical model for investigating spillover effects in a very widely distributed terrestrial system. In closing, I'd like to pose this question. Do we first identify patches that might yield spillovers, and then via direct experimental manipulations study the dynamics of transfers between patches? Or do we first experiment with the entire complex heterogeneous system using techniques such as PCR, stable isotopes, etc., to infer the dynamics of spillover effects between discernible patches within the system. I don't have the answer to that question right now. Gracias por su atención.
Hola. Okay. So I would like to ask you something because I'm very curious about that. I'm sure you have much more experience than I, David. When we go to the field, we observe that actually movement and spillovers are not in a 2D framework, but rather in a 3D framework, because you have the XY axis, but also the depth. Many insects, many prey, and many predators also do migrations during the day and the night. How important do you think is the role of these axes that we not often consider?
Very important, likely.
Yeah. Okay.
In general, I would, very important.
Okay.
Thank you, David. I'm very impressed about your talk on the spider webs. Very good. Okay, let's talk. Next talk is by Marta Montserrat, Food Web Engineering, Building Community Models to Improve Biological Pest Control.
I'm sure it's the on. Ah, I could feel it. So it demands among other goals to supply to an exponentially growing population food via the implementation of sustainable and resilient agricultural practices that increase productivity, improve land and soil capacity, and strengthen agricultural systems capacity for adaptation to climate change. Well, biological control is at present one of the most sustainable and resilient agricultural practices because it basically relies on the very mechanism upon which herbivore populations are regulated in nature. Furthermore, it complies with two very important directives, international directives, one of them aiming at reducing the amount of pesticides in agricultural systems, and the other one aimed at promoting the use of native biological pest control agents for biological pest control. But there is a problem that the biological pest control has a very low rate of success. We believe that is because it basically fails the establishment of the animals that either are released in the fields. There is a big problem in the establishment rates of these animals in the crops. So what we think is that there are several reasons of this, among probably many other ones, is that biological pest control strategies assume that species engage in simple trophic chains. They ignore the effects of dynamics that may occur at the meta-community level. They ignore the effects of abiotic factors on the dynamics of communities. And because the three above can become strong forces of selection, they ignore eco-evolutionary dynamics as well. This is why back in the 2012, Jordi and I proposed food web engineering as a tool to improve the performance of natural enemies, especially those that are supposed to be released in hostile environments. We define it as an extension of biological pest control that integrates community ecology and evolutionary sciences into the management of agricultural systems potentially exposed to hostile environments. Basically, what food web engineering proposes is to exploit the inherent intraspecific genetic variability of relevant traits in biological control agents to prepare the organisms to perform better when they are introduced into these hostile environments. The idea is to do that via artificial selection when traits to be improved are known a priori, or experimental evolution when they are not known, the traits that you are interested in. The goal is indeed to go faster than the natural adaptation process that would occur at much slower pace under natural selection. And our specific goals were our scenario of interest was to test what happens when biological control agents are exposed to two factors of stress. One is the presence of a top predator, so an ecological or biotic factor, and the other exposed to an abiotic factor, which is very common, and it becomes commoner and commoner with climate change. In the literature, you can find a comprehensive list of traits that are amenable to be genetically improved via genetic breeding of BCAs. But the problem is that it is not so straightforward to know what traits to improve under specific hostile scenarios that agricultural systems might be exposed to. What we do know is that we seek for traits that hold enough genetic variability so that they are able to respond to perturbations. So we seek response traits that at the same time are also effect traits. That is, traits that affect the properties, functions, or services of ecosystems. In our case, we would be interested in the strengths of trophic cascades. So to do this is not that so straightforward, because what we need to do first is to know the genetic architecture of as many traits as possible you can evaluate in the species of your interest, and then cross them with whatever source of stress, evolutionary stress, that you are interested in. As I said, we are interested in biotic factors, specifically this one, and abiotic factors, that is an increase of temperature. Then use this data to fit Weaver, and through the simulations of Weaver, find out what combination of traits maximize trophic cascade, go back to evolution experiments to improve these species, to improve these traits in the species of interest, test them in the field, and go back and follow the same rule, the same loop, if you need to go the in silico world closer to the in vivo world. In other words, in here we found out what traits are response traits. In here we find out what of these response traits are effect traits. And in here, is this thing working? Does the animal selected works better in the field? That is actually what we are most interested in. And so in this part, I'm going to explain you basically the first step of the scheme of proposing food web engineering. And most of the talk is the work done in the PhD of Diego Serrano Carnero, who is sitting or hiding over there. And he's going to deposit the thesis this month, so we hopefully will have a new doctor soon. The system that we chose was a predatory mite, phytocyte from the phytocyte species, Ambliceus swisky, and we chose it because it's the species that is actually being used by most as a biological control agent. Is very good at controlling 3 of the most important super pests, that's the spider mites, whiteflies, and thrips. And it has very short generation times, what makes this species very amenable for evolution experiments. The first thing that Diego did when he got his PhD contract was to go to Israel because to capture a wild population of this species, because this species is originary from the Middle East. So in the lab he came back with 500 individuals, which is quite a lot. And am I yelling too much? No? Vale. He came back with, so in the lab we kept 2 populations, one wild and one commercial. Next thing we did was to create, to use the technique of isofemale lines to redistribute all the phenotypic variants of the populations across subpopulations originated from one single founder female. He managed to obtain full-grown and very well kept 20 isolines for the wild population and 22 for the commercial population. Next he did was to evaluate a total of 23 traits to all the isofemale lines of the 2 populations. And most of these traits were measured in the same very individual. So we divided this trait into life history traits, morphological traits, behavioral traits, and some other traits, physiological, that were measured in other individuals because measurement implied the death of individuals. Then what we did was to find out all the correlations between traits to know the genetic architecture of the traits in this species. In here you see the significant positive and negative correlations between traits within the same category and measured in individuals, in the same individuals, and here using the average of each isofemale line. Also positive, negative within each category and between, and I will come back later to that. The next thing that we did was to use PCA to reduce the number of variables of the number of traits to be further studied. But in this graph, you already can see that the variance of the commercial population in blue is contained within that of the wild population, both for traits measured in females and in males. Next, we estimated the intra-class correlation as a proxy of broad sense heritability using generalized linear mix models that were implemented through the MCMC-GLMM function in R. And here we see the traits that we found with significant heritable were mostly related to size, development, reproduction, and physiology. Also, it's interesting to note that when only the commercial population was considered to estimate the heritabilities, none of them were significant. And the next we did was to test, okay, now we have found out the potential response traits, but are they really response traits? So we test them following the scenario of our interest, whether traits evolve when they are exposed to two selection forces, the presence of a top predator or high temperature, so an abiotic selection force. We did experimental evolution in a two full factorial cross design with low temperature, high temperature, presence of predator or absence, and subpopulations were exposed during 10 generations to either of these treatments. After that, we moved the subpopulations to a benign environment, left them there for 2 generations to eliminate the maternal and grandmaternal effects, and after this, we measured the traits. I say we, but it's actually he who did it. Yeah. So these are the results that we found, and indeed, traits evolved, some of them. Basically, the ones related with size and the ones related with development. Indeed, egg sites here you see each of the replicates, and in dark blue is the average of all the replicates. What you see is that the size of the egg decreased with temperature, but only when the predator was present, both for eggs that yielded females or eggs that yielded males. And the same pattern was observed for the size of the females, which is measured in the dorsal shield because it's the most stable piece of the body, and it's exactly the same. They became smaller, but only at high temperatures, but only in the presence of the top predator. As for developmental time, juveniles took longer to develop at higher temperatures. Yes, both for juveniles that yield females and juveniles that yield males. So basically, we got two outputs that are quite against, or at least partially, to the temperature size rule in ectotherms. As you know, this rule says that at higher temperatures, metabolic rates increase, therefore they develop faster, but this is as a cost of becoming smaller. But we also know that there has been, even described by Atkinson itself, several cases, several exceptions to this rule. And actually the tendency is that now there are some authors that try to define or to limit the conditions upon which this rule might hold, or other authors that directly say that there is no evidence that warmer temperatures are associated with selection for smaller body sizes. But be aware that we found them. We found individuals smaller at higher temperatures, yet when acting together with another selection pressure, an ecological selection pressure. The nice thing of knowing the genetic architecture of traits is because it may contribute to explain some results for evolution experiments. For instance, in here, we found these two negative correlations that relate the size of the egg with behavior related to movement. So we can sort of say that the more active the individuals are, the smaller the eggs will produce, because it makes sense. They spend a lot of energy moving. And also we have this positive correlation, highly expected in here, that says that the smaller the eggs will produce smaller individuals. So considering that Orius, that is a top predator, is a highly visual predator, we believe that our results are a combination of an increased activity of the top predator at high temperatures, an increased activity also as well of the prey, in this case, which is also a predator, at high temperatures, and this would yield these individuals to produce smaller eggs, and a selective predation on bigger individuals if the predator just follows the optimal foraging rule. And these two clearly favoring smaller individuals over bigger ones. As for the developmental time trait, this trait, we believe that phenotypic adjustments, well, no, it's an hypothesis, phenotypic adjustments in response to changing temperatures may not be possible under all conditions because of energetic constraints. It is true that increasing temperatures increases metabolism, but there might exist a compromise between catabolism, which is highly dependent on temperature, and anabolism, which is relatively independent of temperature, in the sense that if developing faster at high temperatures requires an energy tank that is not filled by individuals when they go to acquire food, so to acquire energy, this could result in longer developmental rates. If cost of anabolism at temperatures, if there is a high cost of anabolism at high temperatures beyond the optimal temperature. So the next step in food web engineering would be, okay, now we know more or less what traits are response and towards what direction they evolve under the conditions of our interest. So the next things would be, okay, what of these response traits are also effect traits? And for that, we just got this project recently in which Dr. Odin Moron will work on Weaver to parameterize it using our data, using other data from other people, to try to respond to these questions we are interested in. As now we pass through the Spill Island. Okay, now we are dealing with traits that are related to spatial structure or to movement. So what we are interested to, we are doing in the lab is the artificial selection on the trait dispersal propensity. We try to create two lines. The reason of doing that is because if we create these two lines, we want to test them against resource islands of different sizes and cross with different distributions of the resource islands inside the greenhouses to see what different effect on the trophic cascades each combination has, or to find the optimal combination that would result in a better biological control, which is of pests. It is our goal. Don't forget this. So to do this, we use this type of experimental setup. As you can see, it's composed of 10 cells that are connected via this bridge, which is aimed at this direction, but make very difficult the other one. In the strain with high dispersal, what we do is to put 200 individuals here with plenty of food. And after a certain time, we collect the ones that are found at the farthest away patches from the patch with food. Therefore, we obtain individuals that migrate from the initial patch despite this has abundant food. Parallelly, we run a replica control in which these individuals, of course, are taken to the growing phase to be used in the next run. And parallelly, we have a control situation in which all the individuals that are introduced here at each round come always from the outbred population. So our expectations would be at the initial rounds to find most of the individuals close to the patch with food, and at the end of the selection, if it works, we should find in this treatment, in the selected treatment, more individuals farther from the patch with food, but not in the control. For the low dispersal strain, we do the same, but a little bit different. We don't put food in here, and what we do is to collect, after we introduce 200 individuals, and after a certain time, we collect the individuals that remain in the nearest patch, the 50% of the 25% of the individuals, that they are found in the nearest patches close to the patch where they were relieved. The idea is that we obtain individuals that don't migrate even though there is no food, so that they have low dispersal rate. Our initial expectations would be to find at the initial rounds most of the individuals far away because they are supposed to go foraging or try to find food, and at the final rounds to find most of the individuals in here or close to here in the selected population, but not in the control. Here is some pictures of Diego either preparing the arenas or doing the experiment, but I also show it for you to realize how the size of the experimental arena. It's quite big. It's 1 meter long, And here the results. Okay, what you see here is, in this axis is the experimental setup. So patch 1 is the one with food, because we are now showing you the high dispersal rate. Then, in here, you see the expectations during initial rounds. We have 11 rounds so far, and the expectations during the final rounds. And what you see in the graph is the average of the median of the distribution of the individuals across all the patches. Yeah, and then it's important to say that we are still in the process of kind of fine-tuning the experimental setup, because as you will see, we found that some problems. First, oh yeah, it really worked. Look, they are all here. But yeah, after 6 selections, Jesus, they are still here? So until Diego realized that most of the animals were hiding on the shelters that we had added on top of, we had added on each of the cells. So they were very cozy there and they were not moving. So removal of the shelters. Now they start moving. Okay. There is some sort of weird dynamics in here, and we reach the end in which, yeah, it looks like if there is some difference, but there is a big problem that also the control population is moving when they were supposed not to move. So we think that maybe there is a kind of follow the leader behavior, that one moves and all the rest go behind. But we'll see. If you have any idea on how to improve this setup, it would be very welcome, actually. But anyway, we continue with the selection, and even more now that we start seeing some sort of divergence. The low dispersal rate, the structure of the figure is the same. And in here, we were expecting at the initial rounds, all the individuals move, and again, they were not moving, so we removed the setup, and now we found that we found a situation that is highly similar to this one. So we have the feeling that we now found the experimental setup that actually drive us or has directed us to the expectations that we need for the initial rounds. So again, we will continue and see. But this is also for you to realize that are not easy to select traits that are based on behavior. It's not so straightforward like selecting for becoming resistant to a pesticide, for example. And last, whoa, it's great. Last, just to show you the people that is involved in here, that would be Jordi, of course, Sara Magalhães from the University of Lisbon, Diego Serrano, the PhD student, soon-to-be doctor, and myself. And thank you very much for your attention.
3 minutes for questions. Okay.
I'm just wondering, so what are your thoughts about the trade-offs between the traits that you select and other traits? How that may
I think I prefer the micros, if you don't mind.
Thank you for a very nice talk. I was just wondering, so when you select for some of these traits in your predators in the lab, do you think there might be some trade-offs with other traits?
Yeah, there are
that
lots.
may
all the negative correlations are potential trade-offs, and we
Right,
found
that-
a lot.
that may influence
Yeah,
their performance in a
that-
field?
That's why one of the part of the message that to know the genetic structure can really help to understand what things happen and why. So it's a really important path.
Thank you.
Marta,
Hola.
thank you for your very interesting talk. I don't know if, I'm not sure I've understood your experiment. You start some population with just one female founder, right?
Female.
Female. And after that, you have a maternal population.
No, this is to create isolines. So to reduce its subpopulations, that you aim at reduce the variability within the population and try to increase the distance between isofemale lines. And this is a technique you use to calculate things like correlations, like heritabilities.
And to look for the response traits, what did you do? You have those populations and you
Yeah.
submit those populations to different environmental conditions or whatever? Or?
Yes, well, first of all, the traits that show that are heritable are supposedly candidates to be response traits. Then what you do is to just measure how they change, how traits change when you expose the populations to the selection force or that you are interested in, and we found that the trait changed.
Okay.
So that indicates that it's a response trait. It responded to a selection, to an environmental pressure.
And you estimate the response when you change the population to different environments, right? For example, different temperatures.
Yes.
Or present or absent of predators, or
yes,
something like that.
It was a full design.
Okay,
design.
but then I think that what you are looking for, what you are finding is phenotypic plasticity for those traits. That is also very important. Then no, because you are changing the same individual between different environments, and how those traits are expressed or not. Because
it's populations. This is the whole population.
It's experimental evolution, so it's through time,
I know.
and then there are two, three generations to remove any maternal effects, any false plastic effects, and you see the final response that is evolutionary and genetic. It's the isoline technique. That is based.
Okay.
Anyway, we can talk about it because I'm interested in
Yeah.
what you have to say about it. Very much.
I had a related question. Fantastic talk, though, Marta. For those evolutionary steps, did you rear those individually, or did you keep them as populations?
Population.
So did you fix density?
Sorry?
Did you fix population density?
Yeah, we always start with the same number of individuals at each round, or at least the closer individuals that you can reach to. Because of course when the selection is, for instance, with the presence of the predator, sometimes predation pressure was extremely high. So we also have to find how to reduce this pressure so that you can get enough individuals to start the next round.
So each generation.
And sometimes it's the whole individuals that are in the population after the first round. Then you remove everything except you keep all the individuals. And you put them again in a new, completely equal setup.
Cool. Okay, that was.
Yeah, it's not that you leave them there. No. You have, at each round, you create the environment as similar as possible to the previous one and to the next one.
Right. Okay. Cool. Thank you.
Okay. Sorry. Do you hear me? Yeah? No? Thank you, Jordi. I represent the practical point of view of this meeting. Well, I work at the Andalusian Institute of Agricultural and Fisheries Research and Training, IFAPA, for its acronym in Spanish. IFAPA is an organism dependent of the regional government, La Junta de Andalucía, from Spain. And our objective is to offer solutions to resolve problems in the agricultural, fisheries, and agri-food sectors. To achieve this, we work on three main different areas. Applied research, we develop a finalist project, control pest in my case. And knowledge transfer and training. We organize many technical meetings and training courses for farmers and technicians throughout the years. This is my research center, La Mojonera. It's located in the middle of the plastic sea. We are here. We are here, and my research center is here. And obviously, it's focused on horticultural protected crops because it's the main agri-food sector for our province. In particular, I work in integrated pest management and biological control of pests that attacks these crops. Well, currently we know that agriculture is a major use of land. It's estimated that agriculture covers about 38% of the global land surface. About 1/3 of this is used as cropland, while the remaining 2/3 is used for grazing livestock. Unfortunately, we are also facing a significant losses in our natural habitats. It's estimated that about 70% of our forests are fragmented. But it's not just that agriculture covers a lot of land, it's that the current model of agriculture intensification is based on agrochemical inputs, large monocultures, and landscape homogenization. Certainly, this type of agriculture has successfully increased yields. However, it's also responsible for negative environmental impacts in our ecosystems. Over the past several decades, there are many works showing that intensive agriculture exacerbated by climate change are associated with losses in insect biodiversity and the global insect decline, and also with important ecosystem services for agriculture and linked to insects, such as biological control or pollination. In Europe, agricultural policies that are from farm to fork and the European biodiversity strategy are in line with the European Green Deal to mitigate or reduce climate change. From farm to fork strategy aims to accelerate our transition to sustainable food system, and we have to reduce insecticides by 30%, fertilizers by 20%, and increase organic farming by 25%, all of this by 2030. And the European biodiversity strategy aims to reverse these losses in biodiversity by increasing 10% of our agricultural land dedicated to non-crop habitats. So the final goal is to reinforce these important ecosystem services. In this context, certified organic farming is often claimed to be the fundamental alternative to conventional farming. Certified organic farming in Europe is a type of agriculture where synthetic agrochemicals are prohibited and where some practices are recommended, such as green manure, crop rotation, mechanical weeding instead of herbicide use, or restoration of natural habitats. But note that all these practices are recommended, they are not mandatory. And it's also important to me to remind you that natural pesticides are allowed in certified organic farming. And sometimes natural pesticides are dangerous as synthetic pesticides for insects. So the contribution of this type of agriculture to stop losses in biodiversity could be overvalued. In fact, switching from conventional to organic practices increase local species richness by just 1/3. However, it leads to considerable yield losses, so more land is needed to produce the same amount of food. So maybe 35 organic farming is not enough. If we really want to stop or to reverse these current trends in agriculture, we need a paradigm shift in agriculture. We have to redesign our farming system and our agricultural landscape. Surprisingly, there are many measures, environmentally friendly measures, that can be implemented even in very conventional farming systems. For example, land sharing or land sparing perspective. Land sharing perspective focuses on conserving non-crop habitats inside the agricultural matrix for biodiversity. This perspective favors those species that are well adapted to agriculture and promotes the spillover of ecosystem services to crops. On the other hand, land sparing promotes large, well-conserved and separated areas from intensified agriculture. Land sparing is the only way for in situ conservation of those species that require undisturbed habitats and are incompatible with agriculture in a land sharing context. So these areas could act as source habitats through spillover effect conservation. The last studies are showing that both perspectives are not mutually exclusive, both are needed and should be combined to reach a multifunctional ecosystem. So how can we implement and how can we transfer all this nice theory to an artificially system like plastic greenhouse agriculture in Almeria is really challenging, and is one of our objectives from Monica and mine for the past 10 years. So it's very exciting for us to be part of this project and this symposium, because we feel that all these results allows us to optimize the use of biodiversity for pest control and transfer this knowledge quickly and efficiently. So thank you very much, Jordi. The province of Almeria hosts the largest concentration of greenhouses in Europe and one of the largest in the world. More than 3.5 million tons of vegetables are produced on nearly 33,000 hectares of greenhouses. About 12.5 of this surface is under organic production, and the majority of this production, 75%, is exported to countries in Central and Northern Europe, mainly Germany, France, or Holland. The promotion of this intensive horticulture has resulted in very significant social and economical benefits for our province. However, having also important negative impacts in native biodiversity and our natural resources. In this slide I show you on the right here the transformation of our landscape since later 70s until now, and the destruction of important semi-arid endemic habitats unique in Europe. On the left, I've marked in red the few remaining patches or the few remaining plots harboring these specific habitats. From a pest control point of view, since early 60s, there are many works showing that agricultural landscape simplification reduces natural pest control, increased pest outbreaks and insecticides use. And as seen in this picture, Almeria is the best example of agricultural landscape simplification, and indeed, this system suffers a high pressure of pests. But even inside the greenhouses, there is a high simplification. The specific greenhouse cultivation conditions favor the proliferation of pests. I mean, good temperature, humidity, all the plants are genetically identical, and so on. Therefore, many pests attack horticultural crops, but I highlight two of them, whitefly and thrips, because both are present in all protected crops, and both act as vectors of plant viruses, which are very aggressive to certain crops. The high incidence of these pests has been controlled in the past by means of chemical treatments, and as expected, these pests developed quickly resistant to these insecticides. So this unsustainable situation continued like this until 2007, when many farmers started to adopt biological control as a main tool for pest control. 15 years after, the situation has completely changed, as seen in this figure, and in the upper line, the surface of greenhouses using biological control has continuously increasing, reaching 52.5% of the total surface. In fact, the market value of biological control in Almeria is about 30 million euros, which is more of the total market in the rest of Europe as a whole. Here, only I show you some of the main arthropods that farmers use in their greenhouses. Farmers purchase these organisms in biofactories and release them inside their greenhouses. They are mainly predatory mites and parasitic wasps, anthochorids, mirids, and also hoverflies, ladybirds, and lacewing. Since 2007, and with the reduction of insecticides, the abundance and diversity of spontaneous insects colonizing crops and assisting farmers in pest control are becoming increasing. Here I show you some example of this. So, although our natural ecosystems are degraded or are fragmented, some ecosystem services like natural pest control seems to be active, seems to remain active. So we think we have to show it and to promote it. And this biodiversity is not only evident with insects, it's also evident with vertebrates, especially in the case of reptiles. This is the big lizard. And also with birds. So based on all mentioned before, we developed the following hypothesis that I summarize in this slide. On the left, I show you the current situation and agricultural matrix of greenhouses supporting a high incidence of pests and plant viruses. And we propose, on the right, I show you the future strategy, where we propose to conserve and restore the few remaining semi-natural habitats and connected them with the new establishment of hedgerows. These hedgerows could act as phytosanitary barriers and should be composed of native species and specifically designed for natural enemies rather than pests. We have been working on the new establishment of hedgerow for the past 10 years, but we hope to start working on landscape scale in coming months with a new project. So I summarize the result of this point. In this slide, I summarize our background. The first step was to know the role of native plants as reservoirs of these plant viruses. So we collected more than 300 samples belonging to 28 different plant species and 20 botanical families in different places of Almeria, where are greenhouses. Analyzed them by immunological and molecular techniques for the presence of the 10 most harmful viruses that attack crops. Here is an image with some result. This is the positive control. This is the sample, negative sample. So the results show us that none of the native species analyzed was naturally infected with these plant viruses. So apparently, pests prefer to feed on weeds or horticultural crops rather than on native vegetation. So we propose the substitution of these weeds that are growing in the surrounding greenhouses and which are frequently removed with herbicides with small native grass species. So the next step was to select the best candidate species for design these hedgerows. Accordingly to the bibliography, we established the following plant selection criteria. For example, using always native species, uses species commercially available in forest and nurseries, uses shrubs rather than weeds or trees, uses species combine over their overlapping bloom periods, combine nectar and pollen-rich species, use some species belonging to the Apiaceae family, and use species offering microclimate, alternative prey, or shelter. Finally, we decide to select 22 plant species belonging to 13 botanical families. I show you a nice picture of them. Each species fits 1, 2, or some criteria. And with all these species, we designed and established a field experiment, again in the middle of the plastic sea, in the experimental station of Cajamar. This is a nice aerial photo of this area. And as seen in this picture below, 5 years after, we realized that we have the ability to change the landscape. Once these plants were well established, we sampled them for insect abundance, mostly for 1 year. So the next step was to know how these pests and their natural enemies were using these plant species. In summary, in this figure, I show you the abundance of these pests in each plant species. Each bar corresponds with one plant species. We found that native plant species supporting low, medium pest levels, suggesting, again, that pests prefer to feed and reproduce on crops rather than on native plants. Regarding specific natural enemies, we evaluated the abundance of one specific predator and parasitoid of each pest. And as seen in this table, the best model shows us that the presence of prey rather than floral resource was better predictor for the unnatural enemies abundance. I mean, the specific natural enemies were collected also in those plants that also supported the high pest abundance, suggesting that there was a potential biological control of the pests. We also evaluated the abundance of some generalist natural enemies, in particular web weaver and hunter spider in these plants. And again, as seen in the table, we found a positive relationship between spiders and pest abundance. The positive relationships was higher for whitefly than for thrips, And also we found a positive relationship between web weavers and the adult mobile stages of the pest, of whitefly, and a positive relationships between the hunter spider and the nymphal immobile stage of the same pest, suggesting that there was complementarity among these spiders. The next step is to demonstrate and quantify biological control in this ecological infrastructure. So to achieve this, we use sentinel plants, in particular cereal sentinel plants, which are inoculated with a specific aphid, cereal aphid, which attacks cereal but not attack horticultural crops. And we put these plants on open air and therefore expose it to the action of natural enemies that naturally occurring in the landscape. And some plants we put inside these insect catches and therefore excluded from the action of natural enemies. And in summary, we found that there were four more aphids in sentinel plants excluded from the action of natural enemies that one exposes to them. These results for us show that natural pest control even occurs in very simplified agroecosystems like greenhouses in Almeria. Last but not least, I like to point out our work on knowledge transfer. Here I summarize some items regarding transfer, training, digitalization, information, and governance. But from all of this, I like to point out this web design. We developed this website for any user, any farmer, any technician can design biodiversity and hedgerows among greenhouses. Well, the cost of installing these hedgerows is not expensive. In fact, it represents less than 1% of the total cost of an agricultural holding. So as seen in this figure, the proposal has been well received by farmers, but and we calculate that there are more than 200 kilometers of hedgerows established in Almeria now. But to be honest, we expected that the proposal would be results in more hedgerows, and which would be more successful. And in conversation with farmers and technicians, they say to us that farmers recognize the benefits of biodiversity for pest control. So they decide to introduce all this biodiversity inside their greenhouses. So the benefits of biocontrol and biodiversity is for themselves, not for everybody. So this is actually the current situation in Almeria in the greenhouses. Biodiversity is increasing, but much more inside than outside of greenhouses. So more questions are needed to be answered. No? Ideally, we should to combine outside biodiversity outside and inside, but we have to need how many islands of biodiversity, density, how many plants, how many species for island to optimize this biological control. So we hope that the Spill Island can allow us to answer this question on how to manage this biodiversity inside of greenhouses. Thank you so much for your attention.
Thanks very much. That was really interesting. Have you looked at any other benefits apart from biological control? I'm thinking specifically, I saw a syrphid there, syrphid fly. Have you looked at pollination of tomatoes or anything like that?
No, not yet. No, not yet.
And another benefit, there was someone in New Zealand who looked at something similar in pastures, planting native plants for a totally different reason about soaking up nutrients. But they found one of the biggest benefits was in staff retention, that employees wanted to work in a nice environment. So there might actually...
I understand well, I think.
No entendiste?
Sorry, can you?
Hubo un estudio en Nueva Zelanda donde estaban mirando en pastos los beneficios de biodiversidad y uno de los mayores fue la retención de empleados en el pasto que disfrutaban más del ambiente más lindo con plantas nativas. Así que es
Sí, sí, lo hemos
una idea.
estudiado. If we studied. Yes. Yes. Aesthetic, yeah, sure. I think that is one of the things that farmers recognize, that the benefits is only the landscape, aesthetic, ecosystem services, yes, sure, because our landscape is very dirty with a lot of residuals, so yes, sure, they recognize these benefits, and it's really nice to see insects and flowering, yeah, sure. But we didn't measure it.
Cool. Thank you.
Thank you for the nice talk. Have you considered to use this activity also to conserve some specific plant species? I
Yes.
mean, to introduce them if they
Yeah.
are threatened in the...
For example, some endangered species. For example, we think much of them think that the only way to conserve Mytenus senegalensis, is the species endangered in Europe, is the only solution to conserve this species. Or maybe with I don't know, I don't remember the name, but the bog.
A buxus.
Buxus, but I think that the only way to conserve Mytenus senegalensis is this, to show farmers that they can receive some benefits from the habitat, not only the species, the habitat that contain these species.
Okay, yeah, sure. Thank you.
E, ei, a.
Hello. So we are starting more than 30 minutes late, but we were a lot of people, and we had to have lunch, so organization. It's a pleasure to me to introduce the talk of Mónica González, who's going to talk about using functional biodiversity as a new tool in IPM programs. She works in Agrobio, in a company in Almeria.
And all of you for being here. It's not the best hour to have a talk, so I try to be a little bit, I don't know the word. For me, it's an honor to be here and to learn about you, because it's necessary to improve your knowledge, to achieve more functional biodiversity as a new tool in IPM programs. Let me introduce briefly Agrobio. It's a factory, biofactory, located in the so-called Plastic Sea in Almeria, and we provide services of biopollination and biocontrol in more than 40 countries. We produce 700,000 bumblebee hives of different species of bumblebees, and also insects, the most important natural enemies. Most of the principal mite predations, as well as meated bugs, anthochorics, and some parasitoids, for more than 50,000 hectares of crops. We are focused on greenhouse crops, but also we are spreading our company to other outdoor crops, also vegetables or fruit trees. And as Estefania and Marta pointed out, intensive farming systems have led to a fragmentation and simplification agricultural landscape, making crops weaker and more vulnerable to pest and disease. Also, recent scientific publications show the massive loss of isopod biodiversity as a perverse effect of this intensification. The negative effects of the biodiversity loss have not been apparent until now, but is faced with today's agriculture is faced with new phytosanitary alerts, with new pests and diseases that have no effective control till now and seriously threaten our crops. And certainly, one of the reasons maybe of this situation is that intensification system go against the agroecological theory, which states that cultural practices that tend to diversify the habitat toward the use of wind-based crop rotations will increase the presence of natural enemies and its diversity, so it provides an additional control of the phytophagous insects. On the other hand, consumers are more aware about where their food comes from and are responding, asking for and demanding products with zero risk. And also, every day, less products, phytosanitary products, are registered, and we are suffering the resistance of the pest to the pesticides. So, in this scenario, biological control is becoming a trending topic. Greenhouse horticultural in Almeria experiences a paradigm shift, as Estefania explained a few years ago. And in this sense, biological control has been successfully implemented in many protected horticultural crops, becoming a key tool in greenhouse IPM programs since 2007. And being considered a model for other agricultural systems, because this change was too quick, only in 1 or 2 years. Nevertheless, as I said, every year new and old-known pests are becoming more difficult to control using inoculative biological control, as Marta also said. In the case of intensive outdoor crops, it's not easy to develop an effective inoculative biological control strategies. These crops are not able to provide all the natural enemies the resources they need. In these crops, the main challenge is to maintain them, the release natural enemies in the orchard and prevent their escape or death. And we are also talking about hectares of the same crop. It is clear that inoculative biological control protocols are not sufficient for success in intensive agricultural systems. Prevention and low pest pressure are essential. Moreover, effective biological control requires an abundance of different types of natural enemies and their arrival at the right time. And this is a big challenge. In this sense, agroecological management of the orchard will ensure this purpose. And is independent of organic crop systems. Is the management independent of its conventional or not. Therefore, we believe that biological control by conservation is an essential complement to enhance inoculative biological control in any IPM program. For Agrobio, the company where I'm working now, improving biocontrol strategies effectiveness by promoting ecological intensification through the establishment of green infrastructures is a key issue to increase the abundance and diversity of natural enemies. We propose the use of different types of ecological infrastructures based on native vegetation. All of them are complementary in their uses and the purpose. We propose the hedgerows establishment, biodiversity islands, and cover crops when it's possible. Since 2010, we have been investigating, Stephanie and I and many other people, how biodiversity can contribute to make more stable agroecosystem from the pest control perspective. In this sense, functional biodiversity is the key question, and Stefania's work is milestone because the identification of that attractive plants to the natural enemies is crucial to design functional ecological infrastructures. We propose hedge as a permanent ecological infrastructures composed of shrubs of different species and different size and structures. This infrastructure usually is planted around the perimeter of the plot and act as a barrier and a highway to interconnect the permanent the natural patches that still remains in the landscape, interconnecting them and inviting the native natural enemies to colonize the agroecosystem and act as a barrier, as it's our desire, as Jordi shows in the first talk. It's like a mid-term investment. but as we are aware that the functionality of this kind of ecological infrastructures depends on its ability to look like an habitat, and it's really difficult to design one because this is the reason that I'm feeling so happy, because it's complex and needs to be well designed, the sufficient complexity to be balanced, but not too difficult to implement in the field. So we try to meet different criteria to make them useful for this purpose, and to provide all the resources that natural enemies can't find in the agroecosystem. Provide them shelter, food, whatever. The work that Stefania and I made during the last 12 years make growers be aware of the usefulness of this kind of vegetation, and they are very innovative, and now we are in the point to advise them and make sense in the design of hedgerow, because they interpret that it looks like a garden hedgerow, and it's not. So here you can see some hedge that you can find in the field, made by only one species, an exotic one or ornamental ones with flowers. They understand, wow, we have flowers, it's okay, but not, because these plants only attract pests. And this one is making problems to the growers in the future, because they have to prune, and they are not gardeners, they are growers. And this kind of well done and well designed ecological infrastructures are autonomous and stable, and that don't need management, and it's a place for the native natural enemies. Don't disturb them. So in AgroBio, in my job, I have 1 hour and a year working in AgroBio. We try to advise and design customized hedgerow depending on the space, because lack of space is main problem, but we offer different kind of plants also, and facilitate the access of its plant, and try to help them in this question. But I'm going to change for the really useful biodiversity islands, that is the infrastructure that make efficient, make possible an efficient inoculative biological control. These are really fundamental, because as bigger the orchard is, as more important these little islands are, to maintain the natural enemies, the released ones, and also the native ones. Their purpose is to improve biological control, so the conservation, and by encouraging the presence of the released natural ones, as well as the non-commercial enemies from outside, in inviting them to colonize, maybe also from the hedge, the inner parts of your orchards. Also favoring the preventive and early establishment of the auxiliaries in the crops inside the greenhouse, also outside, and allow the conservation of natural enemies in the absence of pests, as they provide the alternative prey or hosts. So for us, it's really crucial to use this kind of plants. We propose the combination of 2-3 small species of shrubs that we can insert in the plot, providing more or less, maybe after this symposium I will change my mind, between 18 and 27 little islands for 1 hectare placed in that places at the beginning or end of the tree lines, or in the perimeter, the corridors. The question is to interconnect them, and any natural enemy, the release or active one, can find a resource at least no more than 50 minutes, make them more efficient. They don't have the natural enemies to spend most of their energy flying or moving, but preying, reproducing, or what else. The selection of these plants, of course, is the same, with the same criteria than hedge. So, and also in the greenhouse, it's difficult to select the plants because we are always working with native shrubs, and not all these plants support the greenhouse conditions, lack of light or very high humidity. So we have to make a good selection of this plant, and also because these kind of agroecosystems are so simple that any plant you introduce can change the balance. And the last one is cover crops that are also fundamental part of biological control strategies in outdoor crops, because they integrate the soil as indispensable component of this agroecosystem. They support different natural enemies and complementary ones than the hedge, but we have difficult challenge to work with, because we are in the Mediterranean basin with a little rainfall, so it's difficult to establish them. They should have low crop competence for the resources, and they should be attractive for the fauna, auxiliary fauna, and compatible with the crop management. There are many challenges, but we are working in some projects to select a good combination of seed mix to apply for this purpose, and we are looking for rustic and native species of seeds that can grow in dry conditions, herbaceous character of annual plants, with reseeding capacity, low size for not need tillage or something, low vegetative development, so on. And we are in that point. And this is my last slide. It's a reflection. We have many natural enemies that we can integrate in our crops by means of innovative and resilient strategies. This is for really modern growers, and I think it's the future. So thank you so much.
Thanks, Monica, for the talk. I have a question regarding the managing practices. So when you sell natural enemies to the greenhouses, do you put several species of predators within the same greenhouse? And this is because you try, or growers try to find the complementarity of having different predators for a given pest, or they select one predator for a pest, another predator for another pest?
They don't decide. They prefer to use only one or two natural enemies, but our job is advising them and introduce more natural enemies to have different niche. So our work every day is to make them understand that for the aphids, for example, it's not worth it to use only Aphidius colemani is the natural enemy they mainly known, but you need different kind of predators, so you have to integrate them with this kind of ecological infrastructures to be a little bit more successful. Aphids till now are impossible to manage, so now they are understanding that you need more. But they don't decide.
Can I make another quick question? So, have you noticed whether the pests are starting to have resistance to the predators that you are putting in? So, I can imagine as a red queen hypothesis, you introduce a predator with a given genetic variance, and insects, they evolve fast, and pests are becoming adapted to the predators, and you create somehow a red queen hypothesis.
Yeah, the use of only Aphidius colemani is making aphids to escape from parasitism, so it's really important. It's a small system, so it's necessary to study this and introduce and combine different kind of predators, different kind of parasitoids to balance and look for someone who at the end control the pest. I don't know if I have answered your question, but we are in that point right now. After 9-10 years working with biological control, we are now in the point to continue advising and make them understand that with 2 or 3 natural Swirsky, Aureus, and Aphidius colemani is not enough.
Thank you very much. I've got a couple of comments and questions. So the first is a suggestion, then something in response to the nice question you just got. So you were talking about diversity islands and the row crops and so forth, or row plants. You know, you ought to be thinking about doing experiments, basically, because there's a lot of uncertainties in all of this. And it's nice to think about what is the optimal way of doing something. But there's a big error bar around that. And I think that one thing that's often missed in applied ecology is the need to do controlled, replicated experiments where you try out different things and try to really understand what's going on, treating your applied mission as an opportunity also to get more knowledge about the world. And you have an opportunity, it seems to me, to do that here, okay? The people who are making money out of this, the agriculturists don't like it. They want to have an answer.
Yeah.
But pragmatically, we need to have a plethora of answers for different situations, and it's hard to determine those in advance just by thinking about things. You really need to do experiments. And then in response to this, I had some papers with Mike Hochberg at Montpellier some years ago in ecology, pointing out that when biological control is successful, it doesn't break down because of evolution of resistance by the target species nearly as often as the evolution of resistance to chemical control, okay? And there are a variety of reasons that might happen. And one advantage of having a diversity of natural enemies is it makes it harder for the target species to adapt to multiple selective challenges simultaneously. So there's a really important evolutionary question at play here about how you design the community of natural enemies to minimize evolutionary responses to avoid control by the target species.
Yeah. So that's why I'm so happy to listen to you, because I have to take into account all of these things, but the growers need an answer. So we are starting. I know the importance of all of these questions, but I try to do my best and learn and take notes and, of course, observe. And here we should adapt in the future to the coming problems. But the question is, like this, everything is lost. So we'll see. Thank you. Thank you for you.
Yeah, one short question.
I had two long questions, but I'll try.
But we have tomorrow to discuss.
Well, maybe one I can summarize as a comment. I think in relation to what Bob's talking about. Have you thought about bringing in pests from the greenhouse, like aphids from the greenhouse, into your cultures to grow them?
No.
Because what's happening is you have enemies that are not that are always encountering the same aphids in the culture, but the aphids in the field are able to evolve. So you're putting your enemies.
When we started this study 12 years ago, in our hedgerow, we found lots of herbivores, aphid, different kind of aphid, because most of this plant has their own aphid. Yeah. So they also is alternative prey. So we are not introducing pests in the hedgerow to improve the key natural enemies, but at the end they arrived, and we also have a work that found that a parasitoid for Liriomytha, a pest in the greenhouse, stay during summer in one or two plants, Thymus species. During the season, when the crop is inside the greenhouse, they go into the greenhouse to parasite the pest. But we don't know where they are in this shrubs because no pests were there. So many things are in the table to be done.
Cool. Thank you.
So now we go on to the next talk by Jose Maria Gomez from this institute, who's going to talk about modeling ecological interactions, a Heraclitean view.
Now? Okay. Well, I would like to thank Jordi and the organizer for inviting me to stay here in this really very interesting symposium, and for giving me the opportunity to talk about very strange things today. I hope you are going to enjoy. Well, my idea with this talk is to show you the way I think ecological interaction used to work in nature. This is based in a framework that we are developing, and together with the framework, we are developing also a modeling framework or modeling approach to cover this framework. Then, everybody knows what is a biotic interaction. We have talked a lot about biotic interaction today here. It's any relationship between different species or individual of different species which affect the fitness or the population growth of the species that are engaged in those relationships. In general, biotic interactions are defined by the direction in which they affect the interactor. And this is this is the classical grid that is used in most ecology textbooks to classify or identify different type of interaction. Mutual imps, when the effect on both species is positive, commensally, when this is 0 for one and positive for the other. Antagonists, when it's negative and positive, like for example predation. The most interesting interaction we are talking about today. or a mensalin, where is a negative for one of the species involved in the relationship, and 0. This static, fixed, idealistic Platonic view has been challenged in the last decade, and today most theoretical and empirical ecologists acknowledge that interaction can change between them, can flow from one to the other. In fact, there are many proposals to change the interaction grid for something more like an interaction compass, like we see here, in which you can move from one type of the other, one type of interaction to another type of interaction, just like in a compass, moving a little bit here the degrees and so on. You still have the different type of interaction, but instead of being a grid that they are just like something that they cannot move from one space to the other, they can move from one space to the other. Okay. Two pieces of evidences has been used to change a little bit our view of how interaction behave in nature. One is something that is so-called the mutualism-antagonism continuum. Is something that started to be developed in some symbiotic interaction, like for example, mycorrhizas, but has been observed also in some non-symbiotic interaction, like for example, this here that is a seed zoochory or seed dispersal by seed predators on oak here in Spain. Basically, this framework, this view, what means, or we try to say, is that you can locate any interaction between a pair of species belonging to a specific type of interaction along a gradient running from very positive, in this case, to very negative, to mutualisms, to antagonists. Mycorrhiza is a typical example in which just changing a little bit the condition, the species, the variety of the fungus, you can have a fungus that is positive for the plant, or a fungus that is, or a strain of fungus that is very negative for the plant. Here is exactly the same. The effect that different type of seed consumer have on the seed dispersal success of one plant can change from very, very negative, behaving mostly as seed predator, to very, very positive, behaving mostly as seed dispersal, like for example these two species of corvids. Another piece of evidence used to challenge our static view of different type of interaction is what is so-called the context dependency of interaction. Basically, what this view states is that the sign of many interactions may change when the environmental condition also change. You again are still within the idea that there are different types of interaction, mutualisms, antagonisms, commensalisms, but one specific pairwise interaction may change between each type, just changing a little bit. For example, the spatial condition, moving from one place to another, the temporal condition is moving from 1 year to another year, by abiotic factors, changing, for example, temperature, humidity, rainfall, wherever, or even by indirect effect, or as is also called the community context. And this is, for example, is a very good review published in 2014, in which this author just account the number of signs changes in these three big type of interaction, depending on these 4 factors of community of context dependency. And we can observe that in many cases this is the number of specific interaction, including this in this review. You can change that for example, factor like abiotic factor, predation can change of sign in more than 60% of the example of cases. Okay, these are the two views to explain why we had to move from a grid to a compass view. But if you observe these two explanations, still consider that interactions are different depending on the sign of the interaction. What I'm going to show here and propose here is a very personal view of how interaction can change without assuming that there is a predetermined prefix sign in the interaction. And this framework is based in a long tradition of gathering empirical evidence by a huge community of experimental ecologists. And it's very naturalistic. It's based in the natural history of the interaction, and this is really a very easy to understand framework. There are several points in this framework. I'm going to explain one by one and trying to justify each one. This is the conceptual framework in which is based my approach to the modeling of ecological interaction. The first idea in this framework is that all biotic interaction can be visualized as processes where individuals of different species make contact, encounter, and interact, giving to the other species a kind of service, a kind of resource, or just be killed by the other species. This is what we call interaction event. Then we can divide all the interaction in many different interaction events. And the combination of all the interaction events between one species to the other is what is going to explain the total effect of this species to the other species. Even although the interactions are bidirectional, most of the effects are unidirectional. We can divide the bidirectional outcome of one interaction into unidirectional effect of one species to the other and the other species to the first one. Now, this is very important then later to understand most of the results we have obtained. The second point of this framework is just trying to summarize what we have observed, all of us, in nature or reading the literature on natural history of ecological interaction, is that each interaction event entails an immediate effect on at least one of the interacting organisms. That is something that we call immediate outcome. This is the immediate outcome of the interaction. It's not the fitness effect of the interaction, that we call that individual effect. It's not the population effect of the interaction or population outcome of the interaction that we call population effect. It's the immediate effect that one individual, after one encounter, have in another individual of the other species. And this effect can be positive. It can just provide a service, like, for example, a pollinator moving pollen from one plant to the other, or one bird moving a seed from the mother plant to the landscape, or one symbiont just helping to fix nitrogen for one specific plant, or can be negative, like, for example, in the picture that I'm showing you, just one species that is killed by another species and proceed to eat in this, for example, hunting set. Well, so far, what we have used in this framework is something that is common sense. But something that is new in the framework, and something that I think that is happening in most of the interaction, is that unidirectional interaction even may have two possible immediate outcomes for the same pair of species, positive and negative. It's something that I call dual effect. Like, from my point of view, in most cases, two species that are interacting may have two different immediate outcomes. One is positive and the other is negative. In some cases, in some very extreme cases, like, for example, the predation between one lion and the buffalo, most of you will think that probably all the interaction events are going to have just negative immediate outcome. But in many cases, like, for example, this case here, the interaction that Helicobacter pylori have with us, this species has many immediate positive and negative outcome on us. This dual effect is very important to understand how we have modeled the interaction. Just a very preliminary review of the literature, we can observe that many interactions that we are, in most cases, thinking that the immediate outcome are always of the same sign, can also have dual effect. For example, this is a non-exhaustive list of interspecific interaction sharing positive and negative event, immediate outcome. Here, this is the color means how we ecologists classify the interaction. Red antagonisms, in this green commensalims, a brown competition, in this blue mutualism. For example, here in predation, in some cases, in most cases, the immediate outcome is positive for the predator. It's unidirectional, interaction outcome. But in some cases, the prey can kill also the predator. That means that in some cases, for a predator, the interaction outcome, the immediate outcome, is negative for the predator. Here there are many examples. For example, in brood parasitism, that in between, for example, the Clamator glandarius and pica pica, the magpie and the great bird cuco here in Spain. In many cases, for the parasite, the positive effect is to be rare by the host. But in many cases, the parasite is killed by the host. And so on. We can extend this view of dual effect to many different types of interaction. Here, for example, for mutualistic interaction, here in blue, we can have that for pollination, in many cases, the immediate outcome is a fecundity enhancement for the plant of the interaction with the pollinator. But in many cases, the pollinator is also killed by the plant. Like, for example, in many examples in which the plant produces toxic nectar. That is very common. Like Echium vulgare is an example of this. In summary, if you read the natural history of many interactions, you can find examples of both immediate positive and negative outcome for the same pairwise interaction. And even in some specific cases, you even can see that the authors have counted the proportion of the encounters in which the interaction is positive or negative for the two partners. Here is an example of typical predation. This example you can read in this classical monograph of Schaller in 1972, the Serengeti lion. This author observed that of 1,000 hunting sets, one of these sets ended in the death of the lion. That means that if I calculate the proportion of positive versus negative immediate outcome, I have that for this pairwise interaction for the lion, the proportion of positive immediate outcome or event is 999, and the proportion of negative effect are just one out of 1,000. In the case of the interaction between leopards and baboons, is even more dramatic for the leopard. According to this study of Collishaw, 1 out of 10 hunting set between a leopard and the baboon end in the death of the leopard. Then the next point in our framework that we can locate any pairwise interaction along the gradient between positive and negative immediate outcome. And this is more or less a view that where or how diffuse is the interaction and how far from the both streams are the interaction. This is something that is not rare. If we again go to the literature, we can observe that many interactions are located in a gradient between just one type of immediate outcome or just the opposite type of immediate outcome. This is a non-exhaustive, again, review on many different types of interaction. These are the type of interaction, and these are the number of examples I have been able to locate, and this is the proportion of immediate outcome that are negative, and this is the proportion that are positive. You can see that how different interactions are moving along this gradient. This is giving us an idea that the mutualism antagonism continuum can occur for many different species, many different pairwise interactions. And that the reason is probably because they are dual effect. Again, you have a different blue code depending on the type of interaction we are classifying this. Well, the last part of our framework is that the continua in any outcome of the interaction is defined or can be defined like the variation in the interaction outcomes that result from the coexistence of negative and positive immediate effect and their per-event strength. We can have here like an ideal way of observing this. we have here this, that is again the gradient between mostly antagonistic and mostly mutualist. These are a cladoceran, and each dot is one specific interaction event between one specific individual of duck, Dublin duck, one species of Dublin duck, and one clone of cladoceran. The size of the dot is the per event strength of the individual event, of the individual interaction. And the color means if the event is positive or negative, blue in positive and red in negative. This is ideal, something that is handmade only to show you this framework. Here you have what is happening to the cladoceran when we exclude the Dublin duck. And within context, we can locate each specific pair of interaction between different pair of individuals along this continuum, depending on the difference between this and this, that is the null situation. Okay? For example, between this gray Dublin duck and the cladoceran, the interaction is negative for the cladoceran. Between these three, blue, red, and green Dublin duck, the interaction is positive with different magnitude to the cladoceran. So we have a way to find a continuum between mutualisms and antagonisms within the same context. We can scale this to different contexts if we average this per habitat or even between different species. That means that the continuum has different levels of organization, can scale up to different levels of organization. And in addition, another property of this continuum, as I'm showing you, is that we can find the continuum for both species of interacting organisms. Like here, for example. If we have this, we can have a change between antagonisms and mutualisms for one species, a change between antagonists and mutualisms for the other species, and the result is that we can move from interactions that are positive for both species, positive only for one species, or even negative for both species, using exactly the same framework. Like with this conceptual framework, we can explain all different types of interaction irrespective of the sign of the interaction and irrespective of the classification a priori that we have of those interactions. Okay, this is our framework. What is new here is that we consider that in most cases between two specific pair of species that are interacting, always there are dual effect, positive and negative interaction outcome. Then, what we have done after this is to modelize this mathematically. I'm going to explain the model just very, very briefly, but you can read the model in this paper that has appeared recently in Ecology. But basically, what we did was to create a system of differential equation in which we have 4 terms. The first term, that is a logistic term, is basically the intraspecific effect. This is the per capita growth rate of the species without the interaction, and this is the logistic effect of the interaction of the growth due to intraspecific competition. And then we add 2 terms, one that is positive effect and the other that is negative effect. The positive effect is basically a term indicating how many interaction events are positive every time 2 species are interacting. That this is done by the value of P1 or P2 here. Okay? And epsilon is the per capita strength of each of the interaction events. Exactly the same for the negative term. This is alpha, that is the per capita or per event interaction strength, and this is the proportion of interaction events that are negative. This, as you can see, this is a consumer resource model that we have basically include a new term, negative here and positive here. And this is the cost of the interaction that we are not modeled yet, but we are starting to model this. And what we found? We found that if we change the value of the 2 strength, the alpha and the epsilon and the value of the p, we can move from very different type of interaction. This is the p space, that is the change in the value of p1 and p2, proportion of interaction that are positive from 0 to 100 or to 1, and from 0 to 1. And these are different values that we have modeled for alpha in the two species and for epsilon in the two species. And you can see here, for example, they are moving the value of p1 and p2 when the interaction are weak, 0, 1 for the 4 parameters, you can have all the situation. When the interaction is strong for the epsilon but weak or small for alpha, most of the situation, irrespective of the value of P1 or P2, is mutualism. That means that you can have a mutualistic interaction even when the proportion of positive interaction events are very low. And what is more interesting is that when you make alphas, the negative strength, very strong, you find situation in which one of the species or both just collapse and the interaction disappear. And you only have a very small region of the P space in which the two species coexist and the interaction is possible to occur. And then with this model, using this framework, with just one single model, we can have all the possible situations between two pairs of species. And then just to conclude, our approach would say that interacting organisms have potentially two immediate effects on their partner, one positive and the other negative. It's the dual effect. This is something that we assume. It's a strong assumption of our approach, that the interaction outcome result from the coexistence of positive and negative immediate effect that vary in per event strength is also a very strong assumption. And using these two assumptions, what we have is that we can move from any different type of interaction, just moving this value. Thank you. Okay, thank you. Sorry.
Sí, sí. Gracias. Gracias, Jordi. Ópale. Esa second. Ópale. ¿Dónde está esto? ¿Dónde está el mouse aquí? ¿No hay mouse? Ah, sí, mira. Sorry. Oh. Espera. A ver. Tienes que dar escape, ¿no? Lo hice antes, pero la tengo en el browser. Tienes que salir de aquí. Eso es. Sácala. Sí, sí. Eso es. Dale ahí. No, pero espera. A ver, cómo se le da a la pantalla total y ya está. Ya lo tenía abierta. Eso es. Sí. Ya está. Gracias. Gracias. Ok. Ya. Just a second. Oh. Sorry, everyone. Avanza, ¿no? Sí, antes avanza, ¿no? Ya. Okay, everyone. So congratulations, Jordi, and the team has made this possible. Really happy to be here. I'm gonna try to summarize the modeling kind of we are still doing after a couple of years. I would like the title to be Trait Architecture Predicting Cascades in Biological Control kind of ecosystems. That would be lovely, but we are not there. But the aim is to have a modeling framework that allows us to really using many traits at the individual level for many species to allow predictions to really test with empirical data. For example, if the evolution of correlated traits predict cascades and how these cascades allows for the local extinction of pest, for example. That would be lovely to have a theory that is able to really make predictions around these problems. This is a teamwork for many years. Many of them theoreticians, Pablo Guimaraes, Stefano Alesina, Leonardo Aguirre, Victor Egilus, different countries. It's an amazing team, learning from them a lot of problems. Also interesting here is, I've been working mostly in food webs, trophic interactions, trophic traits, interaction traits, mutualistic, antagonistic, but then when you interact with people, you broaden your mindset, you start to really see how complex life is and how important it is to build theory driven by empirical evidence, by the people that know really the problems in the field, and this is challenging because then models become more complex. Right? If there are many people working in many topics, like most species live in environments, temperature, precipitation, many abiotic variables, but then many preys, many predators. These are already two big set of traits, and then dispersal traits, metabolic traits, and we can add till a large number. This is quite complex, and we don't have a theory in ecology, in evolution, allowing to systematically modeling complex genotype to phenotype maps or architecture to understand coexistence or this type of more applied problem. So this team is helping me to try to move forward. So I have two main questions today. One is, how does trait architecture affect resource consumer dynamics and coexistence? This is a set of preliminary simulations around different scenarios of trait architecture. Classically, we have like two boundary conditions. One is the evolution of modularity. If you go for systems biology, this is full of modularity. Many patterns from the genomics to morphological traits, so modular patterns. But then in the other boundary, you see also strongly correlated characters. So the challenge is to build a modeling framework that allows to test these two boundaries and the whole gradient in between. And the second question is a synthesis, it's a preliminary synthesis to connect complex traits. We enter not in quantitative, but in complex, meaning explicit pleiotropy, epistasis, and regulatory dynamics at the genomic level, and then you have a complex genotype to phenotype map. Still work in progress. I think Jordi have been doing really an amazing work with Weaver, and this kind of complements this from a more maybe pragmatic point of view, meaning simpler models, these kind of problems we are trying to deal with. This is the literature I have been working with for this talk. This is a public, it's been recording now, but it's also public talk. You can download from the GitHub repository and take a look. So I'm mostly be dealing with these things, with trait evolution. So essentially, you have a population. This is one trait, right? And you have a frequency distribution. So you have, in the center, you have most individuals are in the center, and then you have also individuals with some trait value in the periphery, in the margins of the distribution. And then you essentially, depending on the mechanisms you put in the dynamics, these distributions are going to be amplifying, meaning the variance is expanding, but If you have strong stabilizing selection, then the variance is contracting. And you can have also fluctuating selection, many potential selection modes in the dynamics. The challenge is that we have many of these distributions because each one is a trait. So you have many traits, you have these many distributions evolving per species. We'd like to introduce briefly that maybe two boundary conditions for understanding trait architecture. The first is a classic work, right, in adaptive radiations in microbial communities. So you essentially have the formation of clusters and microbial types within each cluster, and each with a specific set of traits, adapting to specific nutrients and microabiotic conditions. And then you disturb this stable state condition, and then you get the formation recombination, horizontal transfer, and then you end up with a different condition, different situation, but also most species are highly clustered. So this is, I think, one boundary condition because it's essentially as an easy system to understand, even if this is complex, but it's quite limited, small, and the boundary conditions are really, really narrow. You can also have a much broader set of boundary conditions. For example, sticklebacks, they have been colonizing the freshwater ecosystems coming from the ocean, and in the ocean they got many predators and many defenses, and then also size is changing quickly, and they are adapting to new abiotic conditions like water without salt. And so these traits, the abiotic, biotic, and dispersal or migration traits, are all evolving somehow. The point is, are they evolving in a modular fashion so they can move in different directions independent of each other, or are they being strongly correlated so they have some constraints in how they are colonizing new habitats? So these kind of situations can help us to understand better how does trait architecture affect resource consumer dynamics and coexistence. Essentially, quick summary. I think most of you know this plot. Is essentially you have the Grinnellian niche, temperature, precipitation, abiotic traits. Then you have the Eltonian niche, depredation, parasitism, biotic traits. And then you have the Hutchinsonian niche, so you combine the two, and then you have the more recent, right? This is mostly biogeography. But in many fields in ecology, we have been arriving to similar situations. We have broad classes of traits. Now we are in a situation where biotic, abiotic, and movement combine each other. The point is how they combine within and between populations to make ecosystem function, to make them work. So a recent review in tree, essentially, this is the classical way we deal with traits in ecology. So you connect them in 2 or 3 dimensions. Here in 2, trait X, trait Y, you have positive correlations, negative, no correlations. This is classically, right, using the covariance matrix. So you have data, morphological data, or any kind of data about traits, and you can really plot them in different ways. Here you have the 3 matrices, the intra-specific covariance matrix, but you can also have the among species covariance matrix. I think most of us get, right, this understanding from the data. The point is how to model this, how to model mechanistically the dynamics of these matrices to grasp a bit how trait architecture can affect the species coexistence or have predictions of interest. We enter here a bit more in when you go to systems biology. Essentially, you have this continuum. You have the continuum from integrated phenotypes, meaning you have the genotype, different genes, you have development, and then you have the genes essentially regulating the expression of different traits, and you also have interactions within traits. So this is essentially the idea of integrated phenotype full of pleiotropy. So specific genes are affecting more than one trait. And so these networks, they have been used in system biology, in many branches of evolution to understand the genome evolution and the architecture of the genetic architecture of many populations. But when you move towards more fragmented genotype to phenotype maps, you essentially end up in modular phenotypes. Connectivity is lower, and pleiotropy is lower, and you have modules. Modules here could be, you have a module of a set of traits that are biotic traits, are responding to biotic predators, or are the traits these guys are using to use resources. And the same with abiotic traits. So you have this gradient. Now the point is how we advance. So there have been a couple of simulations around this. This is one. You have in panel A integration and constraints. So the more integrated the traits are, the more correlated, the higher the pleiotropy, plus other mechanisms connecting genes and traits, the higher the constraint, meaning that, for example, if the sticklebacks, these guys are moving towards one specific habitat, less salty water, they maybe are releasing the stress in one direction, but they are adding maybe new constraints. For example, the adaptations to salt water or salty water are connected to defenses, because there are many predators where they were living in the open ocean. And so they cannot really move quickly towards the freshwater systems, because all these constraints, they have been experienced for thousands of years. So that's a bit tricky. It's not really easy that would be the ideal. But these constraints, essentially you put this in 2D, PC1, PC2, you essentially see that these constraints produce a narrow line, right, around these two dimensions. And when these constraints are not there, meaning that the two traits evolve independently, you essentially have no correlation. So you can, in that kind of a sticklebacks scenario, you can really move really fast and adapt much quicker. That's something interesting, for example, for biological control, right? If we were able to have superheroes, like organisms that really have many dimensions independently evolving, that could be really interesting to see how they can control many of the pests. But taking into account these correlations is a bit more tricky. Essentially, when you add these constraints, what you see is that in time, phenotypic divergence is the variation is much higher when you have constraints. Essentially, that means that when you are adapted to specific habitat complexities, you are entering with these correlations there. And maybe you are getting a trap, because it took so long in correlating all these traits that you are really doing well. But if the conditions change, it's going to be difficult to live from these kind of constraints. On the other hand, when you don't have correlations, you essentially are moving the traits independently of each other. So these are important kind of concepts to see how we can really model these multidimensional systems. So imagine we have a set of traits, morphology, reproduction, territory, physiology. We can put many, and then we can somehow aggregate. That would be lovely to model everything as a dimension, right? But still, we are doing here a big simplification. So imagine we can categorize three big types of traits, like the abiotic traits, the biotic traits, and the migration trait. And you have two boundary conditions, right? In the top, you have trait integration. So you have essentially correlated traits. You have some matrix structure that is telling you that there is some covariance, and you can explore the gradients from low, medium to high covariance on the top. And on the bottom, you have a strong modularity. So these things are independently evolving. And imagine with this framework, you can connect this with fitness, with individual fitness. Now you put this in a space. Essentially, you have here the three pink sites, and then you have like 4 species, the biotic trait, abiotic, and migration. We are assuming here normal distribution, quantitative traits, so an infinite number of loci with extremely weak effects on each trait. So it's let's say, one of the simplest conditions for now. And now imagine that we can model the dynamics of this. We try to say, well, let's try to model the two boundary conditions, strong correlations or strong modularity. And let's say how far we can go. So we can do this. It's the BAM, essentially, biotic-abiotic migration modeling framework. So you have the covariance matrix. You can have this from the data, indeed. Some data already exists. And now you can calculate the fitness of each phenotype, of each individual species I in space X, inside X, at time t, you can obtain the given a trait architecture introduced by this matrix, the covariance matrix, you can obtain the fitness at time t of each individual. And so with this equation, this is the matrix form of the equation, right? This is essentially, you have for each individual, you have 3 dimensions. And so for each individual, then you have the difference, how this is the interaction, right, with respect to an optima. So you essentially, one example here, you are in a site and you have a specific biotic trait. And let's say you are a predator. And now what you do is you count all the trait values of all the prey and you put that predator in one position. If you are really close to all the prey, you are meaning that you can attack them all, then your fitness value associated with the trait value is high. But you can also be in the corner, in the periphery. You are maladapted to that site. Then the fitness for the biotic trait is going to be lower. We can do the same. I think this is collapsing now, right? Hey, hello? Okay, perdón. Sorry. This is good that we stop here in the most beautiful slide. So, so you have this vector here, which is essentially, right, the difference with respect to the optimum for each of the traits. And then you have the covariance matrix, right? That is going to tell you the covariance between each pair of traits. And then you have the same vector here, that is the distance to the optimum. Essentially, you can model the dynamics for many sites and for many complex spatial topologies and explore the effect of the architecture on the coexistence of species. You can model, we have been modeling like 100 prey, 100 predators for almost 2 million individuals now. But a preliminary plot here is a phase space. We essentially explore the migration interaction gradient. Interaction here is this coefficient, right? Is gamma. So essentially, the stronger this value, the stronger interaction. Essentially, it's a co-evolving, right? Because the traits, they keep co-evolving. And the stronger this interaction coefficient, the faster the co-evolution. So the point now is with this gradient, we see for the 2 scenarios, modular is when the covariance is close to 0 on the covariance matrix, and the magic is when there are strong correlations between traits. So explore many priors, many initial configurations of the covariance matrix along this migration, interaction, gradient, and that is one general result. Essentially, the evolution of modularity allows for higher coexistence in the number of species, regardless of migration and interaction. And in the opposite, the stronger the correlations, the lower the number of coexisting species. The metaphor here could be that, for example, for biological control, we can say that when you have species that are with strongly correlated characters, you are going to have in here predictions that allow for faster trophic cascades. So essentially, you can try to fit the different predictions from the modeling to the empirical observations and see how the covariance matrix is able to predict the empirical data. So a synthesis, just quickly, I'm summarizing. This is a an extended framework we are working with. Essentially, you have genes and phenotypes here. You have pleiotropy, right? As I was telling you, right? How genes affect the traits, and you can have epistasis, so it's essentially the total effect. And then you have also regulatory interactions. And so you can have different scenarios, correlated or modular trait interactions. And then from this, you can obtain the phenotypes and the fitness, depending on the functions that you use to calculate the optima. And then from this step, you can go to the interaction traits, distribution of abundances, distribution of traits, and many other properties. So we have been doing simulations with this more complex framework and for the same problem. And essentially, x-axis, you have magic strong, so meaning strong correlations with strong interactions. And then you have magic with weak interactions, modular, strong, modular, weak. And here the Shannon index, as we can see here, you have a similar pattern to the previous simulations without genomics. Essentially, the diversity goes down, is much more variable, the more integrated the characters, the traits are. So essentially, the evolution of modularity is making the system with higher number of species with equal proportions of abundance. Just a take-home message here, correlated traits impose constraints to coexistence. So integrated trait architecture decreases resource consumer coexistence along migration and interaction strength gradients. And the second maybe summary here is like evolutionary dynamics reproduce the same patterns that traits without accounting for genomics, decreasing coexistence under more complex trait architectures. Thank you.
Okay. May I put it here? See? Okay. No. How do I get out? Okay, wait. Management. I think not. I think, well, I just need to figure out where the presentations are. Aquí. Está. Okay, so I just want to say 3 things before I start. One is I hate microphones, so I am uncomfortable. The second thing is I'm going to talk about preys and not predators. But because my plan in the future is to actually use these preys to then feed to a predator, this symposium is actually super great for me, and thank you, Jordi, for organizing it and also for letting me participate. And the third thing is that I am actually an evolutionary biologist, and this represents the first attempt that I got at doing some ecology. So bear with me, please. So I'm going to talk to you about how order of arrival can change coexistence outcomes between 2 competitor species. And so this is the paper, and this is the people that actually helped this paper to come to reality. So if you want to check it out, I think it's cool. So the question here is, and we know, you know this more than me, of course, but there are a bunch of species that occupy the same niche, right? That compete for the same resources. The question then is, how are they able to coexist? And of course that there's a bunch of theories. Carlos actually talked about some of them already, but more recently, modern coexistence theory tried to grab all these concepts of niche and fitness differences to kind of create 2 big
concepts, which is niche overlap, which basically measures or quantifies how much species compete with each other within its own population and between species. So here the idea is that if we have a large niche overlap, then interspecific competition is actually lower than interspecific competition because they mostly fight with each other for the resources and don't fight so much with their own population. If there is a small niche overlap, then competition will happen mostly within the population and not between the two populations. The second concept, which is fitness differences, so this is, the formatting is a bit off, sorry. But basically, the idea of fitness difference is to just quantify how much a species is better than other at using the resources and then competing for it. And the idea is that if we have large fitness differences, then one species is a better competitor than the other, but if we have small fitness differences, then species are more or less equivalent. And the idea behind the modern coexistence theory is that if we have niche differences smaller than fitness differences, which is in the y-axis, and niche differences in the x-axis, then coexistence will happen. One thing that can actually affect how species can coexist is stochastic factors like order or timing of arrival, but other deterministic factors also. For example, if one species is more adapted to a specific environment. And today I'm going to talk to you about stochastic factors like the order or the timing of arrival. And order or timing of arrival during a community assembly can change the outcome of interspecific interactions. This is something that has been known for a long time. And, and basically it can lead to priority effects. And now there's a lot of debate about priority effects, but in this case I'm going to define them as species dynamics are basically governed by positive frequency dependence. Okay, so basically the first one to arrive wins. And Kay and Letton basically introduced or extended modern coexistence theory to incorporate priority effects. And so if you have here again niche differences and fitness differences, you see that coexistence happens on this side. Again, fitness differences are lower than, niche differences are lower than fitness differences, but on this side we have priority effects. Whoever falls within this, the dynamics are not fixed. They will depend on who actually starts growing more in the beginning. And so here, the species that arrives first dominates. And order of arrival should be very important in herbivore communities because herbivores that arrive early, they occupy space, they reduce nutrient availability, they can start the plant defense induction, or they can actually also suppress plant defense induction. So this could lead to priority effects. And so here, my questions are, how does order of arrival determine herbivore coexistence? And what is the role of priority effects on herbivore coexistence? And for that, I'm going to use two spider mite species, Tetranicus urticae, which you've heard about today already, which is a generalist, and it actually is usually a defense inductor. And then we have Tetranicus evansi, which is a tomato specialist, and he's a defense suppressor, and he's invaded from South America. And the fact is that we know that T. evansi outcompetes urtica in tomato plants. But when we go to the field, we often find them coexisting in the same plants. And this got us wondering, how can this happen? So to test this, what we did was we used tomato plants because we know that evansi is the specialist of the tomato plants. And basically we varied the number of females of urtica and evansi that we introduced in the tomato plants. Either we have different initial frequency, or we varied the time of introduction. So either we introduce them simultaneously, first urticae or first evansi. Then, after 2 generations, we counted the number of adult females and registered where they were in the tomato leaf. And then we estimated the growth rate in the absence of competitors and intra and interspecific competitive ability. And we used this data to estimate niche overlap and fitness differences. So what you have here is the proportion of T. evansi in relation to urtica. So basically, if it's above 0.5, evansi is winning. If it's below 0.5, urtica is winning. Here you have the variation in the number of individuals that I put, so either 1 urtica, 19 evansi, 10 urtica, 10 evansi, 19 urtica, no, sorry, the other way around. 1 evansi, 19 urtica, 10 of each, and 19 evansi, 1 urtica. Okay, and then you have here the results when they were introduced at the same time. And you see that when they are introduced in the same time, we already have the pattern that we were expecting. Evansi in generally overcompetes or better, or is able to get the best of Urtica. It's even more obvious when it arrives, when it's in a higher density, but when Urtica starts at a higher density, it actually gets a bit better at dealing with Evansi. When Evansi starts first, there is no doubt that it actually Urtica gets extinct. And then when Urtica arrives first, we actually see that it is able to you don't see the rest of the text. that Urtica is able to actually resist to Evansi. And so when we do the coexistence plot that I showed you before, so niche differences and fitness differences, you see that when Evansi comes first or they arrive at the same time, Urtica is excluded. But when Urtica arrives first, the two species can actually coexist, which shows that order of arrival is able to change how these species coexist. But our question was then, how can this happen? And so what we looked at was the distribution on the different leaves of the tomato plants. And here what you have is the proportion of T. evansi when it's alone, so it doesn't have any competitors besides itself. And we looked at the leaf position in the tomato plants. And you can see that there is a clear preference for leaf number 4. Okay? Then, when we compare the pattern, here again the leaf position, but here is the difference to the occupation on the single regime. So this basically just measures that evansi is occupying more or less, more than it usually occupies when it's alone, or less than occupies when it's alone. And you see that when Teurtica comes first, it actually occupies leaf 4, which allows it to coexist by taking the preferred resource of the better competitor. So basically, we can see that order of arrival can actually change the coexistence probability of the two herbivores, and it does it so via niche preemption by the weak competitor. And we basically just varied the order of arrival by 48 hours, so they either arrived at the same time or 48 hours before the other. And all of these, and these small differences, were actually sufficient for the monopolization of the resources in these two competitors. And so, we can say that small temporal differences can percolate into small spatial heterogeneities, fostering coexistence and the maintenance of diversity. And that's it.
Fascinating talk. I'm not sure what do you mean by leaf position 4? I don't know tomato
so
leaves.
It's so basically the tomato plants, we count the leaves. So when they start growing, right? So the first leaf to appear is leaf 1, leaf 2, leaf 3, leaf 4. So leaf 5 is the most nutritious one. It's the younger one. It should be the better resource.
So could you easily put Vaseline or some barrier to leaf 4 and then repeat this experiment and see what the outcome might be? If leaf 4 is not there, would the outcome be different?
Yes, I could. I did not think about it, but it would be a cool experiment to test whether this would be actually the pattern that allows for, yeah, that is a good experiment to test it. Yes, we can. We actually do the same, we do the opposite way often, which is we isolate one of the leaves and we use only that leaf to then see, for example, if it induces defenses in the whole plant or things like that. But that's a good idea. Yes?
Yeah, great, really interesting system, and thank you for the talk. So you're talking about priority effects on a single patch of habitat, a single tomato plant. But if you think about coexistence more broadly, it requires repeated colonization and extinctions across entire arrays of, tomato plants, if you will.
Yeah.
And so the opportunity for arriving first depends upon how many other tomato plants already out there have the species in question. And so I would suggest you put this into kind of a metapopulation context and looking at competition because years ago I did some work with other people, and you know, if you have just a priority effect and nothing else, if you put it in a metapopulation context, one species or the other will in the long run win out. It's harder to generate the priority effect at the metapopulation scale because of repeated colonization and extinction.
Okay, so I really like that comment because I'm trying to convince Sara to do an experiment exactly about that, because I'm interested in local versus global coexistence, right? So if it's local, so the idea was really to not do it with plants because it's a bit more difficult, but to do patches, leaf patches, that have are interconnected, and then we just distinguish the patches in different ways and vary also the level of adaptation that they have. So it's a I will try to answer to that for sure.
Vale, Yara. Hola. Vale, genial. Gracias.
Hello. We're going to start after the last session of the day with Oscar Godoy from the University of Cádiz and also from the Biological Station of Doñana in Sevilla. The title of his talk is Non-Random Interactions Shape Species Coexistence in Multitrophic Communities.
Perdón, en castellano. Lo veo aquí, pero no lo veo aquí. Aquí no lo veo. Me da igual, pero... Y no está... Supongo que será este. Ok. Ok. Vale. Bueno, pues así. Okay. Well, thanks for being awake, to be honest, because I'm a bit tired, so... Okay. So thanks, Jordi, for organizing the meeting. I've been working little in spillover effects on predators, and I'm learning a lot this morning and this afternoon. So thanks a lot. I feel a lot of privilege from being here. So my name is Oscar Godoy. I work at the University of Cádiz, although I will be soon moving to Biological Doñana Station as a permanent position. And today I changed a little bit the title from what was written on the menu, just because sometimes multitrophic interactions mean something different from contrasted trophic levels. And when I mean multitrophic interactions, it's about the interactions across contrasted species. In this case, I will be talking about plants, pollinators, and their predators. Okay, so this talk is a bit a personal journey. I've been working in this system in Doñana National Park during the last 10 years, and I feel like a child. To be honest, it's like when I was a kid. This is not me. It's just an image obtained by artificial intelligence to make this talk a little more elegant. But one thing that it really reminded me when I was a kid is if I go there, I see a lot of diversity of animals, right? I have the plants, many different species of plants. I have also caterpillars. I have insects that they may look such as Lagorina, that may look like they are pollinating, but actually they are eating other insects. I have also these very nice spiders. I have pollinators over there. And I have snails on flowers, which also they are doing something that is not what you might think. And the thing is that this diversity is amazing and is all together. The question that the ecologists has been trying to answer for centuries is how this biodiversity is maintained. How is possible that we observe within the same area no more than, I would say, half hectare, more than 200 species? How this is possible, right? So I became an adult, and then when I tried to address this question, people told me, well, it depends. It depends the answers to understand such diversity. It depends on whether we are in a competitive system focused between plant-plant interactions, or it depends if we are working on prey-predator interactions. For instance, the predator is the caterpillar and the prey is the plant, or if we are working in mutualistic system. But I'm a bit starborn, and I said, well, don't tell me that it depends. I want to understand it all. How we can understand how a species maintain for more than 100 species. And I want to understand that in the field. I don't want to use computer simulations for a variety of reasons. And to address this question, I think we really need to solve three limitations: a theoretical limitation, a model limitation, and empirical limitation. The theoretical limitations that we need to solve is which are our predictions about how a species coexist when we have species that they are doing positive interactions among themselves, negative interactions among themselves, or neutral interactions, or positive or negative, or negative and neutral, as Jose Maria was saying. The modeling limitations that we need to solve also is that how we can account for all these different types of interactions and species and make a system tractable. So we can follow what will be their dynamics, and we can have predictions about which species will be able to coexist and which not. So it's not just focusing on 2 species, it's focusing on 100 species, and how can we keep track of everyone in our modeling framework. And actually, the last limitation that we need to solve is the empirical one. We need to go to the field, and we need to measure all these species, we need to measure their abundances, we need to know on which resources they feed, we need to know which are the nesting preferences. So it's a lot of limitations that we need to solve, and I hope with this work that I'm presenting today, I will show some of the approaches that we took to solve these limitations. So as I told you, this place in Doñana National Park called Caracoles Ranch, in English is Snail Ranch, because it's full of snails. We have a gradient from these upper parts in which soils have higher organic matter, higher water availability and nutrients, and we have these more empty patches in which there are more salty soils, there are less nutrient, there are less plants, and there are less organic matter. And across this gradient, we established 3, 6, and 9 plots. And you can see how a plot looked like when it was a big flooding event. So you can see, well, we are working within the park. We need to be subtle in the sense that if we put big flags, big colors, a lot of deer will come, also a lot of different animals, and they will basically pick all up. And this is how we measure interactions, or measure species abundances. For each plot is 8 by 8.5 by 8.5 meters, and each plot is divided in 36 subplots of 1 by 1 meter. And we went during 2019 and 2020, during the growing season, weekly, to measure the abundances of the different species that we find. Think that the spring of 2020, it was COVID time. So it was difficult because we needed to go there, but also it was rewarding. While a lot of the population were within the houses, we had the opportunity to go to the field, which was great. And you can see the high levels of diversity that we found. We found 17 different species of plant species, 46 herbivores different, and 45 different pollinators. And when I say herbivores, I'm not just referring to snails, but also to caterpillars, to grasshoppers, to aphids, to many different species. And when I say pollinators are not only truly pollinators, such as bees, we have also bumblebees, we have different species of beetles, we have also butterflies or syrphids as well. And for these almost more than 100 species, we had recorded more than 200 unique plant interactions, more than 100 unique plant-pollinator interactions, and 160 unique plant-herbivore interactions. Okay, so far it seems that the empirical limitation is solved. We went to the field, we had everything under control, we measured that, all set. And this is how the different plots look like across the gradients. You can see the plants here in green, the pollinators in yellow, and the herbivores as beetles or other things as in blue. And you can see that there is quite a variation. I mean, there is a lot of species, there is a lot of connections, but also there are variation across sites that in some cases could be related to the abiotic conditions that we observe at each point. Okay, so how we solve the modeling part. To solve the modeling part, what we took is a lockable terra approach. It's very a simple approach in which if we take the lockable terra model on its discrete form, is the population of one species in the future, t plus 1, will be equal to an intrinsic growth rate multiplied by the present population, and that will be discounted by interspecific competition and interspecific competition. But I don't know how you mine interspecific competition and interspecific competition here, but interspecific competition is what happened. Usually models, for instance, plant pollinator networks, have been measuring what happened on the effect of the plant on pollinator and the effect of the pollinator of the plant. But they don't know what happened among plants, and they don't know what happened among pollinators. So our first challenge is how we can parameterize a matrix of interactions that have different intra-gill interactions, interactions within plant, including diagonal, that will be the intra-specific interactions within the gill of the plants. Also, the interspecific interactions within the gill of the plants. The same happens for pollinators, the interspecific interactions of the pollinators among themselves, the interspecific interactions of the pollinators, the same I described for the beetles or the herbivores, and also the inter-gill interactions. So for instance, here we will be describing the effects of the pollinators on the plants, the effect of the herbivores on the plants, or here we will be describing the effect of the plants of the pollinators. Okay? So the second challenge that we are trying to solve is using a simple locable terra, but a complex matrix of species interactions, accounting for the within gill intra and interspecific interactions and across gill intra and interspecific interactions. Okay? Because some information can be derived from the field, but others is almost impossible, we also try to address somehow the degree of uncertainty that we have when we assume certain types of species interactions. So for instance, for the case of intra-gill interactions, and this is the case of herbivores, we can describe this matrix of species interactions, for instance, as a mean field approach, assuming that all species within the gill of herbivores interact in the same way. Let's put 0.5, for instance. We can say, okay, based on classic niche theory, those species that compete for the same nesting places or for the same type of feeding when they are larvae, they will compete more than those that they have different nesting places, one in the plants, other in the soil, or they are feeding upon different kinds of plants. So we can say this matrix of species interactions will vary according to the resource competition. Although also we can say it's not only for the plants or the places they compete, but ones occur in early phenology and the other species occur in the late phenology, and therefore this niche segregation based on phenology, it will also reduce the amount of competition that we observe within this gill. Okay? Okay, and then the results. How we reach these results? We reach these results by acknowledging a theoretical framework that allow us to connect all the interactions across these different gills that we observe in the field. And this framework, which is based on structural stability, tells us one measure of how is the overall degree of niche differentiation among all species within an intergill that conform the community. So this feasibility domain tells you how much is the degree of niche differentiation among all the 100 species within a community. Higher values means that there will be higher niche differentiation overall. And lower values will mean that there is lower niche differentiation. And you can see the niche differentiation for plants, for pollinators, for herbivores, and the combination of plants, pollinators, plant herbivores, and the three of them. And you can see the values for the three different parameterizations, the mean field, the resource competition, or the resource and phenology competition, okay? The first thing that you observe is that there is no systematic trend in how niche differences change across a gradient of biotic complexity. So the prediction will be that the more gills I put, the less likely that we have so much amount of niche differences because species are filling up the niches. And therefore, there will be less opportunity for species to coexist because the whole space is more filled up, right? If I have two species, they will tend to have high niche differences, but if I have 50 species, it's less likely to find axis enough of variation in nature to maintain their niche differences, right? So you can see that for any of the three parameterizations that we observe, there is no systematic decrease in niche differences with increasing the complexity of the network. Indeed, because of the parameterization that we obtain, we find higher niche differences over these different trophic compartments when we Species are differentiated in the resource they use and in their timing of their events, compared to just a scenario of resource competition, or compared to a scenario in mean field in which we assume that all interactions within the same guild, the competitive ones, occur in the same way. Okay, that makes sense. But this is interesting. So the question is, how is possible that when we have not only plants, but plant and pollinators, but not only plants and pollinators, also plant pollinators herbivores, how they maintain the opportunities for coexist? How is not that they are filling up their niches available in the community? Well, the question, or some suggestion to answer this question, is like, are the interactions that we are observing in nature random? Is some kind of neutral processes in which some species interact with others, but there is no systematic trend, and this is more like neutral theory, and it's more about immigration and emigration, and extinction and colonization? And the answer is no. The answer is, if we have the mean field parameterization, the results and competition, and the results of phenology, this is the observed distribution of interactions that we had in the field, and this is the distribution based on simulations. And we observe that for the three types of interactions, the distribution, how these interactions are in nature, differ from random. So we are not in a random scenario. This is not neutrality. Actually, the interactions among species in highly complex systems occur in a particular way. Okay? Why occur in this particular way and not in other particular way? We have no idea. But at least we document the pattern that they are not random interactions. Okay? This is important. Then the question is, okay, if they occur in a particular way, which are the network properties that describe this non-random pattern? Is because they are more modular, as Carlos was saying? Is because they are interaction strength distributed evenly across the community? Is because we don't have very strong competitors, but neither we have strong facilitators? So how this is occurring in the network? And what we observe is that the higher overall niche differences in the community, and therefore the higher opportunities for coexist, occur when intra-specific interactions tend to be higher. So regardless of what happens across a species, if a species tend to limit themselves a lot more than they limit others, that overall increases the degree of niche differentiation, which is a classic result so in many models, but applied to a highly diverse and complex system. Okay? And the other thing that we observe is that this niche differentiation decrease as long as the interactions within a guild, so this is the intra-guild interactions, so these ones are the ones on the diagonal, not the ones on the off diagonal. When they are more overlap, so when species within the guild try to interact more with each other of different species, but within this guild, then the overall niche differentiation of the community is reduced. So a modularity within the guild, and therefore less connectance, increase the overall degree of niche differentiation in highly complex systems. Okay. So just to wrap up, because this was a short talk, so coexistence in multitrophic communities. Indeed, it requires to have an integrated perspective, but not just for the observational point of view, but also how we connect the observations with a particular models that they are tractable and they tell us overall patterns that we observe in the community, such as the degree of niche differentiation, which is related to the maintenance of biodiversity. We also see that the importance is not what happened on the whole network. The importance is that the particularities of the different parts of the network are important. It's important what happened for the intraspecific interactions across the whole network, both the intros and the inters, but also what happened within a guild of each part of the network, because they are not random and because they are structured in a particular way. Okay? So if you want to know more about the paper, we published that, I think is early view right now, with the incredible lead of David, who is in the audience. And this is the paper you would like to know more. And I will finally thank to the people at the University of Cádiz and the Doña Nava local stations for their incredible support, particularly during COVID times. Thank you a lot.
Thank you. Very nice talk, Oscar. Have you considered to include the potential, the putative interaction between pollinators and herbivores?
We avoid it because we had very few information about how they interact. Based on our recordings in the field, we knew for instance, the nesting places or the plants that were feeding the pollinators and predators. We also knew the phenology based on when they appear in the field. But we have very little information about the interactions between herbivores and pollinators that we know, and the literature has support for in the interactions. But because it was a black box for us, we decided to not include that.
And related with that, just a small additional question. Do you have many herbivores that are herbivores during the larval stage?
Yeah.
And pollinators?
Yeah.
Have you dealt a different way that?
Yes, so they were included as either pollinators or herbivore, depending on the life stage. So butterflies when they were caterpillars, herbivores when they were adults, they were pollinators.
Okay,
yeah.
Thank you.
Yeah, really interesting talk. Thanks. Really fascinating system. It seems to me you'd have the opportunity to do some really great thought and maybe some actual experiments where you've got your network and you've got your estimates of interaction strengths. Now you remove a species, for instance, and say what happens to the rest? Are there sort of percolating effects? What additional species might disappear or something like that? And then you could imagine even maybe doing it in the field for key species, right?
Yes, actually this is a research line that we are conducting right now, and we could see that not with experiments because it's kind of difficult to make experiments with more than 100 species, but because we have been following the populations during the last 10 years, there is indeed a strong environmental variation, for instance, in temperature. We have had, such as the picture, flooding events in which we have water, 1 meter of water above the plants for 2 months, and we have other years that they were super dry. Almost no rainfall. So this is gonna create a perturbation in the system. And the question with the tools that we have now is whether the system is gonna be reactive to this perturbation and increase the noise and some species will go extinct, or rather is gonna the perturbation is gonna come in the system, the system is gonna absorb it, and the perturbation is going to decrease. And we can do so with very specific tools. So we can say, okay, these are the species interactions that we have, and these are the intrinsic growth rates that we observe. We can do some degree of sensitivity analysis. We can say what happened when we vary the intrinsic growth rates and when we vary the strength of interactions. We can predict which species will go extinct first. And we can compare, these are the species that the next year they are not supposed to be here, and we can see whether the populations are decreasing because of this environmental variation.
Okay, thank you very much, Oscar. We go to an ultimo talk. It's Paula Lillo from the INEA, CSIC in Madrid, who's going to talk about parameterizing soil nematode food webs. Thank you very much.
So, hi everyone. I'm Paula Lillo, and I'm currently doing my PhD on the effects of climate change on soil webs. And I study in free-living soil nematodes community. And well, together with my supervisor, Sara Sanchez Moreno, we are working with Jordi to parameterize the first nematode soil food web. Our target is to explore soil suppression and other soil function and service in nematode-based food webs. For this purpose, we are going to test if whether Weaver simulation reproduce reality. And to carry out this simulation, we are parameterizing the very first nematode food web. We'll also will increase the complexity of these nematode microbial webs for study pest control suppression. And we also will working on optimal combination of parameters and food web structure for test pest control under different climate change scenarios. So starting with top-down control by soil grazers, let's talk about bacterial field nematodes and protozoes. These two groups of organisms act as top-down regulators affect in soil bacteria communities, so they are affecting basal resources. If we look at nematodes, they are as compartments in all the soil webs. And well, apart the bacterial field nematodes, there are fungal field nematodes, plant parasitic nematodes, and also predatory and omnivorous nematodes that prey to the other nematodes and to another groups of organisms. And that's why nematodes play a really important roles in soil functioning, since as nutrient mobilization and pest control. So, well, we have chosen the bacterial feeding nematodes for the very first parameterizing soil food web in Weaver, and in particular Acrobeloides species. And why the bacterial feeding nematodes? Well, nematode community are mostly composed by taxa that prey in bacteria. This group of nematodes, they are easily grow under laboratory conditions. That's why we have a lot of experimental data that is available. And they are the main grazer on soil bacteria. And that's why they stimulate mineralization of nutrients, and they can control the population of the soil microbial organisms. And that's why they are a key component of soil biodiversity involved in soil fertility and plant productivity. And furthermore, well, this is the first time that we are including bacteria as a basal resource in Weaver. Well, about the nematode life cycles, they have 4 juvenile states separated by molts until they become adults. They can reproduce asexually through parthenogenesis. They have a facultative sexuality, and well, they can, their sex can be determined by environmental conditions. So let's talk about the parameters and traits in Weaver. Well, due to its size, estimates of its biomass need to be made. That's why Andersey developed this width-to-length relationship that with the diameter and length of the nematode, you have the mass. And with this data, we can calculate this allometric relationship, mass-length allometric relationship, and we also taking into account this length-width relationship. Another characteristic is that they have eutely, that is defined that they have a constant number of somatic cells, generally fixed in the adult states, and they have indeterminate growth. With this data, another parameter in Weaver is the growth rate as increase in mass per unit of time. And we are calculating this parameter as the variation around the value of key value parameter of the von Bertalanffy function. For this purpose, we have fitted this 4-parameter logistic model with the growth curve that are published by Jager et al. And we calculate the first value, that is the size at birth of the individual. The second value, that is the adult size. The third one parameter of the model is the inflection point. And the fourth one is the inverse of the key on von Bertalanffy equation. Another parameter on Weaver is the energy tag, understood as the fraction of the body mass that is invested in maintenance, growth, or reproduction. And that can relate that mass with fat body content at those energy storage. Nematodes store their fat in these fat lipid droplets in the intestine. And well, another Weaver parameter is the assimilation efficiency, understood at the variation or at the proportion of the ingested food that is converted into own body mass. And in the literature, we have found that this value, that it seems really low efficiency, that this can suggest that the ingestion rate need to be high. And well, that can be explained because nematodes morphology in the group of bacterial feeding nematodes, they have these specialized lips in their mouth that help to attract bacteria in the soil. And the last parameter in Weaver that we are working on is the metabolic rate. That is the assumption that length is related to mass and thus to energy. And well, we have found in literature that this data of metabolic rate for 8 different species. And well, with this data, we can fit the model, the metabolic model. And we also see that there is a variation in the weights of the different species of nematodes. And one of the next steps in this parameterization is working in these 3 threads: voracity, speed, and search area, that represent variability and performance with different temperatures. And well, our next steps will focus on soil suppression of microbial pathogens and pests. We also will study the effect of these microbial nematodes food webs on soil fertility and the composition rate and nutrient mobilization. And we will look into the possible spillover effect based on the fact that nematodes themselves can transport bacteria in their cuticle, so they can distribute and inoculate soil microbes into new soil patches. So thank you very much for your attention.
Very, very cool system. Very nice detail. Is your group developing a kind of a detailed population model, sort of stage structure population model for the nematodes? it seems like you would have all the material to do that, right? Growth, fecundity, survival, and so forth. I think that'd be a valuable tool.
Yeah. Well, we are working with individual-based models, but yeah, that is really interesting thing to.
Yeah, that the kind of data that she's collecting serves to that too. Yeah.
Thank you.
Thank you for your talk. My question is very similar to Bob's question, but it's related with the metabolic rate, because it's just a curiosity. I have seen that the information you are using is coming from 1905 or something like that, right? Very old paper. Are you trying to do something like that, or is it very difficult to work on that now and getting new data on metabolic rate of nematode? Is just a curiosity.
It can be. Yeah. It's a really interesting question. But I think, I don't know, that we can start working with this data and maybe do this.
New data. No?
Yeah, new data. Yeah, for sure. Yeah, but in our laboratory, we don't have the instrumental to do this. But yeah, it can be.
Okay, thank you.
Okay.
Holt, thank you very much for coming. The ecological effects of spatial subsidies and temporal pulses, a theoretical perspective.
I may need help getting this up on the screen. I'm not sure. Oh, there it is. Okay, well, Jordi, thank you very much for this invitation and for organizing this really interesting symposium today. I've learned a lot. It's been really fascinating to listen to both contributions to basic ecology at very detailed levels for the nematodes that we just heard about, up to more complex systems, to the applied realm, which is really, really fascinating. And you know, I was trying to think about what I could do that would be maybe slightly useful here today. And before I start, I'd like to start, rather than ending my talk, thanking the people I've worked with, some of whom have already been mentioned and some who I'll mention. So Gary Polis, I worked with for many years before he died in 2001. Mark McPeak at Dartmouth, Richard Gama-Kaiwas, who died earlier this year. I'll mention him, and Andy Gonzalez, who's at McGill. My long-term research associate, Mike Barfield, we've written about 60 papers together. Former student Mike McCoy, Michelle Leroux, who many of you know, Mike Hochberg. I've worked with all these people on things related to the issue, one way or another, of spillover in ecological systems. I'm primarily a theoretician. I'll be talking about models, not necessarily complicated models, but models that provide useful conceptual insights. But I did, when I was a beginning assistant professor, set up a large-scale landscape experiment at the University of Kansas, looking at how patch size and distance from source areas, in this case a forest, influenced the rate of succession onto patches that varied in size. I started this we set it up in '83, got the first data in '84. It's still going, so it's now one of the longer running experiments in community ecology. And in a system like this, I was motivated by kind of spatial ecology, island biogeography, metapopulation processes, and so forth. All taxa at all trophic levels, not just predators, can show spillover effects. That's what colonization is, a spillover from an occupied place to an unoccupied place, if you think about it, right? This is what it looked like 20 years later. You can see you got little woodlands developing there. And spillover, there are a lot of patterns that we see, and I'm not going to talk about this in detail. It's just I like to keep natural, examples like this in the back of my mind when I think about theoretical things. So, for instance, we had a large rodent called a cotton rat, Sigmodon hispidus, which was on these big patches, but there would be individuals that would show up on the smaller patches, and they were clearly spillover individuals because they didn't stay very long. So they were sink populations maintained by source populations. And then there are interactions between the patches and the surrounding matrix. And so we've been talking about spillover of predators. There's also spillover of resources, something I wanted to mention since David was here in the front row. Gary Polis visited me in the mid-1990s or something like that. And unfortunately, we never pursued this, but we went out and with some of his students, we looked at the orb-weaving spiders, because you can get out early in the morning and there's dew on the webs, so they're easy to census. And orb-weaving spiders need a structure to build their webs. So you don't find them in the matrix, which is, by the way, mowed to a low turf. But they were really common, more common on the edges of big patches and on the little patches than in the interior. Why? Because there was a lot of grasshoppers and other prey that were found in this productive mown habitat that could flow into the patches, supporting these relatively sedentary predators, right? So that's the spillover effect of resources. And this project is still ongoing. I've got a former student who's been getting great recent data out there. And that's sort of in the back of my mind because there are source sink dynamics, metapopulation dynamics, trophic interactions, and so forth. So when you strip down the notion of spillover to its most basic level, you've got a local community or ecosystem that you're studying. And a lot of what we've heard about today has to do, you have to understand what's going on at the local level. What are the interactions? Competitive interactions, mutualisms, predation. Is it a constant environment or variable environment? But then it's connected by some sort of input from somewhere else. And you can think of a conceptual schemata here where you've got these spatial flows, but both of these are constant environments. Or the environment you're studying is variable, but you have a constant input from some external source, okay? Or the environment you're looking at is actually constant. It's the source that's variable through time, leading to temporal variation in the magnitude of spillover. Or both could be happening. You could have variability in spillover effects and local variability. And interesting things, everything that you understand about one of these feeds into the next. And so what I refer to as here, and what I'm going to be talking about could be, let's start at the simplest level. We've heard a lot about very complex systems with hundreds of species involved, okay? But if you don't understand what's happening with spillover effects with a single species or pairs of interacting species, you probably won't really understand at a deep level what's happening in much more complicated systems as well. So I'm going to start with single species that can persist without input and then say what's the effect of input on its dynamics. Then source sink systems where the species requires input to persist. And then you could have pairs of interacting species, and then you can build up to more complex, what I call modules, up to entire food webs, like what Oscar was telling us about. And these could be simple differential equation style models or very complicated IBMs. So I'm going to be talking about some relatively simple scenarios that have interesting and maybe even surprising results. And some of this is, I've been nibbling at this issue for many, many years. And I'm going to be going back to some really old papers of mine. And I think that understanding this low-dimensional spillover, what's happening with one or two species, is really essential for understanding more complex systems. And the other thing that has come up maybe a little bit today, we're talking about spillover into your focal community from some other habitat, but you can also have spillback. So what's happening in the system that you're looking at can affect the external environment in some way. And when that happens, that leads to what I call spatial feedbacks, because then the magnitude of spillover may itself depend upon what's happening in this local community. And then you can build up to not two patches, but lots of patches in a complex landscape, heterogeneous landscape. And I'm going to argue that when you take into account these spillover and spillback effects, you can get really interesting, emergent, unexpected, and I think at times really important phenomena. I'm going to give you one example of that. So we're going to start with kind of basic population ecology with a twist. So the basic population ecology, we could be talking about like logistic growth or exponential growth or whatever, continuous time, and then you add an input to it. So here we have our constant, focal area plus a constant source with constant rate of spillover. But I'm going to tell you about what happens in discrete time, because this is relatively simple. This is actually really pretty interesting. So this would be, you got numbers at one time, an organism with annual generations, for instance, and then you're going to project it to the numbers the next time. And the G here includes competition for resources, cannibalism, whatever you think is important biologically. But you're going to add an input from an external source. And so the functions that we might look at this is a discrete version of the familiar logistic equation. This is what's called the Ricker model, e raised to the power, that should have a minus, 1 minus n over k. Okay, that's used in fisheries all the time and in insect ecology. And then we're going to add this input to it. So these models were actually used in a very famous way by Robert May, Lord May, who died in April 2020. He had a paper in Science, 1974, which is a very celebrated paper. It's got over 2,000 citations. The paper is only like 3 pages long. And what he did was he pointed out these very simple models, which actually often do okay. People use them for practical management purposes and so forth, have surprising consequences. Namely, that depending upon the growth rate of the population when it's rare, it can either show stable population dynamics like here, or cycles with a little higher growth rate, or more complex dynamics that came to be called chaos, because small changes in initial conditions would lead to totally different trajectories. So kind of a fundamental lack of predictability about the world, our ability to forecast into the future. And in his 74 paper, he used his model, I forget, one of these was the discrete logistic that I just showed you, the other was the Ricker. And he said, okay, these models are somewhat different, but they have sort of similar consequences. Whereas you increase R, if you're between little r of 0 and 2, you got a stable equilibrium. As you increase R a little bit, you go to cycles, then finally you get to this so-called chaotic behavior, okay? And chaos there was a huge flurry of activity in the 70s and early 80s about chaos, and people said, well, maybe it's not important. But there's been a resurgence. There was a review paper just a couple of months ago saying, hey, there's actually a lot more evidence in time series for this behavior than people believe. So it's still out there. So now we're going to take this, I'll go back a second. We're going to take this model right here. We start with some general function here, which we can plot as a graph, and we're just going to add a constant input to it. So you start with n individuals. They're going to produce a bunch of offspring, and they're going to add a bunch of immigrants, and that's when you census. And so when you do that, this is n of t plus 1 versus n of t. This is just some random possible g function, which in this model is, the line here is a slope of 1. If n of t plus 1 equals n of t, you're at an equilibrium. This is a locally stable equilibrium because you go above it and you tend to decline to it, and vice versa. This is an unstable equilibrium. This is an alternative higher abundance equilibrium, okay? And models for insect outbreaks often have these kind of alternative equilibria, where the low equilibrium is determined by generalist bird predators, for instance, that have saturating functional responses. But the insects get very common, say gypsy moths in the eastern US, or a lot of insects in boreal forest. They can escape that predation because the birds get saturated and they blow up. And so this is part of some very famous models of spruce budworm outbreaks and so forth. Something looks just like this. Now, what is the effect of adding spillover to the, constant spillover to this simple model? Well, what you do, remember, this is the G, numbers of t plus 1 is a function of numbers of t, and we just add I to it. So you just lift that growth curve by a constant amount. It's very simple. Well, what you do is you tend to make these low equilibria disappear, and you push up the high equilibrium. So the effect of spillover in this single population model is that if you have alternative equilibria, the low abundance equilibria tend to vanish because your spillover is knocking you above that. And the upper equilibrium is pushed upwards. So that says that and the same thing occurs in continuous time models, constant immigration from an external source increases equilibrial density. And this is a very general statement about population dynamics. Okay, and we're going to see, does this hold when you look at multiple species interacting, for instance, in a little bit. So then you can say, okay, so that's looking at the position of the equilibrium here, right? Where n of t plus 1 equals n of t, but there's no guarantee that that equilibrium will be stable. How do you know if you perturb for it, you go back to it? If the dynamics, so we could ask two questions. How does immigration affect stability? If the dynamics are unstable, how does immigration influence, say, the average abundance over time in the fluctuating population? And then you can make the immigration itself temporally variable, and how does that happen? Well, it turns out that not long after I got my doctorate and moved to the University of Kansas, I published 2 really obscure papers in book chapters, which I've learned is not where you publish things if you want people to read them. And they weren't just, they were really obscure books too. This is in honor of my advisor one of my advisors at Harvard, and his students got together and published a book. And then this is a lecture notes in biomathematics, which biologists never read, basically. But I kind of worked out this problem. And a guy named Louis Stone in Israel came back to the same problem 20 years later, and he published it in Nature with exactly the same figures I'm about to show you. So that'll teach me. So remember, we had these 2 models, the discrete logistic and the Ricker model, that had very similar stability properties in the closed system. How does immigration affect the stability of the system? Well, it turns out they're really different. So this is the discrete logistic, and here, if you can start out in a stable point, maybe with oscillations, and as you increase, this is immigration on the y-axis, as you increase immigration, you push it to a zone where you get cycles or maybe chaotic dynamics. So immigration can sometimes destabilize local population dynamics. Here's the Ricker model, where it doesn't take very much immigration to make chaos go away. That was the point of Louis Stone's Nature article. And if you have a lot of immigration, you get stable dynamics. So given unstable dynamics, increased immigration can both increase and decrease abundance. Let me see if the next slide helps me explain this. And the reason is, this is n of t plus 1 versus n of t for the Ricker model. And what I do is I'm just adding immigration, a moderate amount of immigration, higher amounts of immigration. And stability is determined by how severe density dependence is near the equilibrium. The steeper and more negative the slope is, the more unstable the population is if perturbed from equilibrium. But with the Ricker model, you have what looks like an exponential tail here. And as you raise that model, this gets shallower, i.e. density dependence gets weaker, so you tend to be somewhat stabilizing. So given that you have immigration and there's some unstable dynamics, is there anything interesting that happens? And the answer is yes. This shows what happens to time. So now you've got a time series, and you're just going to take an average over that time series and say, how is that average abundance over time influenced by the rate of immigration from the external source? And so this is the rate of immigration from the external source. And if you have a stable population, as I said, for something settles to equilibrium, increases immigration just increase your average abundance, okay? But if it's unstable, you can find other effects where a little bit of immigration really pumps up your abundance, and further increases in immigration actually decreases abundance. So you get this non-monotonic relationship between the rate of spillover from the external source and the average abundance in your habitat. Why is that? Well, that's because why do you get when you get this unstable dynamics, like this is with chaotic dynamics here in this example, you overshoot and then you crash. And then you grow up a little bit, maybe you overshoot a little bit, don't crash so much, overshoot a lot, crash. So now you add immigration to it, a little bit of immigration, and you have a floor below which you don't go. You don't go to really low numbers. Because you have that floor, it doesn't take you as many generations to get where you're going to overshoot your carrier capacity to get and crash. And so this shows kind of the chaotic dynamics. Add a little bit of immigration, and you overshoot every other generation basically. And when you take the time average of this, it's considerably greater than this. Okay, but then so that explains why you get these peaks when you can really overshoot with a high R. You can overshoot your carrier capacity and crash. what accounts for this decline here is that you're adding enough immigrants each generation that they experience strong density dependence because you're pushing the population to be nearing carrying capacity. So you make the magnitude of the overshoots weaker. So you get these very interesting effects even in a single species model with spillover from external sources, okay? Now what happens if you, and this is, to my knowledge, unexplored in the literature. So with my students at Canada, we did for this talk and this meeting, I said, let's do some explorations of what happens when we make immigration itself temporally variable. And it's interesting. So we're going to look at what happens if you have pulses in immigration every other generation. You have 1 year with no spillover, and next year you have twice I, and you're going to vary I here. And what you can easily have happen is that as you have pulsed immigration, you go from a stable system to one that shows very strong oscillations. Okay, these are just two different examples of that. So temporal variability in the magnitude of the spillover can drive strong instability in a local population, okay? One that's intrinsically stable. And this shows what happens when this is the Ricker model. Honestly, at the moment, I'm trying to remember what the difference is between these two topics. Oh, this is constant immigration, where with the Ricker model, remember, a little bit of immigration went from chaos to 2-point cycle, up, high, low, high, low, high, low. And as you increase immigration, it finally stabilizes. When it stabilizes, further increases immigration just increases population size. This is what happens if immigration is periodic, pulsed every other year. Again, if you have no immigration, you just got the same phenomena. But then you get a two-point cycle, high, low, high, low, high, low, and then you get to a zone where it's chaotic again. And then you get 3-point cycle, high, low, low, high, low, low. And then finally it stabilizes, then you go on. So you get complex patterns when you overlay intrinsically unstable dynamics with temporally pulsed subsidies from the outside, spillover effects. Okay, so we've heard, and I was very pleased that this came up because I did this independently. Several of the speakers have talked about the importance of evolutionary dynamics, genetic variation, and so forth. So what happens if there's a local adaptation in the population you're seeing? So here, where you are now, the population there is locally adapted. And what you mean by local adaptation is the individuals are more fit there than the immigrants are that come in from the outside for heritable genetic reasons. And so there's a lot of increasing evidence for even micro-scale local adaptation, okay? Like there's some great work done out in Oregon with stick insects, where the stick insects are found in a kind of a complex landscape of several different bushes, and the bushes have different colors and shapes of their leaves, and the stick insects have color patterns that match it. And on each little patch of bushes, you'll find a local population that's got maybe a stripe down its back. Whereas right next door, there's another little patch of bushes that don't have the stripe on the leaves, and so the stick insects there are just solid green, okay? And then you move individuals between them. The individuals that have the quote-unquote wrong color are conspicuous to bird predators and get picked off. So they're maladapted. So if you have local adaptation, what that means is that immigrants on average are less fit than residents. So then we can go back to all the things I just talked to you about and say, how does genetic variation, i.e. local adaptation, how does that affect how does that alter how immigration influences population stability and abundance? Well, if immigrants tend to have lower growth rates than residents, it's high growth rates in these models that lead to that instability. You're lowering the average growth rate of the population, so you expect it to be kind of stabilizing in general. But beyond that, there's some other interesting effects. And this is work I did. I mentioned my friend Richard Gommakaiwas, who sadly died. He and I and Mike Barfield, my research associate, over 20 years ago, worked this out for so this is a diploid major gene locus where there's density-dependent fitness of each, type 1, each homozygote, 1, 1, 2, 2, and the heterozygote. But we assume that the type 2 is in homozygous state comprises the immigrant pool,
And then you can say, given that you have a polymorph, we know what's going to happen if type 2 is the only thing present and the system's stable. Increases in immigration will increase abundance. But what happens if there's a polymorphism for the locally adapted type and the immigrant type that's always coming in and being culled out to some degree by selection? So this shows some examples. So this is the superior type in homozygous form has the same fitness as it does in heterozygous form, okay? So there's no heterozygous superiority or anything like that. And if that's the case, you get this kind of interesting effect that as you vary the immigration rate from the outside, and you look at total population size, instead of going up, it's flat. Where immigration shows up is in changing the genetic frequency of the better adapted type until finally it disappears, and after that, then numbers start rising with increasing immigration. So if you're looking at a system where spillover involves individuals who are maladapted relative to residents, changes in immigration rate might not show up as changes in population size at all. And now here, what happens if the heterozygote has actually greater fitness than the homozygote? Well, in that case, depending upon the details, as you increase immigration rate, you can actually depress total population size because the homozygotes for the superior type are mating with immigrants who have the other type, and they have the heterozygote offspring they produce have lower fitness, and the net effect is you reduce the population size at which the population is equilibrium. So seems perverse at first, an increase in immigration can depress population size because this is increasing the frequency of the maladapted type in the local population. Okay, so you can get a variety of consequences for how immigration affects population size depending upon what's going on relative to genetic variation and local adaptation. Okay, I don't know anybody who's empirically demonstrated this phenomenon, but I'm sure it's important out there. So the next part of my talk will have to do with another single species scenario. So what we were just talking about was on where the species can persist just fine in local habitat if you were to cut off immigration. Maybe it fluctuates, maybe it settles to an equilibrium, but it's going to persist just fine. But what if it doesn't? So little r there is negative. So it goes extinct in the absence of immigration. Well, that's what's called a source sink system, where the sink is a population that's maintained necessarily by immigration that balances the negative growth rate experiences. So here's a real world example that I'm very fond of that Ketty did 40 years ago. This is the sea rocket, Caecile aedula, which is found in the maritime provinces of Canada, like Nova Scotia, in sand dunes. And if you go to the sand dunes, what you find is there aren't very many individuals near the edge of the dune, on the seaward side. If you go on the landward side where there's a lot of vegetation, there aren't many either. You find a lot of sea rockets often in the middle of the dune. Well, that suggests at first glance that the middle of the dune is the optimal place to be a sea rocket, right? Well, what made Ketty's work really almost unique still is he went out and actually measured demographic rates, birth rates and death rates as a function of density near the sea, in the middle of the dune, on the landward side of the dune. And when you go to the middle of the dune, this shows death rates and birth rates, and you can see that death rate exceeds birth rate in a density-independent manner over all the whole range of densities that he looked at. So little r is negative. This is a population that ought to be going extinct, looking at local demography. Why does it persist? And not only just persist, but at high numbers. Well, he pointed out very sensibly that you have an asymmetry in the direction of movement of seeds in the world, namely strong winds that come off the ocean that strip seeds away from the productive plants there, depressing numbers, and depositing them here where they can live for a while, because they're not dying, they're dying, but not at a horrible rate, unlike the landward side. So you get a large sink population maintained by this recurrent immigration. So then we can start asking questions like, okay, what is the effect? What if you have good years where actually the birth rate is greater than the death rate there? Maybe rainy years where it's cloudy in the summer, maybe it's okay. It turns into a temporary source. What's the effect of temporal variation in growth conditions in the sink? And so this is something I worked out a long time ago, and I'm going to get back to the spillback effect. Remember, we're talking about spillover and spillback, and one very practical consequence of people not understanding the interplay between spillover and spillback. So with Andy Gonzalez, we had a paper in PNAS over 20 years ago, and we followed it up with various papers. And so here was the scenario. Going back now to the continuous time model that I sort of skipped over. We've got some growth rate that may be varying with time, plus we're going to have a constant spillover from external source. And so if this is constant and negative, this population settles to an equilibrium. This the rate of input from the outside divided by the absolute value of the rate of decline. So as in Ketty's example, you can have a big sink population maintained if you have a lot of input from the outside and you have a slow rate of decline, okay? And now, and this is what I did, 20 plus years ago, let's let the growth rate be variable through time. And you could think of this is like maybe a probability distribution of growth rates. This is, remember, continuous time intrinsic growth rates. Here's 0. To the right, the population ought to persist, deterministic. To the left, it ought to go extinct in the absence of immigration. Here you have variability, but the population is always a sink. Here, it's not a sink at all. But here you have a site where, on average, the growth rate is negative, but there are times when it's positive. And you can then say, what are the consequences? So it's kind of a weak sink. So there are times, if a population is there, it'll grow for a little bit. Well, so I worked that out. And moreover, you can say, well, is there a structure in the temporal variability? So by structure, I mean, is there a correlation from one time step to the next? If there's no correlation, that's what a physicist would call white noise. If there's short-term positive autocorrelations, that leads to what's called red noise, which is very common in the natural world. I mean, if it's cold today, it's probably cold tomorrow. If it's warm today, it's probably warm tomorrow. It's not just a random draw from the temperatures that are available over the year. So autocorrelation is related to short-term predictability. So what I did many years ago, and there's kind of a funny story here, I'd worked out the math of this, where you can show that that if in a time-varying system with variable growth rates, that the harmonic mean of the time series is equal to I divided by the average rate of decline. Just remember what you get, the absolute value of the rate of decline. That's what you get in a constant environment. But the harmonic mean is always less than the arithmetic mean. So this is a very general, like, three-step argument saying that temporal variability increases time averaged abundance in the sink population compared to what you find in a constant environment. I call this an inflationary effect. Now, the magnitude of the effect depends upon the structure of the temporal variation. This is where the autocorrelation issue comes in. And so if you have, here we have, in the abundance environment, I've set this up so that unity is I divided by the average rate of decline. Then you can say, what happens if you have temporal variability, but now there's a positive correlation, so that occasionally you get a good year, but it's followed by a good year, maybe by another good year before it goes bad. So you get runs of good years and runs of bad years. Well, in runs of good years, you have exponential growth. And the population is maintained by immigration during runs of bad years. Okay, so there's kind of a floor to abundance. And this is a log scale here. And this is just, math, solving an equation mathematically on the computer. You can get these spikes of outbreaks during runs of good times. And the effect, looking at the average density, it's a log scale here, can be quite large. This is a measure of variability. This is a measure of autocorrelation. If we went in a constant environment, you'd have unity population. You can have hundreds to thousands of individuals in a variable environment. So I was at Imperial College at Sylwood Park. I was a visitor there. And I would go over there every year, and they would kindly put me up. But they didn't often have an office of my own. So they stuck me with a bunch of scruffy graduate students, including Andy, who was even scruffier than in this picture. And I told him about these ideas I had about temporal variability in sinks and so forth. He says, I can do an experiment to test this idea. Because he was working with Joel Coyne looking at the effect of the noise of the environment, red versus white versus blue, on population dynamics generally. And he was working with this organism, a paramecium, a bacterium, and he knew this is kind of the Hutchinsonian niche, the thermal response curve, little r of paramecium as a function of temperature. So you get above 40 degrees or below about 5 degrees, there's a negative growth rate, okay? And so he could control this by varying the water bath in which you have the culture. And he says, we'll have a source, which will have a constant, it'll be an optimal environment, and we'll just have a constant input, and then we'll vary. So this is a theoretician's rendition of what his much more complex system looks like. These are 2 jars, and you're moving from a source to a sink. And so what we did, and this is probably Andy's idea, you know, we just used a random number generator and went hot, cold, hot, hot, cold, 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 hot, cold, hot, hot, cold, and so forth, right? And then you took that same random number generator, the same string of numbers, and then you resort them so that you have a positive autocorrelation between them. So the mean and variance of the growth rates ought to be the same, but the positive autocorrelation would be larger. So this shows a white noise treatment, where you can see you got numbers that are mainly pretty low. It's not a good environment for them at low temperatures, okay? This is a log scale, this is an arithmetic scale. So then he just rearranges those hot and cold numbers, and here you get these runs of good years, and it gets up into the thousands. And clearly you do you take the time average of the abundance here. You've increased the abundance in the sink population by a couple of orders of magnitude. So the effect was very real. So we got the paper in PNAS because we had the theory, then we had an experimental assessment of it. So this is where I'm going to tell you about a practical application of this that I'll just give you a little teaser about. So this was for a single patch, okay? And then what if the source is so our conclusion, by the way, was that if you have temporal variation in a sink, maintained, remember, by spillover from a source, you can get these outbreak phenomena, and that increases the time average abundance in the sink. And there was an experimental demonstration. And what if the sink is really, you know, if the source is really a bunch of patches just like the one you're looking at that just aren't correlated closely with what you're seeing in your local patch. And so what you're seeing is a metapopulation, each of which on average has a negative growth rate. The question we then asked was, can this inflationary effect permit persistence of metapopulation comprised entirely of sinks? Well, if you think about it, in the long run, you know, the only way this works is if you send something to other patches while you're growing, that seeds them so that when those patches get good, they can start growing, and then it feeds you back. So that's the spatial feedback that I was saying with this spillover, then spill back. And so I worked this out in American Naturalist, you know, some years ago, and I'm going to go through the math relatively quickly. And the answer is yes. You can have a metapopulation comprised entirely of sinks where if there's enough variability, particularly if there's an autocorrelation, the whole population can persist because of this constant spillover and spillback effect. It's kind of an interesting emergent effect of temporal variability. And the magnitude of the effect can be really quite large. This shows the probability you move the patch. If nobody moves, everybody goes extinct. If everybody moves, you just experience the average. This is where the finite growth rate is 0.6, so the average growth rate would project a rapid decline towards extinction, but at intermediate rates of movement, you retain enough capital locally, you can exploit those runs of good years, but still seed the other patches so that when they go good, they can themselves run and feed back to you. And then Andy did a very nice experimental test again, where he had 2 sinks connected by movement, and you make the patches synchronous, and they go extinct, but you make them asynchronous, particularly if there's red noise, and the ensemble persists, okay? So metapopulation of sinks coupled by dispersal can persist if local growth rates are variable, even if the time average growth rate in each location seems to predict local extinction. It requires asynchrony among patches fostered by autocorrelation. And 2 points here. So this, getting back to the theme of this symposium, this all involves spillover and spillback, okay? The recurrent interplay of those things lead to this phenomena. And I had a paper in PNAS in 2020, and one of our former students who's at Virginia Tech had a paper in Science pointing out that this act, and I can explain this to you later, but I'll do it kind of quickly now. This, an absence of understanding of this phenomena almost surely contributed to the failure of the EU and the US to control the pandemic. And the reason goes as follows. So what we showed in the PNS paper, kind of in a theoretical model, and what Nick Ruckton and Choi showed using actual data from the EU, was what was happening. You had lockdowns, people wearing masks, and so forth. That means the growth rate of the pathogen goes negative. So you go negative for a while, things are declining, things are getting better, then people relax, and it opens up again. And so let's say you have 2 locations, in each of which you're doing that. So you have periods of positive growth, periods of negative growth. And on average, the population is declining towards extinction. And now we have 2 locations, and if they're doing the same, everything synchronously, and individuals who are asymptomatic are moving between them, it doesn't matter. You're still going to go to extinction. But if you're asynchronous, if you're not coordinated in your policies, one's going up while the other's going down, the whole ensemble can grow, even though the conditions at each location would have predicted extinction, okay? And there's very good reason. And so, you know, in the UK, Boris Johnson said they had a whack-a-mole strategy. They're going to go in and they're going to shut things down in this town. But people were still moving in between that town and other places in England. Cuomo in New York said, we have a block-by-block strategy in New York, right? We're going to control this block. Well, that's when you have movement between you. You can't think at just the local scale. You have to think more broadly. And I think that's true for understanding the consequences of spillover and spillback in general. So now, that was all for kind of single species stuff. And as I said, I think that this issue of spillover and spillback was really slapping us all in the face during the pandemic, okay? Now I want to return to a little bit of community ecology to go back to the most familiar model. Ignore the text at the bottom. I forgot to take that out. That was for my student. The Rosenzweig-MacArthur model, where you have a prey population with logistic growth, a predator population with a type 2 saturating functional response. So the more prey there are, the more they're eaten, but it kind of saturates out for a fixed number of predators. And then there's going to be a subsidy, which is the spillover predators from the external environment. And so the question is, how does that subsidy affect the dynamics of this familiar model? And to remind you, you can get a lot of information by looking at the shapes of what are called the isoclines of the Rosenzweig-MacArthur model, okay? So this is prey, this is predator, this is the number of prey at which the predator has a 0 growth rate in a closed environment. This hump here, this is the carrying capacity of the prey, and the reason there's kind of a little bit of a positive slope here is this saturation effect, okay, in the predator's functional response. And so as I've drawn it here in a closed system, the system settles to an equilibrium. So what happens now if you start adding a spillover for the predator, so its predator numbers you would think would go up, right? Well, you can see that by looking, when you add a little bit of immigration, even if there are no prey, there can be some predators over here, all right? Because they're coming in, they're dying at a certain rate, and it's just like I said before, if there was nothing to eat, you would still have I divided by M as the expected number of predators in a constant environment with constant input rates. So what happens is as you increase the rate of immigration, well, you push the predator isocline more to the left, okay? You're going to support more predators if you can make it so that there's so many predators spilling over that you eliminate the prey it needs. They're still showing up. There's still some predators there because they're still immigrating, and you can still it tends to be still stable, okay, if it starts stable with this system. But in contrast to the single species model we started with, and we say, well, what happens to predator numbers? Remember, we started with the model suggesting that as you increase the immigration from the outside of the species you're looking at with direct interdependence, its equilibrial numbers ought to go up. Well, what happens here can be seen by inspection of what happens to the intersection between the 2 isoclines. Well, because of this hump in the prey's isocline, predator immigration rate's going up, but it doesn't get reflected in the equilibrial abundance of the predator. So this shows predator immigration rate and predator numbers and prey numbers as a function of prey immigration rate. There may be tiny effects here, but you add a little bit of noise there, you wouldn't see any effect. Where the predator input, the spillover, shows up is in the lower trophic level. The numbers go down there. This is one of the kind of surprises you get in even simple community population models sometimes, okay? And remember, if you increase immigration sufficiently, you can cause extinction of the prey. And when when you have a system that's initially unstable, which is when the predator isocline is to the left of the peak of the prey's hump there, then increasing predator immigration tends to weaken the magnitude of the cycle and can even stabilize the system. But sometimes what happens is, before that happens, is you get these interesting changes in kind of the amplitude of the oscillations. And now let's go back to the same model and say that you're going to pulse, you're going to vary the immigration rate, you know, maybe seasonal variation in the spillover, or variation driven by, you know, rainfall that varies from one year to the next. And this is all very simple. We're assuming that there's sinusoidal variation in the rate of immigration. And this is a scenario where, with constant immigration, you would have an unstable cycle here, just shown here. Prey numbers go up, predator numbers go up, prey numbers crash, and the predators crash, like lynx hair, for instance. And you add now sinusoidal variation in immigration. It's like what's called a forced oscillator system in physics. And now you can get more extreme oscillations. The numbers are higher here than here and lower. And so the prey is going to actually be vulnerable to extinction. And notice that also with that sinusoidal variation, this is the same time scale. Is that right? I'm not sure exactly. Yeah, I think it's 500 generations. We now have 4 peaks rather than 8 peaks of the prey. So you change the period between the cycle and the amplitude of the cycles by having spillover that's itself temporally varying, superimposed on an intrinsically unstable system. And sometimes you can get complex dynamics. So here's an example where with constant immigration, you just get stable prey and predator numbers. You add sinusoidal variation, and you get kind of this inner loop followed by this outer loop. And so you get these big peaks, and then little peaks, and big peaks, and then little peaks, and so forth. You get a lot of interesting complex dynamics. So that was all when you have just recurrent sinusoidal variation. But often when you're doing experiments or where you're looking at short-term effects, this is something we've gotten really interested in ecology in the last several years, much of it due to Alan Hastings out at Davis. It's what are the transient responses of the system to single perturbations, okay? So if you've got an unstable predator-prey cycle, and then you're going to pump in a bunch of predators, okay, a single spillover event, how does that affect the dynamics over the short term? Does it matter when you hit it in the cycle? Okay, so what we've done, and like I said, this isn't a published paper, I did this for this presentation. We're going to imagine we can have, you know, a pulse of a spillover event, a pulse of predators that come into the system. They could all come in one instant, or you could spread them over a period of time, but you keep the integral, the total number of predators you introduce the same, okay? And it turns out that matters. So this shows what happens when you have an instantaneous pulse of predators, little tiny wedge versus a larger wedge versus fatter wedges, and fatter wedges. And it's hard to see, but the magnitude of the effects are smaller here. The y-axis is varied. And if you put in a pulse that's spread over a long period of time, there's just very minor changes in the system, okay? So the effect, the transient effects of, so here's another thing. So let's put in the same size pulse. So here we have a predator-prey cycle, but you can introduce it here with a lot of prey and a relatively modest number of predators. Over here where there are a lot of predators, few prey. Here where they're both low in numbers, and so forth. And it turns out that the dynamical effects you get in terms of the magnitude of the transient effect, how long it is before the next period arises, and so forth, all depend upon where you hit it in the cycle, okay? So I'm not going to go over this in gory detail. So this is something that I think really warrants a lot more exploration to say, okay, what are the transient effects of a single spillover event? Say in the biocontrol system, you got a farmer who's going to introduce a bunch of lady beetles. You could do it, all in a week. Is there some optimal time period when that should be done if you have kind of an unstable population cycle describing the basic interaction between the lady beetles and the target pest species in the system? So I'm going to talk about a few other kind of scenarios here. So the reason you get cycles and chaos in the discrete logistic and the Rickers because their density dependence is strong, but it acts with a time lag. And it turns out when you put those two things together in ecological models and multi-species models where you have species A affects B and then species B affects A, those are density dependence with a time lag from the point of view of species A, okay? So what you can find there is sometimes species become abundant, so abundant that predators overexploit their prey and then go extinct. That involves, so here's the classic example. Reindeer were introduced onto St. Matthews Island off of Alaska. They were in a fat city, food everywhere, but that food was growing very slowly because it's the Arctic. And they overate their food supply and crashed to extinction, okay? And intense consumption can lead to this kind of wipeout of the food supply. And I had an annual review article with some colleagues a few years ago where we pulled together examples of this. And what you can do, so that's like on the island is one thing. If you crash, there's no place to go and you just die out. But in an open landscape, if you're mobile, you can move, okay? So what you can find with intense consumption is kind of waves of consumption moving across space. So from the point of view of any point in this intertidal, there's a wave of starfish that's moved along. So a pulse of predators that came in, ate out its food supply, and then disappeared. And it turns out these kinds of feedback effects are actually very, very important. This is another interesting one. I'm a keen bird watcher. This is the bell miner, and it took me forever to see one out here in south of Sydney, and I finally found some. They've got that name because they have this ringing sound they make that you can hear for miles. It's like a bell in the forest. And they're colonial, and they're highly aggressive towards other birds, and they eat all these insects. And what are they doing? What are they protecting? Well, it turns out there are psyllids, which have a sugar coating, and that's what these miners eat. The psyllids build up because the birds are chasing off all the insectivorous birds. They would eat the psyllids, and they're eating the parasitic wasp and so forth. They might parasitize them. And over time, the psyllids build up to such a large number that they kill the eucalyptus trees that they're on. And then the birds have to move someplace else, and there's always a very low density of psyllids out there. They found another place where there's enough psyllids to get off the ground, and the process starts again. And this turns out to be a serious, the foresters complain about the graveyards of gum trees out there, this sort of very patchy landscape where there's this spatial dynamic because it depends upon psyllids getting around, bell miners getting around, and overexploitation of local resources. And so if you think about kind of a classic niche space where the purple area here is where little r is negative, here's the little r is positive, and this is high r in the middle. This is where you expect populations to be able to overshoot their carrying capacity, eat out their resources, and go extinct, okay? So here was the paper I did with Brian, Consumer Fronts, Global Change, Runaway Collapse. And we just put together a lot of examples and some simple theory related to this. This is Brian's own work in the coast of Georgia. This is a phenomenon very common in salt marshes around the U.S., where you can see there's this barren here, and here's salt marsh grasses, and if you go up to the edge of these grasses, you find they're just chock-a-block with snails. Okay, why are the snails so abundant? It's because people have over-harvested crabs. They were eating those snails. And so there's been a kind of release from predation, kind of a trophic cascade. And here's an experiment. Brian just put up a fence, and the snails have marched by. And there's a nice little patch of spartina grass inside that fence. So it clearly doesn't have to do with the external environment. It has to do with you've kept snail herbivory away, okay? Here's an example of periwinkles in the intertidal of New England going along, eating up these green algae, leaving bare soil, bare rock face behind that gets colonized by inedible brown algae. Here's a locust moving as a wave across an agricultural landscape. Isn't that amazing? And wildebeest do the same thing. There are a lot of examples. And so we talked, our paper was titled Consumer Fronts, but when I was looking at it, I said, my goodness, all those studies have to do with herbivores, knocking out plants, foundation species, in kind of monocultures almost of plants. Are there comparable phenomena out there for predators or parasitoids interacting their prey, or just to get washed out in the complexity of food webs because of intragill predation and all the other complexities that we've talked about today? Now, we did have some examples in infectious disease systems where you similarly have wave fronts that move along, and the infectious disease is eating up its resource, namely susceptible host, and it's able to persist because it can move along in space. Okay, so the conclusions for my talk, and I hope this has not gone on too long, Jordy. For single species dynamics, so much less complicated than the multi-species, multi-partite systems we heard about, is the basic conclusion: interplay of immigration and temporal variation can have diverse effects on population stability and abundance. In the simplest models, constant immigration is expected to boost equilibrial abundances. And the more immigrants you got, the higher your abundance ought to be. Okay, so this is when you have direct density dependence. Immigration can be stabilizing, but sometimes it can make things unstable. And temporal variation in immigration can interact with unstable dynamics in complex ways. All these ecological effects can be strongly altered if immigrants are genetically distinct and inferior in the local environment, i.e. if there's microscale local adaptation. So an increase in immigration, in the model that I was showing you at least, can even depress local abundances. And in sinks, populations that only persist because there's recurrent immigration, temporal variation in growth rates boost average abundance. And I've argued that if you couple that to spillback effects among other patches, that you can have a metapopulation of sinks that can persist, and that phenomena, I'm afraid, was probably at play in the pandemic that we did such a poor job of controlling in much of the world. So then if we go to predator-prey interactions, rather than where now the predator is indirectly experiencing density dependence via its impact upon prey abundances, as you increase predator immigration rates, you don't necessarily boost predator numbers. Instead, you depress prey numbers, and predator numbers may be relatively flat. Constant predation input can lead to prey extinction, and it tends to stabilize dynamics. But if the spillover of predators is pulsed, that can interact with intrinsically unstable dynamics to create more complex dynamical patterns that have yet to be fully explored, or even at all, really, in the literature. And then something that I think is important to think about, that's important if you're thinking about doing experiments where you may add a pulse of predators, is the transient effects of that pulse can depend upon the timing of that pulse relative to what the intrinsic dynamics of the system. And so some understanding of that intrinsic dynamics can be very valuable. And with that, I took this picture of a lemur when I was in Madagascar with my wife a few years ago. Thanks for your attention. Carlos, oh yeah, I see two hands. Carlos and then Dave, I guess.
Thank you, Bob. I think you fantastic talk. I mean, it's almost a theory of spatial feedbacks in meta communities. I have a couple of questions, but I think I'm going to focus in one, relatively maybe easy for you, but it's because I don't understand a few things. It's about the persistence.
About what?
The persistence of spillover effects in spatial landscapes.
Of the psyllid?
The persistence. So how, for example, I'm going to put you an example. So I understand that if you have spillover effects in many patches, and if the the population moving around has the same fitness or lower fitness than the local residents, then this is going to be essentially gone, right? It's going to go to extinction. So you had to have.
So we're talking about this system, right? Where I was talking about the metapopulation.
Yes, in fluctuating. Right. Yes.
So remember, it does depend upon there being asynchrony among patches. So there may be temporal variability.
Yes.
But in the examples I was giving you, there's temporal correlation locally, but there's no spatial correlation. We looked at in this paper what happens if you have some spatial correlation in, say, a landscape. So nearby patches are kind of tending to go up and down together. The effect is still there. It's weaker, but it still exists. Okay. But and also if you just have two patches and they're going up and down together, that just like the COVID example I can in that PNAS paper, that's not going to let you persist. But if you have just two patches and they're asynchronous and their growth rates, that's enough for you to persist. Okay.
So it's then about a fluctuating selection somehow.
It's not selection. So this is just ecology.
You don't have selection.
There's no variability. There's no variation here.
Okay.
Remember, I talked about variation a little bit.
Yes.
And that's one thing that could be done would be to say, okay, now what happens if you add evolutionary dynamics to this?
Yes.
And we've done some work on that. And I'm handling as the editor for a journal right now a paper where they've argued that for scenarios that are akin to but not identical to this, that if you have heterogeneities out there, that leads to kind of an overall increase in virulence, for instance, and infection. So there definitely are likely to be evolutionary consequences. But this is straight up ecology here because there's no genetic variability in question.
I got it.
Okay.
Yes. Yeah. Thank you, Bob.
Sure. David.
Thanks, Bob, for a fascinating wrap-up talk. To have a theoretical talk at the end of a conference with such good food and so many other talks, after 6 hours of sleep in the last 48 hours, I was able to follow almost all of it.
I'm surprised you're awake.
Yeah, I know. So I
could put you asleep even if you weren't jet.
Well, no, I don't know. So a question for you: you're making a strong argument that we really need to start looking at single species dynamics model.
Well, the thing is, in the back of your mind, that's where you start to understand that, and you build up to more complex systems. It doesn't mean you end with single species.
No, of course.
Right.
I understand that's a good argument. So, given that there's so many species in the systems we study, how would you advise an empiricist who's looking for a way to test some of these models, which species to study?
Well, first place, the protocol you took where you said let's lump a bunch of species into some functional guilds and assume they're all kind of the same and how they interact with some other functional guild, that's a perfectly reasonable place to start, okay? Then you might ask, the spillover is occurring where in the system? Is it consumers that are spilling over, or is it their resources? Like the example I gave with orb-weaving spiders in the Kansas experiment. It was clearly the prey that, the grasshoppers that were hopping into those webs from the surrounding matrix that was feeding those orb-weaving spiders, right? So that's where, you start with like a little food web, and you say, where are the errors from the outside? Where are they big errors and where are they little trivial? There's always got to be errors because in the real world, all local communities are transient, have all been assembled by colonization from some external source. So there has to be, over some time scale, some kind of input from external sources for any sensible, moderate area, right, that you call your local community. But maybe you can ignore that once the colonists are there. Where spillover is interesting is where there are dynamical consequences of recurrent spillover, or of single pulses of, a big spillover event that occurs that kind of washes through the system. And so, the thing is, if you're an empiricist, you're stuck with the messy complexity of reality. And you have to say which species are you most interested in, and if there may be some focal, just like biocontrol systems, there are a lot of species out there that are sort of irrelevant to the biocontrol of a particular agricultural pest. What you're really concerned with is what does it eat, and who eats it, right? There may be a lot of other things out there. Maybe there's some birds that occasionally eat the biocontrol pest problem, but you ignore them, right? So it's kind of a pragmatic, weasel out of it answer to your question.
Yeah, species of interest, and we also...
You're dying. It's probably because we're past closing time here. Yeah, Oscar.
Okay.
Oh, that sounds
okay. Well, thanks for your talk. It was awesome, I have to say. So imagine that I have to design an experiment, and I'm thinking to doing so, and I would like to know your opinion about that. All the theoretical models that you have shown, the species that immigrates or the dynamic of the species is relatively simple. Either because it's a discrete generations, everybody reproduce at the same time. But imagine that you have a more complex organism, just single one. And then it's not just immigration and the consequences for demographic population, is that the same individual can go at different patches during their life cycle.
Right, so in and out. I mean, that happened in the Kansas system, for instance. Maybe I'll go all the way back to that, so we at least have a picture of a real thing on the board rather than models or graphs or whatever. So in this system, that definitely happened. And I told you the cotton rats were found on the big islands, big patches, and they were sink populations here, but we had other rodents that were resident here. And there was one, and we really surprised us because I made these patches the size they are. These are 4 meters by 8, these are 50 by 100, so half hectares. These were deliberately chosen to be smaller than the known home ranges of the rodents in the system, because I was hoping we'd have a gradient in rodent herbivory or seed predation or whatever. But it turned out the voles didn't read the same papers I had read, and they were just fine. They and deer mice were just fine, and it turned out that what they do is that they live in the patches, they get cover there, but they feed in the matrix outside, and they use it as kind of a home base. Okay, and so they go back and forth regularly between the two habitats. So that's an example of what you're talking about, and that's a different kind of model then. You know, and I can't remember. I think it was you mentioned this paper I did with Tiffany Knight and others on the dragonflies and the fish and how fish can impact plant pollination. So they're, you know, organisms with complex life histories often occupy different habitats to complete their life histories, which means there's an inextricable coupling, but there's a feedback then. So, you know, what happens in the pond affects how many dragonflies are flying around outside. That affects the, you know, how much predation by dragonflies there are there. But those dragonflies have to ultimately end up laying eggs back in the pond. So there's this spillover and spillback that's built into the life cycle there. Right, so there are things like spillover and spillback on individuals that are foraging back and forth, doing different things at different times or different stages of their life history, I think. But that's an excellent question.
Thank you for your very interesting talk. You mentioned that that immigrant in certain situation can be maladapted.
Say that again.
The immigrant can be maladapted in certain situations, right?
Yes, that's right.
I wonder whether there is a magnitude of habitat differences.
Well, this is where this issue comes in. So let me get back to, let's see, where did I have that? I had that here. This is where you had a constant environment, and you have a one-way flow from a source into your focal habitat, okay? And you want to know basically what are the conditions for persistence of the locally adapted type. And if you have too many immigrants coming in that are maladapted, that are mating with the better adapted type, you wipe it out, okay? And then there are interesting consequences for population size. But you would get a different answer maybe if there was movement in and out of this habitat, because then fitness isn't just determined here, it's determined by what's happening here, what's happening over here, and how does the movement determine how you average over that variation in space, okay? Now, if you have too much movement, general theory suggests you ought to just select for generalists that are kind of reasonably good in both habitats, even though they're not the best, quote unquote, in either, okay? And so you don't maintain polymorphism if you have too much movement between the habitats.
And do you
so
think,
that's related to...
Do you think that phenotypic plasticity can play any role in that? For example, immigration or immigrants
yes.
could be more plastic?
I mean, what phenotypic plasticity does, it means one genotype can have multiple phenotypes. And so here you're assuming, there's some, we don't have the equations here, but there's some function that says this is density-dependent selection. So all these are functions of density, total population size, okay? And so if there were no immigration, whichever type can withstand the highest density wins out. It's sort of like the inverse of Tillman's R-star rule, for those of you who know that, right? So that's classic density-dependent selection. So you take that and then you just add an immigration to it, and you get these effects. But if you had a plastic genotype that could do exactly what 1-1 is doing in this environment, even though it's 2-2, it would wipe this out right away. Then you wouldn't have any genetic variability. You'd have phenotypic variability across space. Yeah, so phenotypic plasticity tends to degrade local adaptation, okay? I mean, you can still be adapted everywhere. It's just you don't have a genetic basis for that adaptation, okay?
Okay, thank you.
Okay, thank you, Bob. My question is going to be more philosophical, I guess, or it's not philosophical. It's just here in Spain we don't have theoretical background, or we don't have young theoretical ecologists. Just met one today. So what would you recommend to a person that starts and is interested in theoretical ecology, where to start, given all these simple models from complexity that we have?
I mean, there are two ends of that. So there are these simple models. I don't think you need a lot of complicated math to understand what's going on in these simple models. There can be some complicated math. Actually proving theorems and so forth could be very challenging. But you can do a simple computer simulation and convince yourself that the patterns I just talked about are robust. And then you talked about individual-based simulations, right? There's the other extreme where you write down a computer model that has a lot of biological reality, you let it run, and then an exercise that's not done as often as it should, it says, okay, now here's this interesting pattern I've got. Let me start stripping out the details in this model. Which ones are essential? Does it need the entire set of biological details to get the results? Or maybe you need a much simpler scenario, that there's something in there about age structure, stage structure, the allometric relationship you assumed for the nematodes or whatever, this kind of the prime driver of the pattern that you're looking for. That's an exercise that's not done as much as it should with people doing IBMs, but people who run IBMs, you don't have to be a mathematician to do that. You're running a lot of simulations, and these days with supercomputers, you can do thousands of replications and so forth. And to me, that's a very powerful approach, right? Like the food webs you were talking about, David. They're very complicated, right? And then you do an experiment. You add detritus, and you get a certain pattern. And you say, okay, now let's start lumping some species together, and trying to simplify it, right? And often the thing, the assumptions that are important are the ones that you make that you're not conscious of, right? So we were talking about this earlier, where I think we're talking about that relative to your results with IGP, where you increase productivity and all levels go up. Well, those simple models all assumed that there's pure prey dependence in the predator's functional response, and there was no direct interference among predators. If you change either of those assumptions, then you pump in more production at the bottom, and all the trophic levels rise in abundance, albeit to different degrees. But that's like an assumption that a lot of theoreticians make. They don't even think about. They just run with it, right? So that's how I would suggest it. It's not exactly philosophical. It's more like the art of how you think about complex systems. Is you retain the complexity, but you try to simplify them whenever you can. Okay. I hope that sort of answers your question.
Thank you very much. We are finishing this now. Okay, so now we are closing this date today. Eva is not here because I believe that she went to pick Pops back to the airport. So, but I wanted to thank her very much for helping with all the organization and everything that we did here. So. And thank you very much, everyone, for coming. And well, a few of us are going to continue talking about these things tomorrow. Everyone else that came, I hope that you enjoyed the meeting and that you learned something, or that you have some new insights that are going to be helpful in the rest of your career. Now, we are going to go have dinner. Yes.
Let's all thank you.
Okay. Thank you very much. Thank you, Christian, because he's been helping a lot, and this was unexpected. So. And of course, thank you for the people in the back, the IT crowd in the institute, and the people that came and recorded this. Okay, so we are going to have dinner. The speakers and Odin in La Peña al Taranto. For that, what we need to do is arranging who has cars and who hasn't, and so forth. Pablo Barranco, who's here, and he's going to Almeria, is willing to take some of us there, because we're expecting to have more cars, but one of our... Diego is back? Oh, okay. Okay. That's good. What else? I don't think that we have anything left, so we are going to meet here outside and just arrange for